He is a distinguished microbiologist and a dairy scientist recognized nationally and internationally for his pioneering work in the human microbiology, microbial biotechnology, and probiotics. Currently serving as a Dean, College of Dairy Science and Technology at Guru Angad Dev University, Veterinary and Animal Sciences at Ludhiana. Professor Punia earlier held the position of Principal Scientist at the ICAR. As a mentor, innovator, and a scientific leader, Professor Punia continues to inspire young researchers while contributing to the development of Indian dairy and microbial biotechnology sectors. His association with the AMI underscores the commitment to promote microbiological sciences and fostering collaboration among academia, industry, and research institutions. With this, I request the chair of this session to please join us on dais. Audience, can we have a huge applause for the chair for this plenary session, Professor A.K. Punia. It is our honor, sir, to have you at Dev Bhoomi Uttarakhand University. I welcome you once again.
Hello.
Thank you very much. Good morning, everyone present here. I just make minor correction because presently I am not at Dean Dairy Science and Technology, Ludhiana. It was almost 7 years that I completed that assignment, and presently I am Director, National Research Center on Camel in Bikaner, the only one institute of related to this camel in India and Asia. So we start with our session, though we are late, but still I see that majority of the audience or attendees or participants are not here, but we must obey and maintain the time. We are already half an hour late, and I would be happy to introduce Professor Yogendra Singh ji, a very well-known name in the field of microbiology and microbiology sciences in India and world over. Professor Yogendra Singh is a distinguished scientist and NASI senior scientist. He is currently serving as an advisor to Delhi School of Public Health at University of Delhi. He holds his PhD from University of Delhi, and his research career has focused extensively on bacterial pathogenesis and the molecular biology of infectious diseases, particularly tuberculosis, Mycobacterium tuberculosis, and you know about this strain Microbacterium tuberculosis, and of course tuberculosis, how this is causing havoc world over, and a lot many people are suffering from it, and almost more than a million of deaths they occur annually, and nearly 10 million new cases are there. The good thing is that nowadays there is a good recovery or good success rate of the treatment, up to 90%, and so Dr. Singh will be discussing all these things. He is a noted person for his work on bacterial protein toxins, studying their mechanism of action and host pathogen interaction. He has also investigated bacterial signaling molecules such as serine and threonine protein kinases, crucial for regulating the virulence and survival of pathogens. Besides also, he has he served at a lot of coveted positions and he is highly respected academic of, and is a J.C. Bose National Fellow and an elected fellow of the Indian National Science Academy, and it is really a very rare achievement for scientists, and it is dream for many scientists to be the fellow of Indian National Science Academy. Besides this, he is in fellow of Indian Academy of Sciences, and recognizing his significant contribution to the biological sciences, we invite him to the stage and deliver his lecture. And since thank you very much for the appreciation, but I would like, because he is such a personality, if you give him standing welcome, then I think it will be very good, for especially for students. Thank you, sir.
It is an honor to have you here, sir, at Dev Bhoomi Uttarakhand University.
Thank you. Please. Thank you very much. So now I invite Honorable Professor Yogendra Singh ji to deliver, and I would like that young students, because we are missing one presenter over here, speaker, so we have plenty of time. So please keep your questions ready, so that it is both way, and you can interrupt him in between also. You can ask whatever you have in your mind. So it will be, sir, kind of interactive session. That would be better.
Yes. So thank you very much, Dr. Punya. Good morning, everybody. It has been very nice for last, I think, 3 days now. So students are excellent. They are so disciplined. And in your uniform, you look so great. So thank you very much for all the help you people have given. Dr. Punya has already told about certain things. Thank you, Dr. Saab. Thank you very much for elaborate introduction. But before I start, I'd like to thank the organizers, especially Dr. Nabil and Dr. Nirjara, who worked in our lab, Dr. Ruplan in my lab when she was doing PhD. So it has been great to come here and discuss with you what we have been doing last 40 years or so. So we work on 2 diseases. One is called anthrax, which you may not have heard, but I am sure you have heard about biological warfare. अगर लोग न्यूज़पेपर पढ़ रहे होंगे तो अभी आपने देखा होगा कि गुजरात में एक टॉक्सिन बना रहे थे, रिसिन. I am not sure whether any of you have read or heard, but ricin is a very, very potent plant toxin. Similarly, जो बहुत सारे bacterial disease होते हैं, for example, cholera, tetanus, many of those diseases, anthrax, in those diseases actually the pathogen makes toxin. And that toxins actually eventually makes you sick, and finally it kills. Anthrax, I started working long back, almost 40 years back, when I was working for US Army in Washington. Then for about 8-9 years, I worked at National Institute of Health at Washington. And then I shifted to CSIR here in Delhi, where I started the work which I am going to talk today. So I'm not going to talk about anthrax, biological warfare. It can be used for killing large number of people because it is a bacillus, it makes spores. It can float in the air. If people inhale, they will be sick. But today, I am going to focus on tuberculosis. I hope this will move. Can I have the pointer also? Do we have? Or I have pointer here. Pointer hai kya? So anyway, let me, tuberculosis, believe me, it's a deadly disease. And, ये तो move का pointer है इसमें? अच्छा ये वाला है? तो move करने के लिए pointer, जो light वहाँ पे दिखाए जो मैं बोलना चाहता हूँ. अच्छा कोई बात नहीं. That's okay, no problem. So you can see that by the time I finish my lecture, almost 100 people will be dead. So you can imagine how deadly the disease is. This disease, actually, people get infected from the air. If there's a patient who is a tuberculosis patient, sneezes, coughs, so with the air, those small molecules come there, and once you inhale, they will go into your lung, and then you can get infected. You can get tuberculosis eventually. So infection can start by inhalation of a particle, aerosol you can say, which will have the bacteria, and eventually the lung can get infected. Now when it comes to the lung, then there are defense systems. We all have a robust defense system. That's why we all, most time we are okay, except for when we get sick. So now our defense system, especially the macrophages, they engulf. They're not, I mean, it's a very general, innate immune mechanism that when anything goes into your lung, those cells engulf, and eventually they destroy those molecules. It can be a pathogen, live virus, bacteria, or even particle, dust particle, or any other thing goes into lung. The macrophages actually destroy them. So TB goes there, macrophages take them, they try to destroy, but I will show you later that this damn bacteria is very strange, that it can modify our defense system. That's what I will show you in the later slide. You can see WHO in 23 report I have given that almost more than 1 crore people get infected every year, and 10% of that, which I have written on the top, that 100 people will be dead in next 30 minutes or so. It is sad that all the cases of the world, 25% cases are in this country. Our population, 25% population gets tuberculosis. Now tuberculosis has two forms. I told you about the lung, but from lung it can go to any other organ. Except nail and hair, it can travel to any other organ, and those organs can get infected. That is called extrapulmonary tuberculosis, or EPTB. EPTB is even more bad than lung TB, and I'll try to tell you why. Then comes the multi-drug resistance. I'm sure last 2-3 days you have heard about AMR, antimicrobial resistance. Many of these pathogens are becoming resistant to the antibiotic, and I believe that the time is coming when we cannot use the antibiotic because they are not going to cure us. Therefore, we need new antibiotic as well as vaccines. Since the disease is very deadly, the WHO made a strategy in 2015 that let's put every effort, and 3 pillars they have given: patient care, preventive treatment, HIV, all those things. Then they said government supported system, research, all these things they said we should do in such a way that every 5 years there should be a reduction in death as well as reduction into the infection. So WHO said 2035 end TB. But our Prime Minister is very enthusiastic, very dynamic, and he always sets higher goal. So his goal was that why we should follow WHO, rather we should try to eradicate TB even in much before that in 2025. But then comes the COVID in between. So COVID derails the process a little bit, but I can tell you one thing, that during COVID, actual TB cases reduced. And the reason was that people were, they were mainly staying in their home. Even patients were not allowed to go outside. That's why the cases got reduced during the COVID time. ऐसा नहीं है कि इंडिया ने कभी किसी चीज़ को eradicate नहीं किया है. अगर आप 15 साल पहले जाओगे, maybe very young people may not remember, but if you go 15 years before, big holdings were in all over India. ये लिखा होता था कि 2 बूंद जिंदगी के. And that advertisement was done by like Amitabh Bachchan and many celebrities. ये पोस्टर लगा होता था सारे शहर में कि this Sunday bring your child And that child used to get 2 drops of polio vaccine, and because of that vaccine, actually this country got polio eradicated in 2014, and you can see the celebration that 2014 में मार्च 27 को ये celebration था that India is polio free. We can be free of any disease, but what you need is willpower and of the means by which you can eradicate the germs. Only problem is that we don't have a very good tuberculosis vaccine, and I will show you why all those problems comes and why we do not have a good vaccine. Now I'm going to tell you that why this bacteria is so successful that it can kill so many people, it can cause infection in so many people and create so much problem into the population. So one problem is transmission. I will show you some of this in the later slides, that how transmission helps bacteria to cause disease. I told you about macrophages that in lung they are, every tissue they are there, but in lung when anything goes, these macrophages kill those pathogens. But this bacteria has ability to modulate the killing mechanism of the macrophages. That means the bacteria is single cell. You can imagine then single cells, bacteria, trillion cells human body, it can manipulate. So you can see the power, and I'm sure COVID even is smaller. COVID genome is only in KB, about 30 KB or so. So you can see this, even single small cell can be so dangerous. So anyway, this modulates our immune mechanism, and that mechanism is modulated because it wants to survive in macrophages. So macrophages are supposed to kill, but it modifies, no, you will not kill me, I'm your friend. And I will show you how it makes that friendly atmosphere within the macrophages. Very recently, but it has been published, soon people who are working in TB will see, that it not only causes disease and sickness, all those things, rather it takes over most of the metabolic pathway of our body. You know, they right from glycolysis to TCA to HMP pathway, many, by which we derive energy. So the TB patient loses weight, muscle mass, sick, so many things happen to the TB patient. So I will show you, and the one most dangerous problem is actually drug resistance. Because once people get infected with the drug-resistant tuberculosis, it is so difficult to treat because the normal drug do not work on those things. So I said transmission helps the bacteria to spread the disease in more and more people. Because TB bacteria has got oil on the surface, oil. And that oil, we call it lipid in the language of biology. That oil actually allows bacteria to remain viable in the air for longer time. So because it stays for longer time in the air, large number of people inhale, and eventually that causes the disease. People who actually study biology, then we call it, oh, lipids are very cool guys, they don't do anything. But lipid is actually very dangerous also. Lipid not only can give signaling, but they also can even modulate our immune mechanism. That's what TB bacteria has on the surface, that when it goes inside our lungs, it changes its cover, and that changing actually modulates the immune mechanism. So transmission means staying more in the air so that more people can inhale, large number of people can get infected. Human body has got very good, see, we are all machines, an excellent machine God has made. And our immune system is very, very robust, as I said in the beginning. That's why we remain fine most of the time. To do that, and people who study biology, they know that we have innate immune system, we have adaptive. There are so many things. Skin doesn't allow anything to go. Our gut flora, skin flora, we have microbes all over open body. Even our skin has got a lot of microbes. So all these things, the flora, then the macrophages, the phagocytic cells, all those help to keep us in good, good health, because they destroy all the dangerous things. Now, these immune system and all our system into the body runs by a signaling mechanism. And I'll give you a simple example, which many times I give in a class, that suppose you are sitting on a chair, you do not need much energy. But if you have to run, then you can run and more energy will be required. So body has to produce more energy. To do that, signaling goes into the cells that you have to create more energy so that the person can run. So these signaling molecules are one of the enzymes, this called actually kinases and phosphatases. I'll tell you what these are. So human, we have large number of 500 kinases. Similarly, these microbes also have these enzymes because they also need signaling for survival and causing the disease. Kinase is something where ATP actually is a molecule by which we get energy. वो कहते हैं ना कई बार कि ATP level down हो गया, अब नहीं कुछ कर पाएंगे. So kinase actually takes the phosphate group of the ATP and adds on a protein, and that allows the protein to change the structure and perform the way the body wants or the microbes want. Then the other enzyme is phosphatase, which removes that and brings it back to the normal. So there are one set of enzyme, kinase and phosphatase. One adds and modulates the activity. The other removes that phosphate and brings it to the normal state. These, as I said, these signaling molecule actually, they are the one by which all our metabolic pathway, our energy generation, protein formation, everything is controlled by those signaling molecules. So now, I'm going to tell you that how the macrophages kill the pathogen when it goes into our lung. So this is a pathogen, and it goes. So it is engulfed and a vesicle is formed. And that vesicle actually travels and eventually it gets degraded. So a pathogen when goes into the lung, these cells, these yellow color cells, they take it, they make a vesicle, and that vesicle travels and goes to a particular place where it is degraded. Now this path is actually defined. ऐसा नहीं है कि वेसिकल को छोड़ दिया गया कि तुम चलो, वो जानता नहीं है, उसको रास्ता दिखाना पड़ता है. So that trafficking is governed by many proteins which join from the beginning as soon as it is engulfed, and those signaling molecules take it to the end where the pathogen get actually finally degraded. But many pathogens, and especially the tuberculosis pathogen, TB पैथोजन ने हमारी सारी इम्यून सिस्टम को पढ़ लिया है कि हम कैसे अपने को प्रोटेक्ट करते हैं. उसने उसका काउंटर बना दिया है कि अच्छा तो तुम मुझको यहाँ ले जाओगे डिग्रेडेशन के लिए, बट मैं तुमको रास्ते में रोक दूंगा. तो व्हाट इट डज इट secretes enzyme, makes enzyme. Those enzyme actually inhibit this pathway. Eventually, the final step does not happen and it does not get killed actually. So I told you there are enzymes, kinases and phosphatases. They are the signaling molecule. So long back, we were trying to understand how the TB bacteria does, what that enzyme is. So we took many enzymes, but one enzyme I'm showing you. That enzyme is called tyrosine phosphatases. That tyrosine is the amino acid. Amino acid makes protein. So on tyrosine, if a phosphate joins, there is a phosphatase which can remove that. That's why it's called tyrosine phosphatases. So bacteria has got two tyrosine phosphatases, and they are secreted. So we knocked out these genes long back, and we found that if you knock out these genes, the infectivity of the TB goes down. That means these two proteins were helping the bacteria in causing the disease. When we knocked out the genes, what we noticed that it cannot infect very well, but it can survive very well in culture. If you grow on plate and culture, it can grow very well. That means these two enzymes were meant for us. The bacteria has acquired these two genes, these two proteins, so that it can modulate our immune system. Similarly, we knocked out another phosphatase. It's called, again, there are other amino acids also, serine amino acid, threonine amino acid, they are phosphorylated. So this enzyme also, phosphatase actually removes the phosphate group. And when we knocked out this serine threonine phosphatase, we found that the bacteria does not survive very well. Multi-septation means many septa happens. You can see the knockout strain, the septa has reduced first, here the first one, green color, and eventually the number of bacteria, matlab bacteria is not dividing, is not able to divide quickly, the septation, so uska capability goes down. If I remove this enzyme and the previous enzyme, then माइक्रोबैक्टीरिया जो टीबी करता है उसके लिए बड़ी प्रॉब्लम हो जाती है कि वो सर्वाइव करने में उसके लिए मुश्किल हो जाती है. Dormancy, many people, I'll just quickly explain you. तुम लोग कभी जो बच्चे खेलते हैं या हम बड़े भी, और कभी खेलते में कट लग जाएगा धूल उल से, तो ये कहा जाता है कि टिटनेस का वैक्सीन लगवा लो, क्योंकि टिटनेस एक ऐसा बैक्टीरिया है जो धूल में रह सकता है बहुत दिनों के लिए. वो सीड जैसा होगा, उसको बोलते हैं स्पोर. तो सम बैक्टीरिया मेक स्पोर, वही एंथ्रासिस भी स्पोर बनाता है जो मैंने शुरू में कहा, जो बायोलॉजिकल वार में यूज होता है. तो जब बैक्टीरिया को स्ट्रेस होता है, प्रॉब्लम होती है, तो वो कुछ बैक्टीरिया, नॉट ऑल, दैट टाइम दे कन्वर्ट देम इनटू स्पोर. स्पोर इज लाइक ए सीड विच रिमेन्स डॉर्मेंट, मतलब वो कुछ नहीं करेगा. बी जैसे आप एक सीड ले लो, सीड को बॉटल में रख दो, रखा रहेगा, तो दैट टाइम इट इज डॉर्मेंट, बट व्हेन इट गेट्स द राइट कंडीशन इट कैन जर्मिनेट एंड इट कैन कॉज डिजीज. नॉर्मली मोस्ट पैथोजन डू नॉट मेक सच स्ट्रक्चर, बट टीबी का बैक्टीरिया के अंदर ऐसी कैपेबिलिटी है दैट कैन ही बैक्टीरिया कैन स्टे इन आवर बॉडी इन अ डॉर्मेंट कंडीशन. Although few proteins were known that how dormancy starts, but 2 years back we found out, we noticed a protein जो कि which was binding to the DNA, DNA जो हमारे सेल के अंदर DNA होता है जिससे all proteins are made. So that DNA was compacted. So we believe that this compaction allows not many protein to get made. That is why the bacteria can become dormant. So this work we did about 2 years back, and that was towards the dormancy. And actually, purpose of the study is to understand that why this bacteria can survive in our body and even can fool our macrophages, those cells which are supposed to protect us. So far, I told you about the bacteria-related problem. Now I'll quickly tell you about the host-related problem. पहला तो I have already told you कि एयर से आता है. तो एयर से आता है तो कोई आसान कंट्रोल नहीं है. अगर वो हम स्प्रेड कर दें तो किलोमीटर्स में that can get spread. People who inhale can get sick. दूसरा होता है difficult diagnosis, delayed. ये क्यों होता है? And I will request you to understand this because TB is a problem of the society, people. होता क्या है कि आपको जब बुखार आएगा, खांसी आएगी, टीबी बैक्टीरिया से जो इन्फेक्ट होगा उसको भी यही होगा जो कफ कोल्ड में होता है. तो पेशेंट डॉक्टर के पास जाएगा, डॉक्टर दवाई दे देगा कि ये लो एंटीबायोटिक, ये पेन किलर और एंटी इन्फ्लेमेटरी, वो 3 दवाई दे देगा तो फिर आदमी 2, 4, 5, 10, 15 दिन उसको लगेगा कि हाँ ठीक हो रहा हूँ. बट अगर वो इन्फेक्शन टीबी का है तो दोज ड्रग्स आर नॉट गोइंग टू हेल्प, और एक्चुअली फिर उसको टीबी का स्पेशल टेस्ट कराना पड़ेगा. दूसरा है ड्रग कंप्लायंस. टीबी का दवाई ऐसा होता है कि व्हेन यू स्टार्ट, एक्चुअली दिस मेडिसिन वन हैज़ टू टेक मिनिमम 4 months और 6 months. और अगर एक्स्ट्रा पल्मोनरी, वैसे ब्रेन का टीबी है, आई का टीबी है, किडनी का टीबी है, उसमें तो बहुत प्रॉब्लम होती है. एंड मेनी टाइम्स पीपल हैव टू टेक ड्रग फॉर इवन 2 years. और मैं आपको बताऊंगा तो आप सोचोगे कि कितना पेनफुल होता होगा. अगर आई में टीबी है तो उसको जानने के लिए कि ये टीबी है, आई में नीडल डाल के सैंपल लेना पड़ता है. तो ऑल एक्स्ट्रापल्मोनरी ट्यूबरकुलोसिस डायग्नोसिस हैपेंस बाय टेकिंग द टिश्यू, देन सेक्शनिंग एंड स्टेनिंग इट. कि ग्रैनुलोमा, एक चीज़ बोलते हैं ग्रैनुलोमा, जहाँ पे टीबी बैक्टीरिया होता है, तो उसके लिए आपका टिश्यू लेना पड़ता है. तो मैं एक बोलूँगा कि अगर आप लोग, एक प्रोग्राम आया था सत्यमेव जयते, जो शायद बच्चे नहीं जानते होंगे, बट आमिर खान ने बनाया था, उसमें से उस सीरीज़ में 2 एपिसोड टीबी पे हैं. आप देखोगे कि दैट गर्ल who got infected by drug-resistant TB, how difficult was her life. So I will suggest that you should see it, because then you can protect yourself. When you go in the mall, in the movie market or crowded places, then you can protect yourself. So drug compliance में क्या होता है? When they start taking drug, 2 weeks में उनको लगता है हम बहुत ठीक हो गए, बस वो दवाई छोड़ देंगे कि अब तो हम ठीक हो गए. While doctor has said कि you have to take the drug for 6 months, but if they leave, फिर वो वापस आ जाता है TB, and when it comes back, that is even more difficult to treat. Drug resistance मैंने बोला, और I will suggest you watch Amir Khan's those episodes, you will cry कि ये ऐसा भी हो सकता है. यही मैंने बताया है कि डिलेड डायग्नोसिस इसलिए होता है कि सिम्टोमेटिक ट्रीटमेंट डॉक्टर करता है, आपको दवाई दे देगा, आपको ठीक लगेगा, और आसान नहीं है इसका डायग्नोसिस. एक्स्ट्रा पल्मोनरी मैंने बोला कि अगर ब्रेन में है तो यू कैन इमेजिन कि ब्रेन से सैंपल लेना है तो कैसे लेना है, नीडल डालना है ब्रेन के अंदर, सो इट इज़ नॉट ईजी. एक्स्ट्रा पल्मोनरी टीबी डायग्नोसिस इज़ वेरी वेरी डिफिकल्ट. ड्रग रेजिस्टेंट ट्रीटमेंट एक्सट्रीमली डिफिकल्ट. और अभी काफ़ी ड्रग के अगेंस्ट बैक्टीरिया हैज़ बिकम ऑलरेडी रेजिस्टेंट, so we need new drugs also. In addition to, I will like to tell that I think maybe in the next slide, but several hospitals, we collected the data and we found out that women are more susceptible to extrapulmonary tuberculosis. This is a slide which shows how the drug resistance happens. Many of you know कि ये बैक्टीरिया का सेल वॉल बहुत लिपिड है, जल्दी ड्रग इसके अंदर जाता नहीं है. Whatever goes also it can pump out. So there are channels by which it can pump out. Then it has the ability, it can metabolize the drug, so drug बचता ही नहीं है, खत्म कर देता है. Then it can also modify target. So even if you take drug, the drug is not going to work. So there are multiple mechanisms by which this bacteria can become drug resistant. I think one of the slides Dr. Rupal showed in first day, which we worked together, and last down that picture. But I taught actually in the MSc class of DU, the microbiome paper. So you can see your body is mostly microbes. कल मैं किसी से बोल रहा था कि अगर आज के बाद आप गलती करोगे कोई, अपने को ब्लेम नहीं करोगे. You will say our body is mostly microbe, so they are asking me to do this. So you can always blame the microbe because your body is mostly microbe. Can gut bacteria explain our mood? हमारा mood भी regulate कर सकते हैं, digestion, vitamin, पता नहीं बहुत. They can, I mean, you can imagine they can do multiple, so they are working for you. तुम्हारा gut bacteria जो है. But जिसको TB होता है, then it becomes very difficult because bacterial community get changed. जो ड्रग आप खाते हो शायद बहुत लोगों को मालूम ना हो कि उसमें से एक ड्रग का नाम है रिफेम्सिन. रिफेम्सिन एक्चुअली जब आप ग्राम नेगेटिव से इन्फेक्शन होता है, आपका पता लगा you want to go to washroom for very quickly and gases are made and you feel very uncomfortable. So many times actually rifamycin is given. But rifamycin is one of the best drug for tuberculosis. ये वाला पेपर है which Dr. Ruplan and I did, and many of our students were there. Maybe Nirjara was also in this paper, I'm not sure. But so ये मेरा एक पेपर बहुत, I think it will be published soon in Metabolomics and it's already in press. So what we found that TB bacteria also है. अभी मैंने शुरू में एक बात बताई थी और कि in addition to disease, it takes our metabolic pathways. मतलब वो हमारे metabolism को भी control करने लगता है. It can also modulate what bacteria will live in our gut and what not. And in addition, we are taking drug for 4 months, 6 months. So you can imagine eating antibiotic for 6 months, उस patient की क्या हालत होगी. तो हमने यही दिखाया है कि कैसे how the TB bacteria modulates our gut, the gut bacterial metabolism of the gut. And also we did metabolites recently, which as I said will be published soon, that many such amino acids are made which actually are more toxic. I think I have only 1 or 2 slides more. So elimination जो हमारे प्राइम मिनिस्टर ने हमको गोल दिया, आई एम श्योर वी all doctors, everybody wants to do it. But problem, scientists understand कि problem है क्या. And that problem is because it spreads from the air. So एक ही patient can infect large number of people. बैक्टीरिया के पास स्ट्रैटेजी है कि हमारी इम्यून सिस्टम को मॉड्यूलेट कर दे, डायग्नोसिस डिले हो जाती है, पेशेंट दवाई छोड़ देता है, ड्रग रेजिस्टेंस आ चुका है बैक्टीरिया में, एंड ऑन टॉप ऑफ दैट वी डू नॉट हैव गुड टीबी वैक्सीन. अगर हमारे पास जब तक वैक्सीन नहीं आएगी, आई डोंट थिंक वी कैन कंट्रोल द ट्यूबरकुलोसिस, एंड दीज पॉइंट्स एक्चुअली मेक द गोल अनअचीवेबल. इसीलिए प्रॉब्लम है, जब तक टीबी की वैक्सीन नहीं आएगी, और गवर्नमेंट हैज़ टू पुट, यू नो, वी हैव टू टेक डिफरेंट पाथ. वी मे नॉट एलिमिनेट टीबी, बट वी मस्ट ट्राई टू रिड्यूस इट. कम से कम हम केसेस को कम कर दें, वही बहुत बड़ी बात होगी, अगर हम एलिमिनेट नहीं कर सकते हैं. मैं फाइनली ये एक बात कहूँगा कि अगर आप में से जो मॉर्निंग हॉस्पिटल जाते हो, कहीं घूमने जाते हो, तो डोंट गो एम्प्टी स्टमक. यू ईट breakfast nicely, and I know when my students were going every day to the hospital, I used to make sure that they eat well, and also I used to give them vitamin in the lab. I said you take multivitamin, you take vitamin D, because these are required for your good health. तो मैं ये बोलूँगा, अगर आप शॉपिंग करने जा रहे हो, पिक्चर देखने जा रहे हो, ट्रेन में ट्रैवल कर रहे हो जहाँ भीड़ भाड़ है, तो डोंट गो एम्प्टी स्टमक. Also, many of us are deficient in vitamins, so please make sure you get that, because it helps in, you know, protecting from the disease. So we should try to reduce cases if we cannot eliminate it. And as I said, I had a very large number of students, more than 60 students did PhD and thousands worked in my lab. They all are doing so well. They are doing very, very well. And with this, I think I will thank you all for listening. Thank you, sir. Thank you very much. And I will be happy to answer any question if you have.
Thank you very much, sir. And also very many thanks that you could finish really well in time. Exactly 30 minutes were given to you and your lecture, and you finished in exactly 30 minutes. I would invite young generation if they have some questions, so we can take a couple of questions.
You can ask general question. You don't need to ask research related question.
And if you do not have your question, definitely I am going to ask you.
Students, if anyone has question, please raise your hand. The mic will come to you. Okay. Sir, we have one question from one of the speakers here.
So good morning, Professor Yogendra. It's very nice talk. I'm Dr. Syed from NCL Pune. So I want to know why the bedaquiline is not completely given access in Indian.
Is not?
Not given complete permission, retail permission for the bedaquiline, which is the.
Yeah. So I'm very happy with this question. The reason is that bedaquiline drug was developed, and in that team of the.
Sir, mic ke paas aa jaye aap.
In that team of Johnson & Johnson in Belgium, one of our students were there who did MSc in Delhi University and did PhD with me. His name is Anil Kaul, and he was director in IMTech, and eventually he went back and joined as a क्या बोलते हैं वो बड़ी company में big position. Anyway, he is a big shot, and he is in Belgium. Not director, they called it different name. Okay, so what bedaquiline does it inhibits the ATP synthesis. So when it was given to India in 2015, the company said that we will not give it to the drug store. It will be directly given by the hospitals. So it is given in the hospital, but it is little toxic also. But it is given only when people are having drug-resistant tuberculosis. So it helps in curing drug-resistant tuberculosis, given only in hospital, and it is little toxic. But when you are dying, I think you can compromise with little toxic drug also. I'm not sure whether I answered your question. Didn't answer your question? What is left?
Professor Saab, thank you very much for nice talk. once this pathogen lose the virulent gene, can it convert back to pathogen again? Because in our lab we got many mycobacterium which may not be virulent.
Okay, so when you grow in the lab, okay, हमने एक slide दिखाई थी कि bacteria can enter into the dormancy. Dormancy means it also has the ability कि shut down its gene or make it very slow. तो जब ये dormancy में जाता है तो it activates a transcription factor named as DAY-4. एक प्रोटीन मैंने भी दिखाई थी अभी HBHA. तो ये सब प्रोटीन क्या करती हैं कि DNA को कंपैक्ट कर देती हैं. तो those proteins which are required for cell division are not made that rapidly, so it does not divide quickly. वैसे ही वो 24 घंटे में डिवाइड करता है 20 आवर्स में. और इवन इफ इट गोज, सो इट कैन नॉट कन्वर्टेड मीन्स अगर आप उसको ग्रो कराओगे तो कुछ दिन के बाद वो वेरिलेंट हो जाएगा अगर वो S37 RV स्ट्रेन है वो. तो हम लोग वेल्लोर से CMC वेल्लोर से सैंपल लाते थे EPTB का. हम हॉस्पिटल में या क्लिनिक में देते थे ग्रो कराने के लिए. ग्रो होता ही नहीं था. उसका रीज़न ये था कि डॉर्मेंसी में चला जाता था और वो ग्रो नहीं होता था. बट सम ऑफ देम ग्रू, बट इफ यू कीप passaging and putting into new this thing, I am sure eventually they will be out of the dormancy. It may take time. It will take time because coming back from dormancy takes time. जैसे किसी को बहुत चोट लग जाए, बिस्तर पे है, उसको वापस दौड़ने के लिए थोड़ा उसकी रिकवरी चाहिए होगी. वैसे जब ये डॉर्मेंसी में जाता है तो शट डाउन एवरीथिंग, सो इट टेक्स टाइम टू कम बैक टू द विरुलेंट स्टेट.
So there is a question from the student side.
या चल पहले स्टूडेंट का क्वेश्चन ले लेते हैं बिकॉज दे आर नॉट मोस्टली फ्रॉम साइंस, सो आई एम हैप्पी टू इफ दे हैव क्वेश्चन. You can ask any question in biology, of course, including tuberculosis.
Good morning, sir. My name is Anjali and I'm a student of BSc Biotechnology. My question is that what is the role of tuberculosis in dormancy?
When आप किसी को पिटाई करोगे तो बचना चाहेगा. वैसे ही बैक्टीरिया के पास जब स्ट्रेस आता है, हमारी इम्यून सिस्टम कहता है कि आई विल किल यू, तो इट सेज ओके आई विल गो इनटू डॉर्मेंसी सो दैट यू कैन नॉट फाइंड मी. तो डॉर्मेंसी में कई बार ये इम्यून सेल से घिरा हुआ अंदर चुपचाप बैठा रहता है कि हमारा इम्यून सिस्टम उसको देख ही ना पाए. मैंने अभी लिपिड के बारे में बोला था कि जब ये इन्फेक्शन करता है तो अपने सरफेस को बदल देता है. वो बदलने का पर्पज़ ये होता है कि हमारा इम्यून सिस्टम उससे एक्टिवेट ना हो जाए. तो ये चोर की तरह आता है कि कोई जाग ना जाए, हमारा इम्यून सिस्टम जाग ना जाए और मेरे को जाग किल कर दे. सो आई एम ट्राइंग टू से जब बिगिनिंग में आता है तो इट ट्राइज टू कम एज ए थीफ, बट जब वो थोड़ा और न्यूट्रिशन पा लेता है सेल के अंदर तब उसको मॉड्यूलेट करने लगता है कि अच्छा तुम मारोगे, मैं तुम्हारा आई विल चेंज यू. सो इट हैज़ गॉट so many things, virulent factor you can say, by which it can survive and go into dormancy.
Thank you.
हाँ डॉक्टर साहब.
Good morning, sir. I am from MSc Biotechnology. Sir, my question is that कि जब ये ट्यूबरकुलोसिस होता है ये रेसिस्ट कैसे करता है? एयर में रेजिस्ट कैसे करता है?
बैक्टीरिया कैसे
as you said,
बैक्टीरिया कैसे रेजिस्ट करता है?
एयर में रेजिस्ट कैसे करता है?
एयर में तो एयर में क्या होता है, सारे जैसे कोविड था ना, तो जब कोई पेशेंट का स्नीजिंग कफिंग इससे क्या होता है कि वो वायरस या बैक्टीरिया एयर में आ जाता है. तो अगर वो सरफेस नॉर्मल सरफेस है जिसपे ज़्यादा ऑयल नहीं है, लिपिड नहीं है, तो ड्राई जल्दी हो जाएगा इन फ्यू मिनट्स. बट कोविड के भी ऊपर ऑयल था, इसीलिए काफ़ी लोगों को इन्फेक्ट करता था. और इस बैक्टीरिया के ऊपर तो बहुत, अब मैं जो लोग बायोलॉजी पढ़ते हैं, वेरी लॉन्ग चेन फैटी एसिड है, उसके ऊपर ऑयल लगा है. अब यू कैन इमेजिन कि अगर ऑयल लगा है तो एयर में ड्राई नहीं होगा, तो तैरता रहेगा एयर में. तो पीपल हु इनहेल दैट एयर, बैक्टीरिया भी साथ में चला जाएगा. अब अगर आपका इम्यूनिटी अच्छी है तो आप उसको किल कर दोगे, बच जाओगे। अगर आपकी इम्यूनिटी थोड़ी कंप्रोमाइज्ड है तो फिर ये सर्वाइव कर जाएगा, स्लोली ये डिजीज एस्टैब्लिश कर देगा।
ओके, थैंक यू सर।
गुड मॉर्निंग सर।
नहीं, इसके ऊपर आपने वो स्लाइड मैंने शुरू में दिखाई थी, जो लिपिड का लेंथ है 60-70 माइकालिक एसिड, एंड देर आर सो मेनी टाइप ऑफ लिपिड्स। जैसे हमने बोला कि हम लोग पढ़ते थे कि लिपिड बड़ा चुपचाप रहता है, कुछ नहीं करता है, बट ऐसा नहीं है। लिपिड इज ए वेरी डेंजरस मॉलिक्यूल। इन दिस केस it not only helps bacteria to survive longer, but also modulate our immune system. Yes.
Good morning, sir. Sir, it was a very informative session, but I have a query. In one of your slide, you have mentioned that BCG vaccine not effective, but it is the vaccine which everyone is giving to our kids for the tuberculosis. Then why we are giving this to our kids?
Actually, BCG vaccine आज से 70-80 साल हो गया जब इसका ट्रायल हुआ था।
Yes.
मद्रास में हुआ था इसका ट्रायल, चिंगलनूर या ऐसे नाम है उसका ट्रायल का जो स्टडी है। तो उसमें क्या देखा गया कि ये प्रोटेक्ट थोड़ा करता है, बचपन में थोड़ा करता है। एक्चुअली ये MTB का स्ट्रेन नहीं है, ये बोविस का स्ट्रेन है, जो जानवरों को होता है वैसे स्ट्रेन से आया है, और इसमें एक छोटा सा DNA का टुकड़ा निकाल दिया गया है। तो ये खुद पैथोजन नहीं है, बट ये बढ़िया से प्रोटेक्ट नहीं करता। अगर ये कर रहा होता तो इतने लोग बीमार क्यों होते। तो BCG वैक्सीन इज नॉट गुड। हमको मिलता है बचपन में स्पेशली कि छोटे बच्चे उनको प्रोटेक्शन मिल जाए। छोटे बच्चे ज़्यादातर घर में रहते हैं, इसलिए वो बीमार नहीं पड़ते हैं।
ओके सर, थैंक यू सर।
या हाँ डॉक्टर साहब। So diagnosis. Why?
How well we have progressed in terms of diagnosis of tuberculosis?
काफ़ी अच्छा है, हमने डिटेल में नहीं गए टाइम के कारण।
Okay. And secondly, what is the minimal load that we require that causes the pathogenicity of the bacterial cells, mycobacterial cells? What is the minimal load? 1 cell, 2 cell, 10 cell? How many cells can cause a disease?
How much cells? हाँ, अच्छा आते हैं दोनों क्वेश्चन पे। सो डायग्नोसिस के बहुत सारे मेथड हैं। जो एक मेथड है कि आप स्पुटम ले लो, जो स्पुटम निकलता है, स्लाइड बना लो, स्ट्रेन कर दो। जील नेल्सन स्ट्रेन का नाम है। तो माइक्रोस्कोपी में कलर्ड, बट होता क्या है कि अगर नंबर कम है बैक्टीरिया का तो वो दिखता नहीं है, क्योंकि इस स्लाइड पे बहुत डाय हो जाता है। तो जीन एक्सपर्ट एक मशीन आई है जो आज 10 साल हो गया। जीन एक्सपर्ट में सैंपल आप डालोगे तो वो आपका सीक्वेंसिंग कर देगा। तो आप ब्लास्ट करोगे जब इंटरनेट से डेटाबेस में, तो अगर टीबी का सीक्वेंस आ गया, बल्कि वो खाली यही नहीं बताता कि टीबी है, ये भी बताता है कि आपको ड्रग रेजिस्टेंट टीबी है। इसीलिए जो गवर्नमेंट ने रिकमेंड किया है कि ऑल हॉस्पिटल शुड यूज जीन एक्सपर्ट टू डिटरमिन कि वेदर इट इज़ ए ड्रग रेजिस्टेंट टीबी और नॉर्मल टीबी। एक्चुअली गवर्नमेंट बहुत कर रही है। Our government is very very good doing lot of things, but ये डिजीज़ ऐसा और वैक्सीन नहीं है, यही प्रॉब्लम है मेनली। आपका सेकंड क्वेश्चन था कि कितना सेल्स चाहिए। एक्चुअली सेल्स का नंबर जो होता है वो बैक्टीरिया के विरुलेंस पे डिपेंड करता है, और टीबी में भी ऐसे ऐसे स्ट्रेन है जिनका विरुलेंस थोड़ा कम है, बट देर आर बेजिंग स्ट्रेन बोलते हैं जो बहुत ही विरुलेंट है। एंड दिस इज ट्रू फॉर ऑल पैथोजन कि वो कितना विरुलेंट है उसके हिसाब से नंबर उसको चाहिए होता है। बट आई थिंक few thousand विल बी, जो एक्सपेरिमेंटल किए गए हैं वो जो बहुत लेस पैथोजेनिक होते हैं उनके several thousand चाहिए होते हैं, बट विच आर वेरी वेरी दिस थिंग दे रिक्वायर मच लेस। तो इट डिपेंड्स ऑन द लेवल ऑफ द विरुलेंस। नहीं पॉसिबल है। नहीं, एक्चुअली टीबी बैक्टीरिया के पास इट हैज़ गॉट मेनी विरुलेंस फैक्टर। सबसे बड़ी बात यह है कि वो ऐसी चीज़ें, उसने हमारे बॉडी को पढ़ लिया कि ये आदमी कैसे क्या कर रहा है, इसके वो इम्यून सिस्टम सब कुछ, उसके हिसाब से अपने को मॉड्यूलेट करके वो हमारे को बदलता रहता है कि अच्छा जब दिक्कत में होता है तो वो डॉर्मेंट हो जाएगा कि अच्छा मैं छुप जाता हूँ, जब तुम्हारा इम्यून सिस्टम वीक होगा तब मैं फिर से डिजीज़ करूँगा। तो आपके बॉडी में वो प्रेजेंट रहता है डॉर्मेंसी में। or when your immune system is down, it can again cause the disease.
Thank you, sir. Thank you very much. And you have given a very comprehensive talk on why it is difficult to eradicate TB. And in between, when you were interacting, when you were discussing, you have given your talk in very, very simple terms so that the diversity of audience could follow it. Very nice one. We appreciate it. You have also that human being is carrying a lot of microorganisms.
Yes,
and
yes.
There is earlier there was a conception that there is 1:10 ratio of a human cell versus number of microorganisms in the body. But now this has also been established and published in Nature by Israeli and Canadian scientists jointly in 2016 that there is a perfect ratio in a average adult is 1:1.3 यानी कि अगर आपके बॉडी में एक सेल है तो 1.3 माइक्रो ऑर्गेनिज्म आपके बॉडी में होंगे.
Exactly, you are telling the correct number.
Yes, so you never know whether you are a bunch of microorganism or you are a bunch of human cells. So we have to take care, and there is a big fighting between the good and bad bacteria and the body. And as you rightly said, when the number increase and dominates the bad bacteria, you are being considered as infected or diseased. And otherwise there are good bacteria also, they supports our life.
Yeah.
So thank you very much, sir.
Sure.
Then one question I will ask anybody, and it is fastest round first for students only, not to the experts. और कुहाड़ साहब भी आ चुके हैं, थोड़ा पीछे बैठे हुए हैं. वेलकम सर, वेलकम टू द होल एंड टू दिस
लेक्चर. नमस्कार डॉक्टर कुहाड़. गुड मॉर्निंग.
I could notice that he just came and he said. So स्टूडेंट से बहुत तेजी में एक सवाल है, ट्यूबरकुलोसिस को हिंदी में क्या बोलते हैं? I got you do not know.
हाँ ठीक बोल रहे हैं.
नहीं आपसे नहीं पूछा, आप तो एक्सपर्ट.
तपेदिक बोलते हैं.
देखो बच्चों को पता नहीं था कि इसको तपेदिक बोलते हैं.
आपको इसके बारे में जरूर पता.
और दूसरा सवाल में बहुत इस बार एक्सपर्ट भी इन्वॉल्व हैं और बहुत ही जनरल सवाल है. Why it is being denoted as TB, though it is known as tuberculosis? So absolutely and typically and dictionary के हिसाब से ये TC होना चाहिए था, but why it is TB? इस बार मैडम आप भी इन्वॉल्व हैं इसमें जी. एनीबडी?
It is tubercle bacillus.
किसने बताया? वेरी गुड, आई अप्रिशिएट. कौन है जो बताया? बिकॉज इनिशियली माइकोबैक्टीरियम. ओह वेरी गुड, वंडरफुल, आई अप्रिशिएट इट. लेकिन आपको आसपास की इस तरह की चीज़ों पे भी ध्यान देना है ताकि आप जो सुनते हैं चले आ रहे हैं और बस आपके माइंड में टीबी बैठ गया. लेकिन अगर कोई एक्सपर्ट आपसे पता करे तो आप सब लोगों के लिए है ये, जो इस लोग को नहीं जानते थे और मुझे लगा कि मैडम के अलावा कोई भी नहीं जानता था. ट्यूबरकल बैसिलाई था इसका नाम, तो उस टाइम ही इसका नाम शॉर्ट फॉर्म हो गया टीबी, एंड नाउ इट इज़ पॉपुलरली नोन एज टीबी, नॉट एज टीसी. थैंक यू. सो थैंक यू वेरी मच सर.
थैंक यू वेरी मच टू ऑल ऑफ यू.
हम लोग
थैंक यू.
Now I request the
डॉक्टर नबील एंड मिर्जरा. थैंक यू वेरी मच.
Thank you, sir.
सर तालियों से भी पता लग गया कि आपका लेक्चर कितना बढ़िया था. और आपने कल मुझे एक मैसेज दिया था कि टाइम का सर ध्यान रखना. मैंने समय से शुरू किया और समय से फिनिश.
Now I request Professor A.K. Punia, the President-elect AMI, to present a token of respect and gratitude to our plenary speaker, Professor Yogendra Singh. Audience, can we have a 100 applause? So this is the cap of our state, Uttarakhand. Thank you, sir. Now sir, I request you to please stay back. Now I request the Director CRC, Mr. Iqbal Singh Sahani, sir, to please share a token of respect and gratitude to our chair of this session, Professor A.K. Punia, the President-elect AMI. Thank you, sir. Thank you for being a chair for this plenary session. Now, we'll be having a session 11 of Microbial Biomanufacturing Make in India powered by Thermo Scientific in main pandal. Session 12, Microbiome-Genome Interplay would be at the Seminar Hall first floor main building. Session 13, Microbes for Transforming Waste to Wealth is in Ayurveda Seminar Hall after the high tea. I request everyone to please join us once again back at 11:45 sharp for the 11th session would be happening at main pandal, and the rest of the concerned people can join their concerned sessions where they have to be a part of it. And I request everyone to be here sharp by 11:45 for the next session. People can go for their high tea and they can enjoy high tea in their respective locations, which has already been decided. I request the students to kindly be seated as we are starting with the session in next 2 minutes. And the students kindly inform your friends to kindly reach back to the pandal as soon as possible. We are starting with the session soon. So good morning everyone, I welcome you back once again for the last day of this conference, and we are starting with the session 11 now. The session would be at Microbial Biomanufacturing, Make in India, powered by Thermo Scientific. The session on Microbial Biomanufacturing, Make in India, powered by Thermo Scientific focuses on showcasing the India growing strength in leveraging microbial technology for sustainable biomanufacturing. It highlights the role of microorganisms in producing bio-based products such as enzyme biofuels, bioplastics, pharmaceuticals, and agriculture inputs that supports green and circular economics. Experts from the academia, research institutions, and industry discuss innovations, scalability, and policy frameworks that promote indigenous manufacturing under the Make in India initiative. The session emphasizes how microbial manufacturing can drive self-reliance, reduce import dependency, and create high-value products through biotechnology-driven industrial transformation. To initiate this session, I would like to share the profile of our chair for this session, Professor Banwari Lal. Professor Banwari Lal is a seasoned Indian environmental and industrial biotechnologist, currently serving as a Senior Director of the Environmental and Industrial Biotechnology Division at the Energy and Resource Institute, New Delhi. He also holds a position of Managing Director at ONGC TD Biotech Limited, a public-private joint venture between Oil and Natural Gas Corporation, ONGC, and TERI. Dr. Lal holds a PhD in microbial biotechnology from the University of Rajasthan. His research spans nearly 3 decades in microbial biotechnology with a strong focus on petroleum and environmental sectors. He has been honored with several key awards, including the National Biosciences Award for Career Development 2004 by the Government of India for his contribution to the biosciences. With this, I would like to invite our co-chair, Professor Banwari Lal sir, on stage. I request you, sir, to please join us to join us. Audience, can we have a huge round of applause for our chair for this session? Thank you, sir, and I welcome you once again to Dev Bhoomi Uttarakhand University. Since we are starting with the session, so the first invited talk speaker will be our own Professor Banwari Lal, sir. I request you to kindly present your presentation with the people, sir.
Good morning, all of you. First of all, I would like to thank the organizer that they have invited me and given me opportunity to give the talk. So, I think most of students, they are learning, so I will try to make this talk in a form of story, in a simple form, so we can understand this. So, you know that, I think in morning session we discussed there are harmful microorganisms. They can cause a disease and very, very dangerous, very serious disease. But there are very large number of microbes, they are useful microbes. We can use them for different applications. So I think ancient history, if you see when civilization started, microbes were the only one they helped in the civilization. So there are many applications, the microbes we can use. But the problem is that how to domesticate these microbes. In scientific term, we call how to culture them, how to produce in bulk. So how to domesticate them, then we can't use them. Like animal, if we can't domesticate, we can't use. Tiger, we cannot domesticate, so we cannot use their energy. So microbes, only 5% of the known microbes can be domesticated. Remaining, we don't know how to domesticate them. Even in this room also, there may be so many types of microbes, we don't know how to domesticate them and how to use them. Or until unless we will not domesticate them, we will not be able to use them. So, I think the domestication is the important thing. Students, you people can, I think, thoda kam karenge kya? Yeah. So, how to domesticate them? As I told in this room also, there are many microbes, but how to domesticate and how to use them. So, many scientists, generally, whenever we take any student, we try to do that, how to culture the microbes in laboratory. And what we do, try to make the different medium and culture them. After culturing them, we do some test and throw in the wash basin in the lab. But we don't know how to use them, how to produce them in bulk and which type of use these microbes can be. So that is the biggest problem. The biggest problem is that we are not able to take these microbes from lab to the end use. So that is the biggest problem. So in my talk, I will take a story that how we have come out from lab to reach in the end user where we can make money. I think you know that how we can make money from these microbes. I can tell you my story. Earlier, we were struggling to get few lakhs projects, which were difficult to get. And we learned this technique that how we can do the business from these microbes. We are, what I will say that, we have generated 700 crore rupees from these microbes. And 150 crore rupees we have given to government as a tax from the revenue that we have generated from these microbes. So I think this is the problem, that how we can go. This is only a very small example. But if you do many such examples, then we will not depend on grant for small grant or this. We will not depend on infrastructure. Because now I was discussing with the student that they are doing, but practical is not properly here because equipment facility is not there. So for facility, until unless the student will not do the practical, they will not learn the technique. I told you how to make domestication. So once they will not do with their own hand, they will not able to do the things. So I think that is the biggest problem, and the government also tried various schemes, but somehow we are not able to take further this thing. So this is the problem that I am going to tell about my story. So As you know, in the 1990, there was a Gulf War. At that time, large number of oil spill. And this oil spill, we know it's a natural occurring organic compound which can be easily degraded. It's not a toxic, very toxic, and not very difficult to degrade. But it's a very highly problematic. Once it spoils the land will not fit for agriculture or for any other purpose. Oil spill, if you have seen in the sea or land. So for that, in the laboratory we were doing that, we take the oil and culture the microorganisms in laboratory. They were degrading oil very nicely, very clearly. Within few days, oil will disappear. If we feed them oil, they will degrade very quickly. Then we identify what are the pathway of this degradation, what are the enzymes involved. We did all these basic study. After that, we stopped. And we struggling here and there to get some fund to do some further work. And there are only few funding agencies. We are not able to get funds. So whenever we go for funding, the funding agency, they generally tell that you have to go the end user. So then we approached oil company because they are oil producing company. So we told them about this story, but they said these are things that we are listening for many years, but it will not work in the real situation. But we said, give us the chance. After taking lot of things, then they have given the small piece of land, which was a oil spill, farmer's land. And what this oil company they were doing, because oil wells are in the farmer's field, in Gujarat and Assam. So wherever oil spill, because pipeline is passing from oil well to a central place, the pipeline rupture and oil spill occur in the farmer's field and land will spoil. And they used to give money to farmer, and farmer will be happy that they will get money without doing anything. So this was the major problem. After that, many court cases against this oil company, because farmer will come job like next year again, they will not give money. So then it's a major problem for them. So then they gave us a small piece of this. So what happened that the microbe that we produce in the lab, that we tested in the lab, that they are degrading oil, the biggest problem, how to produce in bulk. That was the problem. And in produce in bulk in fermenter, how to make in the product form. Until unless we will not make in product form, nothing will happen. Because the product is not what you will sell. So we have to convert into a product form which is easy to take and then apply. So we did all these things because we are not getting any funding. So we were sort of a jobless, all students, and we were not getting anything to do research work. So doing all these things, we were able to convert into product form. And the product name is Oil Japper, that we gave the name. We obtained the patent. Now it's in science book, 12th science chapter. There is one chapter on this. So it's now become sort of a routine thing. And with this Oil Japper, we have generated large number of money. So earlier we were struggling to get 20 lakh, 30 lakh project. Even in one day we can get that much money. So that type of things that we happen. And so after that, they told other story that when the oil is coming from underground, the depth of oil well is around 1 kilometer, and this crude oil contains wax, candle wax. And in the oil reservoir, temperature is around 90 degree, 100 degree. So at high temperature, this waxy oil would be liquid form. But once it will come from underground to surface, the temperature will reduce, and this wax will deposit in oil well tubing. It's like a cholesterol deposition, okay, in our blood vessel. So for that, we got the microbe, they can degrade this wax very quickly. Wax degradation is not at all difficult. So we did all these things in laboratory, but again to convert into product, that is the problem. I think major problem here, we are not able to go ahead from lab scale to the product form, mass scale production. So I think after that session, the Bio-E3 policy is outcome of this type of thing only. So we converted into product form, and then we started using, and we have done this technology we applied in more than 300 oil wells, and we have generated hundreds of crores of rupees by this process. So the story is that how we can make money from the microbes which are not visible, as I told you. So then we did, because we had started working with the oil company, And this oil company ONGC, which is a big company, their annual profit is 60,000 crore. It's a very big company. So when they drill the oil well, they use one polymer known as xanthan gum. When they drill the well, so this polymer is produced by fermentation by a microorganism known as Xanthomonas campestris. It's a soil bacteria. This produces this polymer. So they told that they are getting this product from China, very costly, 500 rupees kilo. So they said, why can't you do this? So they gave us some research work and they said we have to do this. It's not simple fermentation, it's a difficult. So we developed this, we got this microorganism in a laboratory. Up to 1 year we were doing fermentation, but this polymer was not producing because that proper condition we are not able to maintain in the fermenter. Biomass used to produce but not polymer. So we were about to leave this project, but finally one day the student, they found that the culture broth becomes so thick like honey. Or in the fermenter stopped, the agitator stopped because of such viscosity. So we thought that that is good, but he did not reported all the condition. Next time when he did, he could not get. So after many these things, I said that you have to monitor each and every parameter and then repeat. So he did after repeated and finally we got and we produced this polymer and then tested that it's matching all the parameter and then produced and then tested in a drilling of 5 oil wells. It's giving good result. So now government, the oil company, they are saying you have to set up the mass scale production. So we are going to set up mass scale production, a big factory, large fermenter, 12 lakh liter capacity fermenter, in which we can produce 1 truckload of this polymer one day. So this is the story of this, that how we can learn this technique which we are doing every day in the laboratory to make usable. So that is the thing. So this xanthan gum is also known as Xi polymer. Xi means Xanthomonas campestris, the bacteria name, and it's a biopolymer, which is basically a polysaccharide, and it has a multiple application. It's used in food. Now its application in food is going more than the oil industry, because in oil, the drilling of oils is reduced. So in food, like in pizza, we were not knowing that. Then we inquired from market and we found that India is importing this xanthan gum, large number of quantity. And the cost is around 300 to 400 rupees a kilo of this. In soup, we use in pizza, toffees, lot of food application. So, and same product which used in drilling, same can use in food also. So, if you see in the plate, you can see this type of, it's very difficult. In soil bacteria, you will get this yellow color pigment. It's like a, जो मक्का का आटा होता है इस तरह का होता है, थोड़ा सा yellowish powder. And this bacteria, Xanthomonas campestris, is very common. Even in the outside lawn also we can get this. But only thing, the fermentation conditions are very difficult, very critical condition. If you change slightly, it will not produce. So that is the thing. So we produced this and we characterize this xanthan gum. So it's basically, it's a biopolymer in which mainly heteropolysaccharides, and it contains mannose, it contains gluconic acid and glucose. So these are the carbon units in this, and it's completely characterized. It's not new. In China, there are so many plants, big plant, 90% of global market, the China is producing around 90% of this product. So, very common, but here nobody has given any attention of this. So, and so it has many applications, as I mentioned, it used in food, is used in the oil industry, and cosmetics, in food also, in juice also it used as a thickener. And so initially we did in the lab scale, and the yield is also not much, around 30 gram per liter, 3% of the broth. And these are the characteristics. But for oil application, there are around 15 parameters, and these parameters are identified, set by the US EPA. So it has to be match all the parameters, then only oil company will able to use this. So we were not able to meet all the parameters. Then we did literature survey and found that if we add some additives, generally these additives are the patent, generally secret. If you add this, then it will meet all the parameters. So we did this and mix with the different additive and make a product which will fit for drilling purpose. So this is the process flow chart. That's very simple. You grow in the fermenter, remove the cell biomass, extract this polymer by solvent. After that, you do the air dry it, and then make it powder, pulverize it, make powder, and then you can mix different additives to meet the drilling grade quality. And how much quantity has to added, that is the patent, that how much it has to be added. That is the secret. That is the only secret. Otherwise, everything is in the public domain. So this is in the fermenter, it will become very thick, like very difficult, it's like a honey, thicker than honey. But the viscosity is pseudo, not like honey. Then we produce in bulk. The advantage here is that we have industrial scale fermenter, which generally many other people don't have. So that is the benefit. And this facility also we have created from the revenue that we have generated from this, not by government funding or anything. That we have generated revenue from this, and we have created this facility. And now the plant that we are going to set up is a 100 crore plant. So this is a 15,000 liter capacity fermenter, 13,000 working volume, in which we can produce this product. And then tested in the ONGC, here the laboratory IDT, India Institute of Drilling Technology, ONGC, Dehradun. So that is the nodal agency that they will test this. Once it will pass, there are around 14 parameters. Once it will pass, then only they will be able to use this. So we tested and all the parameters were passed, and then tested for drilling. And this is the application. Now you can see the red color. The demand of this product in food is increasing. And the current market of this product is, last year it was around 500 million US dollar. And in 26 it will go up to 600, 700 million US dollar. So it's a very high demand product. And so the ONGC, they are importing annually around 1,500 tons per year. So we can fulfill their demand once we'll set up this plant. And other oil companies in India also producing. So therefore we are going to set up the production facility of around 3,000 tons per year. So this is the drilling of oil well. I think other scientists will not get the opportunity where it has to be tested. So because we are with the oil company, so they allowed. Otherwise something will go wrong. Around 20 crore rupees will go waste in drilling. If well is failed in drilling, all the money will go waste. So we took this, the oil company, they have given us the opportunity to test this, and we found that, and they give the letter that this product is clear, all the parameter, and good for drilling purpose, and they will able to use this product. So I think this is the story that how we can make a product from the microorganism through fermentation. Fermentation is very common. I think घर में हम लोग रोज दही बनाते हैं, that is also fermentation. So fermentation से how we can make the product which we can use. So I think for young students, why we are studying? We are studying to get the good jobs, good service, wherever we get good opportunity, but there are no jobs because generally in biotech, there are no pharma, there is no industry, so where the student will get job? Therefore, student has to think on this line that how we can do the startup concept, that how we can do. The only problem is that how to come out from lab to the field. That is the biggest problem. So I think that is the important thing, and that we have come out from this. And the company that the ONGC Terry Biotech, it's around 19-year-old company. And we started this company without taking any money from anyone. We started generating revenue, and this company has grown up. And annual turnover company is around 45 to 50 crore rupees. Profit is around 20 crore rupees. 20-25 crore rupees. So every month this company is given 1 crore rupees as a tax to government. So I think then only we can see the transformation in country, in scientific. If we will come out with this type of approach, then student will get jobs. We will not depend on anyone to give the fund for research work. And so this would be a sort of a sustainable model which can be replicated large numbers. So I think that it's the talk that I have given.
Thank you, sir, for an interesting session. Now I request the Director CRC, Mr. Iqbal Singh Sahani, to present a token of respect and gratitude to our speaker, Professor Banwari Lal.
If anyone has any clarification, they can ask.
If anyone has questions from the audience, you are free to ask the questions.
Yes, sir.
Okay, so we have a question. Okay.
So, good afternoon, sir. Sir, I am a student of MSc Biotech. Sir, my question is how this product is useful in pharmaceutical industry.
In pharmaceutical industry, in many formulation it used. You know, in many formulations, this polymer is used to make the drugs. So like food, it is used as a good thickener. Similarly, in pharmaceutical industry, in oil industry, it has a multiple applications. So,
yeah, I was asking, in fermenter, are you able to reduce the cost to meet the China competition?
yeah, cost, it's a highly energy intensive because fermentation time is around 40-50 hours. So, fermentation is not a big problem. Big problem is the downstream process.
Cost, agar total cost calculate.
Production cost would be around 150 rupees per kilo.
Oh, that's great.
Market range is 300 rupees to 400 rupees per kg.
Yeah.
Total cost I am talking.
And you can produce as much as you want.
Yeah. So this is a very simple thing, but in India, the problem here in country that investors are not coming. Because they are not able to take any risk, how they will market. The biggest problem is marketing of any product. For us, the advantage is that because our partner is the end user of this product, so we are not going to waste time in marketing. So marketing is the biggest problem. Many scientists, they have developed some product, but they are not able to market this. So that is the biggest challenge, that how to market. Therefore, we have the benefit that this our partner, they were consumer of this product. Not only this, all other product, and because of that only we were able to generate that much money.
Thank you, Professor Banwari Lal ji. I think nobody better than you can give the idea how to really take these microbes to the end users. And whole AMI and academy is proud of you. But I would like to, I'm sure you can guess what I'm going to ask you. How AMI or via AMI or academy, we can take your help so that we can initiate a startup, or maybe some students via this platform can reach you to develop a startup. What are the possibilities?
Yes,
first part of the question.
So
Second part of the questions, you are a millionaire. I think you should donate 1 or 2 crores to, say, academy so that this Academy of Microbiological Sciences could be in a position to make a mark as far as Academy's contribution in developing the microbiological sciences of this country?
So I think how we can replicate this model. So that is the challenge, that how we can replicate. The problem is that generally the scientists also, they don't do the complete study. They do something and they claim that they have done very big things. Once they approach industry people, they ask the real results. In the real condition, your product will work or not. Therefore, they do not have faith on the scientists. Similarly, scientists, they think that they will cheat us. The user industry will cheat us. So there is no good synergy. Although many attempts were done, but there is no good synergy. So I think how to bridge this gap, that is the biggest problem. Why I was able to bridge this gap? Because I was lucky that we got the partner who was the consumer of this product, plus they were also able to invest whatever investment is required. So I think that was the biggest advantage, but I have not taken a single rupee from anyone. We generated our own revenue and invested on
That's very true. Say for example, AMI takes an initiative to spend 20 lakhs or 30 lakhs.
So I can give you, sir, example. In pesticide, you know, pesticide application cannot be avoided. Without pesticide, the food production cannot be done. Like in cabbage and leafy vegetables, so much pesticide is used. I think now people are saying in Delhi or here, every fifth person is dying with the cancer because so much. This earlier gut cancer was not much cases in country, but now many people are having this disease because of this pesticide. And in the NCR region, so much pesticide. If you go in the farmland, you can see. And pesticides generally, they are not water soluble. So once we bring the vegetable, we wash with the water, pesticide will not go up. But there are hydrolytic enzymes. Until unless this pesticide will not hydrolyze, it will not come out from the leafy vegetables or cabbage. So we have to make this hydrolytic enzyme in the product form. Like ये विम का एक जो आता है ड्रॉप बर्तन साफ करने वाला, एक ड्रॉप डालो, उसमें डालो, अपने बर्तन क्लीन हो जाते हैं. Similarly, you make your enzyme formulation in such a form, easy to use this. Then you can penetrate in market. The demand will go up very largely. That is a small example that I gave you. So until unless otherwise we are doing, oh, I have this much enzyme activities, this much, and it can do this, but until unless we not convert into product form, we will not convert into product form, simple, which has a simple application, then similarly, soil is a gold mine for microbes. You know, I think, which we are not, 1 kilogram soil will contain more microbes than total human population on earth. So we can see how much microbes, I think for students, if you see, but these microbes, until unless you can't domesticate, you can't use. So you have to learn that how to domesticate them, produce them in the lab, and convert into product. You target the product. Until unless you will not convert into product, whether the product may be small quantity, but it has to be, then industry people will come to invest in this, replicate this model. So I think here in next session we may discuss this type of thing. Otherwise, the problem is that no research grant where the students love, they are doing masters, they are spending so much money, their parents are expecting that their kids will get good job, or where they will get jobs.
मैं तो ये पूछना चाह रहा हूँ डॉक्टर साहब.
दूसरा आपने बोला कुछ डोनेट करो. सर, दिस इज नॉट माइन. आई एम like these two organizations. Why I have done? Because I was struggling here and there to get the project. DBT you also know, sir. दिन भर वहाँ पे वेट करते हैं, जब नंबर आएगा आप प्रेजेंट करोगे, आपने 50 लाख का प्रोजेक्ट लिखा, they will give 10 lakh rupees. So it's like a beggars cannot be choosers. अब आप 50 लाख मांगा और they will give 10 lakh, what you will do in that 10 lakh? So we will also write report and that's the finish. So I think that is the situation now also. Scientists are really what I will say, gold mines, but they are not, we are stuck. But in China, the different model. In China, each good scientist is linked with the industry. All particular industry they adopt at least 5 or 6 scientists in their particular field. And they are interacting with them, they are giving revenue also, good understanding them. So industry will get benefit, the scientists will also get benefit, student will get a job, they will get jobs. So this model, I think we also need the China model where each academic professor or scientist is attached with the industry people. But we are saying industry people should come in our university and they should say nobody will come. We have to go there, understand. But problem is that right now there is no good, what I will say, no faith among scientists and industry people. So generally they don't entertain because they will say that if I will tell something him, वो अपनी secret बता देगा और अपने competitor को बता देगा, then so they also don't entertain scientists. Industry people also don't entertain scientists. So I think उसको break जब तक नहीं करेंगे, ये startup model हुआ है, good. But again the success rate is very less. Why success rate is very less? Because many startups, they think there is a big demand of this. Like I gave any product, enzyme, like I said hydrolytic enzyme, which is very common. अब वो आधी अधूरी बात सुन लिया और वो स्टार्टअप कर लिया, उसको पता ही नहीं प्रोडक्ट कैसे बनाना है, हाउ टू मेक इनटू प्रोडक्ट फॉर्म. वो स्टार्टअप तो बना लिया, पर उसके पास कहाँ बनाएगा?
वही मैं पूछना चाह रहा था कि you as a Professor Banwari Lal, how can you help academy to move forward, or through academy, say maybe 5 students they are interested, how you can help so that at least they learn the integrate, say the concept and kind of challenges. If they come out with a kind of an idea or maybe with some kind of the product, but to succeed in initiating and developing that startup based, say, company is also not that easy, that we know. But you have pretty good experience. How we can make use of your services or your participation could help?
So उसके लिए सर यही होगा कि we should identify the product which are ready to market. Because अभी तक if we are so many scientists, आप बताइए किसके पास ready product है जो, यदि उनके पास कोई product है, which I gave you example of this pesticide.
नहीं, मान लिया कि हम ऐसे आदमी को भेजते हैं, उसको आप कितना टाइम स्पेयर कर देंगे? You'll be
आपको मैं बताऊँ याकूट, अब तुम बच्चों को सबको पता होगा याकूट एक आता है, वो कुछ भी नहीं है, वो माइक्रोब्स आपको हम लोग लैब में रोज उसको ग्रो करते हैं, फेंकते हैं, लेकिन उसने एक प्रोडक्ट फॉर्म में बनाया, एक बोतल में मिल्क में वो थोड़ा डाल के देता है और उसमें फ्लेवर डाल दिया थोड़ी, जिससे कि बच्चे उसको लाइक करें. उसका मार्केट देखो कितना है, सिंपल, एक सिंपल फर्मेंटेशन कर रहा है, उसका प्रोडक्शन कॉस्ट कुछ भी नहीं है. एक एग्जांपल बताया मैंने, एक वो करके, अब उनको ये भी नहीं पता कि वो जो हमने जो याकूट ने बढ़ाया वो हमारे गट में रहता भी है या मर जाता है, बिकॉज ये भी कोई स्टडी नहीं है, लेकिन उसने उसके नाम से, बिकॉज उसका टेस्ट उसने क्रिएट कर दिया, उसमें डाल करके ये फ्लेवर डाल दिया, फ्रूट का डाल दिया, स्ट्रॉबेरी का डाल दिया, कुछ डाल दिया, सो द किड्स विल लाइक इट. बट उसका एक्चुअल बेनिफिट कितना हो रहा है, दे आल्सो डू नॉट हैव एनी आइडिया. बट हम उसका बता सकते, बिकॉज उसने कंप्लीट प्रोटोकॉल नहीं बनाया. सपोज आई वांट टू चेंज माय गट माइक्रोफ्लोरा, हाउ टू चेंज? इफ आई टेक एवरी डे गुड माइक्रोब्स, यू नॉट एबल टू मेक गुड माइक्रोब गट, बिकॉज माय एक्जिस्टिंग माइक्रोब विल नॉट गो अवे. वो अपना घर छोड़ के कैसे जाएगा? तो उसको पहले उसको रिमूव करना होगा, आइदर ए स्मॉल डोज ऑफ एंटीबायोटिक, किल देम एंड देन यू टेक, देन यू कैन रिप्लेस देम. सो दीज आर द थिंग्स. तो उसका एक्चुअल बेनिफिट जब आने लगेगा, देन मार्केट इंक्रीज होगा. अदरवाइज एवरीबॉडी इज सेइंग कि हम लोग गट माइक्रोब रोज, हम अच्छे माइक्रोब रोज पिएंगे, फिर भी जरूरत नहीं है हमारा गट का माइक्रोब चेंज हो जाए, बिकॉज आवर एग्जिस्टिंग माइक्रोब दे आर सो अडेप्टेड, दे आर वेरी डिफिकल्ट टू गो अवे. सो ये कंप्लीट प्रोटोकॉल ये डेवलप करेंगे तो उसका डिमांड होगा. ये याकूट का इट्स सो गुड एग्जाम्पल स्टार्टअप का, जैसे स्टूडेंट का आएगा तो उसने कितना अच्छा बनाया, उसका कितना $multi-million बिजनेस. मतलब चाइना नहीं, जापान से होके इंडिया का मार्केट कैप्चर कर लिया. और हम लोग माइक्रोबायोलॉजिस्ट
Thank you, sir. Now I request the Director CRC to present a token of respect and gratitude to our speaker, Professor Banwari Lal, sir. Audience, can we have a huge applause for our speaker, Professor Banwari Lal, sir. As we have the invited talks after this, so the respective time which is given for the invited talk would be 15 minutes. The warning bell will be at 13 minutes. The power talks will be for 12 minutes and the warning bell will be at 10 minutes. Industry talk for 10 minutes and the warning bell at 8 minutes. I request the concerned speakers to kindly concern this thing and maintain that we should be complete during the time. And this session would be industry talk, will be powered by Thermo Scientific. Dr. Arun Kumar will be joining for the same. So over to my chair for the further taking this session.
Next talk is Professor Ramachandran, our friend Ramachandran. So please come and. The title of his talk is a very interesting title, Integration of Ferrous Oxide and Copper Oxide Nanoparticles with Phytoremediation as an Innovative Technology for Eco-Restoration and Metagenomic Analysis of Distillery Waste Polluted Sites. Very interesting topic. Complete your talk in 10 minutes.
Yeah. Forward. Okay, okay. Okay. On कर दिया? Okay. Good morning to all. Chairperson, dear students, respected audience. Actually, ये क्या खोला है? I am glad to be in front of you and thankful to the organizer put my talk here. इसको ऑन करो. ये खोलो तो मेरा. भाई मेरा टॉक आ जाने दो ना. नहीं, नेक्स्ट करो. क्या लोड कर दिया? आपने दूसरा टॉक लोड कर दिया. उसमें मेरे नाम का खोलो. रामचंद्र, बस इनलार्ज करो. इनलार्ज करो. इनलार्ज करो. Yeah, sorry for inconvenience. I will be talking on the actually some aspect of nanobioremediation, which is very fascinating word nowadays in area of microbiology and biotechnology. Ferrous oxide and copper oxide is two actually common nanoparticles which are helpful for the bacteria as well as plant for the bioremediation. And I have chosen distillery waste and pulp paper mill waste, which is a very important known environmental pollutant. If we will see the process of the bioremediation, there are three components: environment, contaminant and microbes. And they play a role in the process of bioremediation in very coordinated manner. The principle of bioremediation, if you will see, there will be 4-5 sequential process which is very essential to start the bioremediation: solubility, availability, interaction, metabolization, energy and biosynthesis. All these processes actually occurs, then only there is a bioremediation or multiplication of the any organism in the process, anywhere. In the nature, there are the two group of the pollutant, organic and inorganic pollutant, and the root of organic and inorganic in the microbial cell, you can see through the photographs, they come in contact of the cell wall or the cell membrane, then they enter through receptor inside the cytoplasm. And there is also further point or receptor protein, they direct where the pollutant has to go. Pollutant has to be accepted or pollutant has to kept at a corner. That is also a very unique process of the microbes and plant. This is further regarding the entry of the pollutant. The pollutant may come in form of the nanoparticle, and this nanoparticle, which is nowadays very well known as nanotechnology also. If you will see the material, their atom at the lower level or molecular level, they have different form. So their molecular structure, their mode of action, their binding property depend upon the nature of the matter. They may be in different form. They may be spherical, polymeric, and they be tubular anyway. So the property of the nanoparticle has some basic property. On this principle they work. The synthesis of the nanoparticle, most of the biotechnology people, chemistry people, they do. And there is a physical, chemical and biological method, different method. And these are the two well-known methods for the construction or the preparation of the nanoparticle. If you will see the use of the nanoparticle, there are multiple use, including the drug delivery, and the bioremediation is our actually directly environmental aspect is very well known. If you will see this picture, you can see the interaction of nanoparticle with the pollutant has a different property or the parameter how they can work. Interaction between nanoparticle, microbes and contaminant, you can see different parameters which actually regulate the process. All the process is very fixed, that not arbitrary, and that's why they can work. This step is very important, how they enter inside the cell. That is important. They can enter through membrane poration, passive diffusion. Endocytosis actually occurs only in case of the eukaryotic cell. Receptor mediated is also very common, and biofilm penetration is very common. Electrostatic interaction is very common in case of the microbes. This cell membrane actually play very important, semi-permeable membrane we know. So regulatory function of the all particle is regulated through membrane only. I have chosen this actually pollutant from the pulp paper industry. This is view is showing after the treatment of the pollutant in the industry itself. So industry actually produces, pulp paper industry, huge amount of the polluted water after the paper manufacturing. Actually, there was a lot of query earlier also, and still industry is facing. Even after treatment, the pollutant which is coming in environment, this contain more than 50% EDC compound. So it is a major source of the aquatic pollution, and bacteria even after treatment of the industry treatment facility, they cannot break further the compound existing. So some silent bacteria exist inside, they are known as autochthonous bacteria. If they get the optimum condition or nutrient, they can grow. So through biostimulation process, I try to reveal them, what type of microbes are present there and they are potential. So because for the lignin degradation, we did separate study also. But when we add our isolate and they can compete with the indigenous bacteria and they can inhibit them. So our targeted performance level was not coming. Then we observed that during the biostimulation process, this bacteria was also doing proper if we provide them optimum nutrient condition. These are the lignolytic enzyme. The lignolytic enzyme actually manganese peroxidase, lacase and lignin peroxidase. They actually work in the contaminated place in coordinated manner, not one single enzyme work in the mixed pollutant or industrial waste. We try to see the bacteria, how they are growing. In the upper one, actually they are showing the bacterial growth after the nanoparticle addition. In the lower part, there is a different category of the bacteria. And all this enzyme stimulation was monitored. And this picture is showing the sequential enhancement or induction of the detoxifying enzyme. So all the parameter and product were monitored. In this table you can see some metabolites were degraded at the end and some recalcitrant or the metabolites were regenerated. So these 2-3 bacteria, Klebsiella, Enterobacter, Acinetobacter were found very potential. In case of the pulp paper waste, actually Klebsiella will be most of the places available. Even also in case of the wood, you will get Klebsiella as endophyte also. When we use the actually these nanoparticle, we found that these nanoparticle actually enhance the enzyme production. So this manganese peroxidase and lacase production was seen, and in presence of the copper oxide and ferric oxide, how the manganese production was enhanced. So all the parameter was actually tested, and we observed the toxicity reduction by seed germination test. And these pollutants are also actually mutagenic or carcinogenic. So we also observed by the simple chromosomal aberration test using the Helium SEPA, and chromosomal aberration was observed before the treatment and after the treatment. Thus chromosome separation was, and this aspect was integrated with the plant, because phytoremediation is one important aspect in the nature. You can see how the pollutant is affecting to the environment, and cattle are taking the same thing as a food, and that comes to our food chain also as a contaminant. This picture is showing very, very hazardous, looking as a charcoal material discharged from the alcohol industry. There is actually we were thinking there will be no any growth of the microbes, but he found that some bacteria were growing. This picture is showing the aspect of the metal, how it affects to our health. This is the status of this sludge, what I showed in picture, how much manganese, chromium, copper, zinc and iron, including the lead, was also available. So that is showing the toxicity along with the organic pollutant. Again I try to detect the pollutant present as a toxicants, because these pollutants are not known so far, and the CPCB monitor only BOD, COD, and still in our country there is no any rule or norms to control about these pollutants. So the yellow highlights are showing actually these pollutants as a under category of the EDC compound, because they affect to the fish or the aquatic flora for their hormonal reproductive system. And further we also identified by using the different solvent to reveal the range of all these pollutants. So these two bacteria were growing very comfortably, Staphylococcus. This showed that this bacteria is pioneer in case of the highly polluted sludge. And further this provides the chance for the successive transfer or replacement of the other bacterial growth in the sludge. And the bottom one is showing the attachment of the rhizospheric bacterial population through the root hair of the plant which grow in the subsequent stage. This is the view, the plant, how they can grow on the sludge itself for the eco-restoration. And we collected several indigenous weed variety. This is the cannabis sativa, which is known as a flax also. And this was very much potential, and we found, and we screened several medicinal plants also, they were growing on the sludge itself. So this showed the contamination, and they also have PGPR activity. So the mechanism you can see quickly, because, and this is the nanoparticle, how it goes inside the plant cell and how they facilitate for the eco-restoration of our polluted environment. The interesting point is the metal. Metal actually does not degrade at all. Metal converts from one to another and it comes into the ionic form and it is accumulated inside the plant cell. And how the plant actually adjust themselves in the highly contaminated area. You can see the picture of different plant cells under the electron microscope. They actually, the bottom one is showing for the Ricinus communis plant, which has been reported very tolerant and potential by making the multi-vacuole, and slowly the accumulated metal actually deposit inside towards the periphery region. And different plant actually has different mechanism of their adaptation for the metal accumulated in their threshold limit, beyond threshold limit. This is the view for the other plant, how the granules of the metals are accumulated inside the plant cell cytoplasm. This is the view of the Ricinus. You can see the bottom one is the multi-vacuole formation. They are coming towards the cell wall. And multi-vacuole, this left figure is showing multinucleolus formation. This is a type of adaptation of the plant at the highly concentration of the heavy metal inside the plant's tissue. And this is again rhizospheric bacterial association. Plant microbe interaction is nowadays also one important aspect of the microbiology and biotechnology. So we also screened the comparative accumulation of the plant, and under this actually study we developed one patent, and this finding was highlighted from the local newspaper and national newspaper. And in this area I published one book from CRC. This book is actually running in almost 80 countries. America has included this book matter into their graduate courses also. And advances of biodegradation of industrial waste, we are also, they have invited for second revision. So we published several papers under this finding. This is my team, and work was funded from DST, DBT, including CSIR, and this I actually, due to the shortage of time, I showed you the just glimpse of the some finding, how the microbiology and biotechnology can help into the nature for your eco-restoration and for sustainable development. But still the path is long. You have to understand many things. This picture is showing we have to go still long distance.
Thank you,
So
Professor.
thank you very much. Due to the shortage of time, I have squeezed my talk. But whenever you like, you can interact. My two books has been also published on bioremediation and microbial biotechnology in 2025 this year. I have circulated on the my site. So all the microbiology, biotechnology student or PG student can purchase that book. There will be a lot of the basic information required to you. So thank you very much, sir. And thank you to our
Thank you,
audience.
sir. Now request our
organizers.
chair for the session to kindly share a token of respect and gratitude to our speaker, please. Dear students, if you have any questions during the lunch time, you can
I will be
interact with the
pleased to
speakers.
reply any query to the young students.
During the lunchtime, you can interact with the speakers. And
If
you
Dev
can check
Bhoomi
your
will
queries.
invite for any detailed talk on bioremediation, phytoremediation, or lignin degradation, so I may be happy.
Would be our honor, sir.
Thank you.
Thank you.
Faculty or students, they want to purchase any of your book. What is the kind of concession from your side they will get?
As an author, what I get my privilege, I will extend my concession to them. Almost 25%.
To 25% from the author they will
Yeah,
get.
even I can make some library also. I can hand over 1 or 2 copy.
Good, good. That you can send. AMI can certainly, because now we have started our permanent office of
Yeah,
AMI
that
as
will
well.
be good, sir. Especially the members, youngers, are actually in need to have a good knowledge, because they actually contact to the Google or online. Many things are not clear on the Google. And there is also fragmentary knowledge. So any textbook, because the fashion of the textbook has reduced nowadays, if you will read textbook as a notebook, and you will get lot of the things, curiosity. If you have knowledge of the basic information, then you will develop your self-interest to the this
Professor
conference.
Banwailal is waiting for.
Yeah, thank you, sir.
Thank you, sir. So students, if you want to buy his textbooks, is available online. You will be getting 25% discount what is mentioned for the I request Professor R.C. Kohart sir to please join us on stage. He would like to have a photograph with you. Thank you, sir. Over to my chair for the next session.
Next talk is Dr. Souvik Kumar, Naipar, Ahmedabad. So try to complete in 15 minutes.
Yeah, please, sir.
You will get the warning.
Yeah, definitely, sir. I will complete, or if I will take more time, please indicate. I will stop my. Yeah. Thank you, AMI, for giving me opportunity to present my research output in this platform. And today I would like to talk about today I will talk about the fabrication of electrochemical biosensor for indirect detection of swine flu. So my laboratory is basically based on Naipur, Ahmedabad under the Department of Medical Devices. And my overall work is related to replacement of traditional electrodes. Generally, what we are seeing, a cost of a 3 electrode system is about 400. So I would like to replace with the traditional, traditional electrode with thread and paper. And its cost will be less than 10 rupees. So this is the first our aim. Second one is material synthesis, that is derivatives of hydrogen substrate graphene, graphene and graphene. So derivatives of these are not available till date in the market. So these two work, we are working with Nexicon India Private Limited. So with this company, I am working this a specific area. Third objective is disease diagnosis, where I would like to develop point of care devices for cancer biomarker diagnosis, swine flu, cardiovascular disease diagnosis, and last one is mental stress determination with the help of some specific biomarker that may be in serotonin or cortisol. So this is the third objective. And fourth one is would like to work in the field of bearable devices. What you have beard in your hand, this type of thread can be used as a biosensor for disease of various diagnosis, for diagnosis of various diseases. So this is my core area where I am working. So first of all, we will discuss about the nanomaterials, biosensor, swine flu, and here I would like to discuss four different outcome. First one is ITO-based biosensor, and two work that is based on paper-based biosensor, and last one is thread-based biosensor. So when we are talking about the nanomaterials, as we know, its dimension is in nanometer, and it has very good applications in the field of coating, biosensors, fluid cells, and a lot of things where we can use nanomaterials. And due to this reduced in size, it shows very excellent properties in terms of optics, in terms of electronics, magnetics, and mechanical properties. So when we are talking about the biosensor, you just imagine that machine available in the market, that is glucometer. So just like glucometer, I would like to develop biosensor that can use for the diagnosis of various cancers or mental stress or cardiovascular disease diagnosis or even swine flu. So there are several components of the biosensor, just like analytes that we need from the biological samples. Another one is analytes, and another one is bioreceptor that may be in the form of antibody, DNA, RNA, etc. Electronic component, there is two different components of glucometer. One is electronic component, another one is chip. So here we would like to develop chip, and another part, electronics part, we will work together with some specific incubator, and we are trying to develop these devices. So when we are talking about the swine flu, as we know, as a WHO report 2024, about 17,201 cases, 11 cases occurs in between 13th May 2024 to 26th May 2024. So on the basis of this one, we have selected this specific disease for diagnosis. There is several biomarkers that are secreted in biological sample. It may be in the form of blood, it may be in the form of saliva, it may be in the form of urine. Wherever we can diagnose this biomarker, on the basis of that one, we can easily diagnose a specific disease. It may be swine flu, cancer, or this one. So when we have analyzed various biomarkers, then we have seen SAA biomarkers is showing prominent changes. That is, when healthy subject, its concentration is about 12.2 plus minus 15 microgram per ml. But when if subject is suffering from the swine flu, its concentration goes high. So on the basis of concentration, we would like to investigate the normal subject, or we would like to differentiate normal subject with the swine flu patient. So this is the output. First work we have done, or we have published in material chemistry and physics, nanostructure zirconium embedded porous carbon modified ITO based biosensor for swine flu detection. So we, in this one we have used two different materials, porous carbon and zirconium nanoparticle, that has some specific property. Several chemical reactions, some physical process is used for the synthesis. This one mainly is hydrothermal process is used. After this one, we have characterized this material with several different techniques, XRD, TAM, we have generally used, and we have also used SAM analysis, step-by-step fabrication of the electrode. These are some other data that we have gained. This is the main data C part, when you are seeing this developed biosensor having ability to diagnose this SAA biomarker in the concentration 10 to 100 microgram per ml with a sensitivity 95.88 and LOD with 6.37. So on the basis of this one, we can see our developed sensor can able to detect SAA biomarker. So we have compared it. This last one is the present work compared with the traditional technique that is used for the detection of swine flu. So this one is this is the first work. Another work we have done, hydrogen substrate graphene, about similar technique that we have generally used, but process is different. Here I have chosen hydrogen substrate graphene due to high conductivity. Its conductivity is about 10 to the 3 Siemens per centimeter. So it make a novel material for the development of biosensor. These are the process that have used for the fabrication of this one. And this video I would like to show you, that will show we have developed 2D nanomaterials for, so you can see where we have synthesized 2D nanomaterials in our laboratory that can easily transfer from one substrate to the another substrate, and it can be used for the development of bearable sensor, solar sensor, or flexible solar systems. And wherever you can want, you can use to develop memory cell or this type of cell. So this is the video what we have synthesized in our lab. So you can easily transfer your one part, your synthesized nanomaterial to the another part. So for the characterization of this one, again we have used XRD, TAM, SAM analysis, FTIR study, and this is the main electrochemistry that have used for the development of our biosensor on the basis of CA. We can see its linear detection range in between 1 femtogram to 1 microgram. Another linearity I have also investigated or found in between 10 to 100 microgram per ml. So there is two different when you are seeing figure C, you can see two different linearity graph you can obtain. So this is the output of the second work. I would like again show to some video that shows our fabricated electrode. That is, this is paper based electrode actually. So here we have developed, so this is the paper electrode that we have fabricated, conducting paper, that is conducting in nature. It is modified with some nanomaterials, that is hydrogen substrate graphene. So you can see here bulb is glowing, and when we are folding again, then we have no lose its conductivity. So we just fold this several times. We are in the process to transfer this technology to Nexicon, Semitech India Private Limited. Whenever time is over, sir, please indicate me. Yeah, so you can see here after folding, we cannot lose its conductivity. So, yeah. So here we have compared first one to second one. Here we got shelf life up to 48 days. We need at least for marketize at least 90 days for marketize any product. So now again we have worked with phosphorus doped hydrogen substrate graphene to improve its sensitivity. Similar technique we have used. We have incorporated or intrinsically doped phosphorus to hydrogen substrate graphene. Here you can see this is the prediction. Red color is showing doping of the phosphorus element. So these are the materials characterization, and these are the linear detection range what we have obtained. Its linear detection range is in between 0.01 femtogram per ml to 200 microgram per ml. So linearity range is very good. Limit of detection is 0.01 femtogram per ml with sensitivity 27.2 microampere. So we have again compared 63 days we have obtained shelf life. Glucometer generally have 90 days shelf life. So commercialize this one, we need at least 90 days, but sensitivity and linear detection is more than enough. So fourth work is published in Biosensor Biology, very good journal, and this is the thread base. So this is the output. Next time we will show you more result. Yeah. Electronics part, I would like to show what type of research we are doing. Yeah. So electrode is okay. Another part is electronic devices. So we would like to develop electronic devices. We have done this experiment in our lab. Point is that where we are stuck, we are not able to convert ampere what I have gained into nanoampere or microampere. So we are contacting with some industries or some company who is working in this specific field. So this is the output. So thank you AMI to giving me opportunity. This is my lab at when I was in Delhi University, and this is my lab left side. And this is my current student, PhD student, and these are my master and PhD student, and this is the previous one. Thank you.
Thank you, sir. Now I request our chair to kindly present a token of respect and gratitude to our speaker, please. I request ma'am to please present a token of gratitude to the stage.
Transfer this technology to industry.
Sir, when we have contacted with 3 different companies, Vanguard and Zydus Medtech is working to develop this type of electrode for various disease diagnosis. In materials, electrode fabrication, material that is hydrogen substrate graphene, and another one is thread-based and paper-based electrode is working with the Nexicon Semitech Private Limited. So we are in the process to this. Okay, thank you. Thank you, sir.
On the behalf of Dev Bhoomi Uttarakhand University, we present a token of respect and gratitude to Dr. Sanveer Kumar and National Institute of Pharmaceuticals Education and Research, Ahmedabad.
Thank you. Thank you, sir. Thank you.
Thank you, sir. For the next talk, I shall announce. It is an industry talk and is a sponsor partner with us, that is Thermo Scientific. I request Dr. Arun Kumar to please join us on stage for his presentation. Thank you.
करूँ सर?
Dr. Kumar, I request you to kindly complete your presentation in next 10 minutes, which is the directed time been given by the chair to you.
Hello. Project नहीं हो रहा है. Hello. Hello. Am I audible? Hello. ये चल रहा है. Sufficient. निकाल दो इसको.
आवाज़ नहीं कर रहा है.
It's here, sir. Please.
Hello.
You can proceed.
Hello. I'm okay. Hello. Hello. Yes. So, very good afternoon to all of you. First of all, I would like to thank the organizers of this conference and giving this huge platform for all. This is definitely a great platform for researchers as well the industry persons. They are collaborating here, they are sharing their views, and definitely this is a transforming time where we are going more in the translational thing. The research is now boundary between in the laboratory only. It is coming out from the laboratory and going to the public domain. This is a great sign for our country, and this is a great sign for the in the way the vision we are working. So as a industry or industry person, we would like to say from Thermo Scientific that we have a lot of solutions in company that can help the industry partners as well as the researchers who can come forward and want to transform their research to the public domain for betterment of the public. So today I am going to discuss about the Kingfisher Plasmid Maxi Pro. This is basically an instrument used for isolation of the plasmid. Plasmid is an extra genomic DNA that is present in the bacteria as well as yeast and used as a tool for the gene editing, mainly the gene editing. So this instrument helps to isolate the plasmid in bulk amount. But before going into deep dive into this instrument, we would like to repeat or retreat our motto, that is make the world healthier, cleaner and safer. This is our tagline at Thermo Scientific, that we in collaboration with researchers are more on this line. We are making the products that makes the world healthier, cleaner and safer. This is our main motto. If we talk about the working areas, we are more in the biology. We have reached, we have touched lot of dimensions in the research. Like we can see here, cell and gene therapy, mRNA vaccine therapy, biologics, and research and discovery. So we have R&D industry. We are working on the mRNA vaccines. If we are talking about the drug resistance and other things, then there are the solutions in such things like mRNA vaccines can help on these things, or cell and gene therapy is the new dimension of the treatment of the diseases. So these are some solutions, like in cell and gene therapy, we have the instrument and the consumables, and nucleic acid therapies, therapeutics. We have the complete solution for the researchers who are more in development of such therapies or such things. So not going much into details of the things, We have all the instruments and everything in as a complete workflow solution for our industry as well as the researchers. So how we fit into the molecular biology is very much explained by the workflow solutions we have. Now, coming to the Kingfisher Plasmid Pro, this instrument, before going into the details, I would like to briefly define what is the plasmid and how it is important. It is more for the students who are sitting in this podium, that plasmid is a circular DNA, as I said, but this is extragenomic. So we can very well isolate it, modify it, and transform it for our purpose, for saying introducing the new gene, isolating and purifying the new protein. So we can work with all the modifications with the help of this plasmid. That's why plasmid is a great tool for the researchers. So in molecular cloning, transformation, and for the transfection gives you the help, gives you the idea that how we can transform these things into your research, your practical forms. So if we see the purification grade, if we purify this plasmid with manual methods, we get the less purity of this plasmid. But the solution which we are bringing here is giving you the highly purified plasmid that is required for the transfection as well as the required in the advanced transfection grade. So this is the major concern which we are able to tackle with this instrument. Endotoxins present in the during the isolation is the main concern when we use these plasmid for transfection into the cell, or we are using it for any therapy. So we are removing these restrictions with this instrument. This is the entire workflow which we face, which we follow during the isolation and transformation. This starts from the plasmid designing, cloning, transformation, cell culture, and then plasmid purification. Plasmid purification is the central point where we need a purified plasmid for further downstream processing or downstream research. So if we check what we have, if we go to the mini preparation, we are limited up to 40 microgram. But in midi-prep and maxi-prep, we need 1.5 mg, up to 1.5 mg of this plasmid. In mega-prep, definitely high amount of plasmid is required. So, considering this midi-prep and maxi-prep segment, we have the solution with this Plasmid Pro. This Plasmid Pro is basically used by the biotech, biopharma sector, CROs, CMOs, and academia labs, where workload is high. So this is giving a good solution, good platform for all these research as well as industry segments, where this instrument can very well be used for large scale production of the plasmids. Now coming to the instrument, this instrument is basically work on the no centrifugation basis. So generally the plasmid isolation is culturing the cells, pelleting them, then after pelleting we lyse the cell and isolate the plasmid by multiple centrifugation steps that are involved. But this system does not require the centrifugation steps. This is having a kit. This is having a kit which is having all the reagents and components in it, and that can be loaded with your plasmid culture directly into the machine, and rest job will be done by the machine itself. So you can see that there are three benefits. We are saving the time, we are getting a plasmid which is having the less endotoxin, and even parallelly we are giving you the large amount of plasmid at the end of the experiment. We are taking only 75-minute time, which generally takes 4 hours of the process which goes through the centrifugation, multiple centrifugation, and multiple manpower involved. So all this is tackled by this system. So we have further we have checked the purity as well as the further downstream application ability of these plasmid. We have found that this is highly purified and can be very well used for your further downstream applications. So this is again the comparison between the manual workflow and Plasmid Pro workflow. We are definitely having the less steps involved and less time involved in this Plasmid workflow. So this is again the comparison we are having with definitely a good solution of this Plasmid Pro that can be very well used for our industry as well as CRO partners. I'm not going into details of this slide. This is basically the working manual, how we can install this instrument, how we can easily use this instrument. So that is one thing. Now I end my presentation. I hope I am well in time, and I hope that you will have more questions from our Thermo Fisher Scientific. One more thing I just want to clarify here, that we as a Thermo Fisher Scientific has an R&D laboratory in Bangalore and Hyderabad. So we most welcome young researchers, scientists to visit our facility if they want. They can show our workflow, how we work. In Bangalore facility, we are developing more than 1,000 antibodies. So we are producing them there and very well selling, used for selling. So anyone can, if you are interested, you can write us. We can definitely support you to visit our R&D facility there. So this is from my end. If you have any questions, you are most welcome.
So this machine is not having centrifuge, or?
No, sir. This machine does not require any centrifugation process. This is basically a kit-based system where you have to load your culture directly.
No, but how will you separate plasmid?
So this is basically our secret, you can say, the procedure. But this, what I can define here, this works on the pressure workflow. So you load our culture, this pressurized system, and it separates the pellets as well as then further it separate the plasmid.
Now I request once again to our chair to present a token of respect and gratitude to our speaker. The audience, can we have a huge round of applause for the speaker who's delivered a beautiful session to us. Over to my chair to invite the next speaker.
The last speaker is Dr. Ashish Vyas from Lovely Professional University.
So I request Professor Ashish Vyas from the Lovely Professional University to please join us on stage and share his valuable insights with us.
The title of his talk is Integrated Management for Control of Verticillium Wilt of Cotton.
The title for the talk would be Integrated Management for the Control of Verticillium Wilt of Cotton.
यहाँ तो है सामने. Done? Okay, good afternoon all. Thanks, respected Chair, Dr. Banwarilal sir, on the dais, and my microbiological fraternity of the dais. Very thank to the organizers, Dr. Nabil Ahmed, Dr. Namita Singh, for having been invited me for this talk. Before I actually start with my formal talk, this work which I'm going to present is a problem which is something which is very, very localized in Bhatinda region. And one of my students came up with a problem, and then we thought of that we should come out with some integrated solution to the problem which was persisting in Bhatinda region. So the title of my talk is Integrated Management for the Control of Verticillium wilt. Now, just to set a pace, when I say cotton, scientific name of the cotton is Gossypium, and it has broadly 4 species with Hyracetum, Arboreum, and 2 other species like Barbadense and Arboreum and Herbaceum. Now, one thing which I would like to say related to wilt disease, that whenever we say wilt, as a microbiologist or as a mycologist, we often think of synonymity of Fusarium. Fusarium has been known to cause wilting diseases in many of the member of family Gramineae, like in chickpea and pigeon pea and all. So, with very specific focus, there has been some cases of reported of verticillium that I'll be showing you in the coming slides. So, if you talk about Punjab, Punjab is on the 9th position in terms of cotton production. And if we look into Gujarat leads in that, and within Punjab region, there are specific districts like Muktsar Sahib, Ludhiana, Bhatinda. Bhatinda leads in cotton production. In 2025 data, if we can see, 2.9 lakh hectares of cotton has been cultivated, and within Bhatinda region, 95,000 hectare areas is specifically being utilized for growing cotton cultivation. Now, this is something which I really need to tell you when I started my lecture, saying that when we say wilt, Fusarium is the most common genera which has been reported to cause wilting. Now, there has been some cases, I mean, not some cases, very specific case which has been reported 6 years back in Tamil Nadu, wherein people reported that the wilt disease being caused in cotton is not by Fusarium but by verticillium, right? So this was the case reported which I came across through this report of ICR Cotton Research Institute. So verticillium which appeared in Tamil Nadu remained restricted mainly to south zone only. So if we talk about the historical context, this is a soil-borne fungi, and verticillium is a fungi which comes under Deuteromycetes group. And very specifically, if we say the plant body of fungi is, the basic unit is hyphae, and the tuft of hyphae is known as mycelium. Within fungi, there are some modifications which are found when the mycelium modifies, and there are 7 kinds of mycelium modifications like aprosorias and hostoriums and other structures. One among them is rhizomorph, which is found in Fusarium, and sclerotia, by the growth of lateral budding in the hyphal structures. The top hyphal structures, they start growing laterally and they form a microstructural structure. These microsclerotia are being found in verticillium. So once they infect at the blooming stage, it goes and it attacks on the vascular differentiation, the xylem cells, and it starts clogging, and as a result of which the wilting occurs. When the first blooming stage, the symptoms which are being observed is it comes at the first flowering stage, it reveals wilting and become yellow roses. Vascular discoloration is light to dark brown in color, and abscission and malformation of younger bolls at the time of blooming and at the time of boll formation occurs. So yellow between veins and leaves, necrosis occurs in the later. There is one specific word which has been said is tiger stripe kind of pattern is being observed on the leaves at the time of wilting. So vascular discoloration has been found. First it attacks on the cortex region, and then it directly attacks on the xylem cells. So you can see these wilting cases. This is the overall cotton development and growth stages, from root development, vegetative development, and then squaring occurs. Squaring is a stage wherein the specific fruit starts coming, and then the blooming comes, and then the boll development starts, and then after boll development, the defoliation occurs. Fungi starts showing its symptoms specifically on the bloom stage. Of course, it comes on the squaring stage also, but then exact wilting symptom starts appearing in the blooming stage. So within the structure, this microzoa which are free in the soil, they start through root exudate formation and then the direct penetration, and then it attacks on the xylem cells and the wilting starts. Now, looking into, there has been certain the fungicidal agents being used, but then we thought of mitigating the problem through certain kind of an integrated approach, wherein we can take some different methods by way of soil solarization and then soil fumigation, and then taking certain kind of microbial bioinoculants by way of seed bacterialization, taking bacteria into consideration, and then some fungi making a liquid bioformulation. So we have done some kind of an experiment. Because of paucity of time, I'll be just skipping some of the slides. So first we isolated the microorganism verticillium, and in Bhatinda there are 320 districts, but we targeted specifically 20 villages. In Bhatinda there are 320 villages, and out of those 320 villages we targeted 20 villages. And there are 8 broad samples which we have targeted, and for isolating the verticillium, just to confirm that whether the causal organism is verticillium, not fusarium. So the isolation was done. So this is actually a culprit fungus, which was verticillium, and very specifically in the initial 7 days it turns dark brown, and the later stages it turns completely dark black. Reason being because the sclerotia it produces, there has been some, we didn't have done that work, but then while reviewing the literature, it forms DHN, melanin pigments being present in it, dihydroxynaphthalene pigments being present in it. So it's 14 days old colony, and you can see the microsclerotia. Then we have isolated this sample, and then just to check the prevalence rate, the prevalence rate actually is written, highlighted here, which has gone behind these flaws. It is 73% actually. And of overall prevalence rate of verticillium dhalii in infected sample fields in 20 villages, it was 73%. And we got that fungus identified through 18S sequencing. It was to be verticillium dhalii. Based on ITS region, we created the phylogenetic tree. Then our aim was to isolate certain antagonistic fungi as well as some endophytic bacteria. So we targeted certain manures like poultry, farmyard manure, biomanure, vermicomposting, compost manure, green manure, oilseed manure, and animal manure to isolate certain fungi. We have taken almost all villages, various sites of Punjab to isolate these endophytic fungi. Then we have gone for testing with the PIRG method. We have isolated 7 different fungi which are showing potential antagonistic effect, that is Aspergillus niger, Nidulins, and Pteris, then Glyocladium, Trichoderma herzianum, Cladosporium, and Fusarium solani. And then we isolated certain endophytic bacteria, and the 3 endophytic bacteria which we isolated was Pseudomonas aeruginosa, Lysine-Bacillus macroides, and Bacillus subtilis. All were get identified by 16S sequencing, and the fungi were identified through Indian type culture collection, New Delhi. So then we did the percentage of inhibition test just to check that are they showing some kind of antagonistic activity. The antagonistic activity which was shown to be maximum by Aspergillus fumigatus and Trichoderma herzianum. So these were the results. Then these 3 bacteria, among them Lysine-Bacillus macroides was reported, to show 69% of the antagonistic activity. Then this PIRG method, this antagonistic method. Then we also checked the hydrolytic enzymatic potential by way of checking their pectinases, cellulases, and proteases. So the results were, these are the results of cellulases, proteases, and chitinases activity. Then the microbial formulation which we developed was from cow urine. Here I would just like to tell a very important point. The problem which was persisting in Bhatinda region, the farmers used to control verticillium wilt through fermented milk, which we generally call it as lassi, khatti lassi. What we thought was that we'll be incorporating some kind of an adjuvant into it by way of maybe adding cow urine and certain kind of an microbial inoculants so that we can upscale the process under in-lab condition. So we did this work. Then in the field trials, we have gone for studying natural field study as well as spot trial studies. And we have also done seed bacterization. And in seed bacterization, we have taken Rasi 134 variety of seed, which is Gossypium arboreum species. And we have bacterized the bacterial seeds with this method. And then we have checked in through pot trial as well as through natural field, we have checked the disease severity index and the biocontrol potential protection. So these methods are being written here. Then we started with our experiments. I think I should come directly to the result part. Now, if you look into the disease severity index, the Herzianum was showing 28% compared to the control. And if you look into some different, Herzium among the fungal strains has gone for giving some wonderful results to us. This was the biochemical formulation which was prepared. Some pot trial studies. So if we compare it without biochemical formulation with that of the biochemical formulation, Trichoderma herzianum came out with 79% of the showing the biocontrol potential. We have also checked in the yield per plant. I mean, in a plant, normally there are 6 bolls being found in a cotton plant, and within a boll we have 2 gram of lint being obtained from the seed when we remove the fibrous part. So compared to control, we have got somewhere around maximum in 23 grams from Trichoderma herzianum as well as from Aspergillus terris. We have also checked in that when we have inoculated the microbial inoculant, the seed bacterization, the fumigation and the soil solarization, we have seen that there is an increased root length, because endophytic bacteria and these trichoderma fungi, they also promote certain biocontrol potential as well as the growth promoting activities. So we have seen the increase in the length of roots also. So the major focus was that we need to work out with some kind of an antagonistic fungi deported from different organic manures. And the seed bacterization with Lysine bacillus macroides reduced the wilt symptoms with natural field as well as pot trials. And most effective was a combinatorial study when we take Herzianum into account, when we take Lysine bacillus macroides into account, when we go for soil solarization. So it subsequently, reduce the tendency to develop verticillium wilt. Thanks to Punjab State Council for Science and Technology for giving this funding for the research work, for our MSc dissertation work. Presently, the girl is working on the molecular aspect of this entire work, that what are the impact on the soil and all those things. We are working on that. Maybe we can work out with the specific serotype culture of verticillium, that what kind of pathogenic potential in terms of the genes it has. And thanks to a lovely professional university. We have filed a patent also for it. So recently its FER has come, and maybe we'll be publishing it and getting it granted.
Thank you, sir. Now I request our chair to present a token of respect and gratitude to our speaker, please.
So this verticillium also, in fact, other plant, how it will survive in soil once the crop is harvested?
Sir, actually it's a Sclerotia. If you talk about Sclerotia, the way we say endospores are found in Bacillus. Amongst the modified forms of fungi, Sclerotia is the most resistant form. The DHN, which I am talking about, it is the specific compound which has been found, making it, if you are not taking any action, it can be found for next 14 years. And even a single Sclerotia has the capacity to start infecting a susceptible plant. So targeting and directly targeting the Sclerotia should actually work out. I must say this thing because we are the second people who are reporting verticillium wilt. Otherwise, first report is from Tamil Nadu region. The problem is somewhat localized. So what I really urge from my MSc students, they should come out. I mean, people are working on global problems. In the pot fields as well as in the natural fields, we have covered the area where we have experimented with plastic sheets at the temperature. So after that we have checked it, the verticillium was not found. And in the control case, we have inoculated it.
Thank you, sir. Now I request sir to kindly share a respectful token of gratitude to our eminent speaker from lovely professional university. Audience, can we have a huge applause for our eminent speaker? Thank you, sir. Now I request the Associate Dean Academics, Professor Kanish Jha, to present a token of respect and gratitude to our chair of this session, Professor Banwari Lal. I request Professor Kanish Jha, the Dean, Associate Dean Academics, to please join us on dais to felicitate our eminent chairperson for today's session, Professor Banwari Lal. Thank you, sir. So it is the time for the panel discussion, Collaborative Dialogues, Bio-E3 Policy and Biomanufacturing. The Collaborative Dialogues on a Bio-E3 Policy and Biomanufacturing focuses on exploring the evolving landscape of biotechnology policy, innovation and industrial biomanufacturing in India. The session aims to highlight the Bio-E3 framework, education, entrepreneurship, and ecosystem, emphasizes how these three pillars can work together to strengthen the Indian bioeconomy. Experts from the academia, industry, and policy sectors discuss how enabling policies, public-private partnerships, and research-driven innovation can accelerate biomanufacturing in the areas of biofuel enzymes, pharmaceuticals, and biomaterials. The dialogues can also examine the strategies for creating a self-reliant, sustainable, and global competitive manufacturing capabilities aligned with the Make in India and Atmanirbhar Bharat mission. With this, I would like to invite the moderator for this session, Professor Prince Sharma, the President of Institution of Microbiologists of India. Sir, I request you to please join us on dais.
Yeah, I can hear you. Thank you.
With this, I would like to invite the panelists for this session, Professor R.C. Kohar, the Chairman, AMAC, Dr. Gulshan Wajwa, DBT is joined online with us, Dr. Banwari Lal. I request everyone to please join us as soon as possible on the dais for the panel discussion.
Hello. Hello.
Over to my moderator for this session.
Good afternoon, everyone. I think this is the session between you and the lunch. So we'll try that we do the maximum, we take out the maximum mileage of this particular program and have a nice lunch after that. This is probably the fourth panel discussion which we are holding in this AMI conference. And this flurry of panel discussions on some important topics which AMI thought are pertinent, burning issues as far as education and research is concerned, as far as the economy is concerned, as far as the role of microbes in circular bioeconomy or biomanufacturing or education or industry is concerned. So we planned different panel discussions. And this last one today is again on the topic where India is trying to excel, where India is trying to do the biomanufacturing and come up on the map where it will be competing with the developed world. And to do that, a lot of work is to be done. And the area Government of India has chosen one area, this particular area, at BioE3, which stands for biotechnology, for economy, for environment, and for employment. Very pertinent. And in this, this particular BioE3 policy was put in place on August 2024. And government since then has been trying to do multiple things to achieve these objectives in the next 10 years. There are 6 verticals to that. The 6 verticals which we'll be discussing shortly in this panel discussion, the 6 verticals which have been identified and which India wants to excel and complete those, we will be discussing, and the fallouts of those. We'll also be discussing what are the challenges, what are the problems during this accomplishment of this can be obtained. When we say that BioE3, biotechnology, we can understand that how important biotechnology is for economy, for employment, and for environment. And biotechnology is not complete without microbial biotechnology, where microbes are going to play a really vital role in all these processes of biomanufacturing. Also, when we talk about biotechnology, in all these verticals, 6 verticals, we'll be talking about the blue biotechnology, the white biotechnology, the red biotechnology, and the green biotechnology. All they are important. So carrying further to this, let me start with our panelist Gulshan Badbaji, if I can ask that. Sir, this BioE3 policy which is in place and government is doing so much to accomplish it, and the first biomanufacturing institute, the National Agriculture and Biomanufacturing Institute has been formed in Mohali, that's the first one. How do you think that BioE3 policy is going to really take India to the map where it will be like a developed economy, highly competent developed economy? Sir, you are not audible. Organizers?
Am I audible, sir?
Yeah, now you are.
Thank you. Distinguished guest, esteemed colleague, members of the scientific and industrial community, welcome to this panel discussion, India's landmark BioE3 policy and the Department of Biotechnology's program on biomanufacturing. Today we will explore these initiatives which will shape India's bioeconomy, accelerate sustainable manufacturing, and create a new pathway for employment and environmental stewardship. As you know, Government of India through Department of Biotechnology recently approved Bio-E3 policy as just has been told. This policy articulates a vision to transition from conventional manufacturing to a regenerative bio-based model, essentially aligning with the circular bioeconomy principles. It is not just about making biopharma, but chemicals, enzymes, smart proteins, even capturing carbon biologically. Carbon capture is the most recent phenomena, which everybody is around the world is talking, and reducing the carbon footprints in different countries at different levels. The vision of this policy is to position India in the forefront of a sustainable future through biomanufacturing that integrates bioeconomy activities while attending to environmental impact. The goal is fast-track innovation to technology in a sustainable way, bringing together fragmented biomanufacturing efforts in India. And the objective of the program is to promote adoption of advanced biotechnologies, align R&D to revolutionize production processes, and accelerate development of high-value based products. So, as has been just mentioned, the policy is structured around 6 thematic verticals. These verticals include bio-based chemicals and enzymes, functional foods and smart proteins, precision biotherapeutics, climate resilient agriculture, carbon capture and utilization, and futuristic marine and space biotechnology research. Let us talk about how DBT intends to deliver this. The policy calls for establishing bio-enablers, especially the bio-AI hubs, biofoundries, and biomanufacturing hubs. These are the 3 major components of the policy, and this is the way it has been initiated. First call for proposal is already given few months back, and it is under process, and I hope many useful new products will be coming out of this call, and this will be developed in the coming few years. These are meant to be geographically distributed, shared infrastructure centers where academia, startups, and SMEs can scale their bioproducts. DBT is already issuing joint calls for proposals with BIRAC. Some calls, industry portion will be handled by the BIRAC, and the academia portion will be handled by the Department of Biotechnology. But translation to commercially viable products, the policy explicitly ties to entrepreneurship, skill building, and scaling up of the products as well. The policy envisages interministerial coordination for biosafety regulation and ethical governance. Also, data governance is a part of the strategy, ensuring balance between open science and intellectual property. The DBT-BIRAC joint call particularly interesting. For example, the call for carbon capture and utilization 6 projects that are energy efficient, that are cost effective, and that are scalable to a larger level, so that we can fulfill the needs of the country as well as we can supply to the part of the world where they need all these products. That could also convert.
Sir, just to interrupt that, can we be very specific to that? How is India going to achieve these? What steps Indian government is taking to achieve this in the coming few years?
This calls and the subsequent calls are the process by which government is going to achieve, and the products developed through this will be given to SMEs, MSMEs, and other industries to scale up and to give them support also.
Okay, sir. Right. Thank you, Dr. Gulshanji. I mean, you have described it in detail. that what exactly is Bio-E3 and how government India is poised to complete it. I come to Dr. Banwari Lalji, he is into much of this biomanufacturing also, that one of the impediments in achieving the goals is that it is very custom, because see, so many of the ministries are involved in that, agriculture or industry or environment and different industries, and DBT also, of course. Is it, how possibly is that they must work in coherence to achieve this, because most of the times they work in silos?
I think here the biggest problem is that how to achieve this. It's very easy that thinking that with this policy there will be complete transformation, but difficult. The reason is that because these MSMEs and this startup, they do not have any good product which will give them good business. That is the one limitation. Another is that the scientists also, as I think in my previous talk I told that scientists have done the basic work, but they have not developed the complete solution. So until unless there is not a complete solution, then industry also confused, because industry technically they are not qualified to do further improvement. They can upscale this, they can replicate this, but they do not have R&D facility, because R&D is costly, they cannot afford. So I think here the synergy is a problem. Scientists, they are doing many things repeatedly. I think if you see, your scientists are writing project after project and maybe 90% repetition. If we sit in the DBT task force meeting at this, I think presentation would be mostly the things, there is no lead. So that is the thing that government thought that how to do some changes in this. So therefore they started this policy and they have allocated the fund also. I think more than 2,000 crore rupees they have allocated. But the problem is they are not getting good proposals. Even DBT also, the BIRAC, they have given the call, but good proposals are not coming. The proposals, because the big corporates and big industry like Adani, Ambani, they are not coming in this. Startup, they do not have expertise. First of all, they do not have complete idea. Nowadays, many startups, they think very easy. They started more than, I think, around 10,000 startup in biotech.
1,000 something are there still
I think
now.
10,000 startup. But I think if you see success ratio is very less. Because they also like us only, they have opened the startup, but they do not know how to take it further. Another thing is that in biotech, because big industry, corporate, they are not coming, so investment is also big. Although BIRAC and this, through this call, they will support. Through this call, they will support, but this is a public money. They will not give them as a grant. They will give as a either interest-free loan or sort of a soft loan. So they can take up these ideas and they start manufacturing. So I think a lot of problem. And this policy also, let's hope that this would be successful.
But sir, my question was that will this be how successful will be that these ministries will coordinate and academia will also come in and coordinate, DBT will coordinate, and that's because till now these are some of the, always have been the problems. In ministries, they never coordinate.
Yeah, that is true, because in here, it's not like China. China is a single window clearance, but here, not that type of thing. Even GMOs, it's very difficult to get approval. If anybody has developed any genetically modified product, very difficult. It will be going in so many ministries, so there is no good coordination. But through this policy, they think that there will be good coordination and there will be nodal person from each ministry. They will help to the startup or industry people to get the clearance. I also submitted a proposal and proposal also approved 65 crore rupees as a soft loan, interest-free loan. But there are lot of regulations and approvals and these things. So, because DBT also helpless, like BIRAC and this also helpless, although government is pushing that there should be good synergy among all the ministries. hopefully I think there will be some good outcome of this. But I think we can see the transformation until unless there will be a big industry or big corporates will not come, like Adani, Ambani, or corporates, profit-making corporates, they will not come.
I know, sir, because that's what my next question was to be, that because regulatory red tape there, which is there, that to cross that and to make the processes expeditious, many of them, they are skeptical about them,
Yeah,
very skeptical about them.
Yes. So you can see like the ethanol is a biofuel. Those who has taken initiative, they have become rich people.
Yeah. Okay. So that we have these problems will always be there. I will go to Dr. Kohar now. He is an academician. You see, sir, there are 6 verticals to this Bio-E3 policy. Do you think that government is going weightage to equal weightage to all the 6 verticals and will be achieving it, or it is as per India's need, it is going to give weightage to 1 or 2 particular verticals more than others?
Thank you, Professor Prince Sharma, for giving an opportunity to share my views. We all heard Dr. Vadwaji, who is from Department of Biotechnology. And this is a very good scheme, very good policy, which has been launched by our Honorable Prime Minister, as you have been told by Professor Sharma, that in August 2024. Actually, every scheme is really very good. But this kind of the scheme is aimed at achieving the sustainable development goals defined by United Nations. And this Bio-E3 is mostly focusing on energy, economy, and environmental sustainability. We need to see that these are very important, say, areas. By getting sustainability in energy, environment, and economy, I think you'll be serving many purposes, and of course all 6, say, verticals, which also have been briefed by Professor Prince Sharma, we can meet it out. Now it depends whether at the same scale or the same, say, the bench or the same, say, level of achievement we can. Of course, there has to be an effort that parallelly, if we could work out, then we need to establish the relationship among these, say, 6 verticals. They are very much interrelated, because when we talk about energy, economy, and environment, because fishery, agriculture, forestry, they have been contributing, and of course Niti Aayog targeted that in the year 2030 sometimes there will be 300 billion, say, bioeconomy. And if you add also the microbial based different technologies and the products, I think it may reach to 500 billion.
Yeah.
My suggestion here that if we really have to achieve this, this cannot be achieved based on the DBT under this scheme has started calling research proposals. If you are calling the research proposal from the individuals, it will be like any other scheme.
Truly, yes.
Therefore, they should advise with the guidelines to all higher education institutions to develop a center of Bio-E3. So that based on the strength, research strength each institution has, or 2 or 3 or 4 or 5 institutions together, they identify, say, a level of research they have reached, depending upon the kind of facilities as well, how best they can make a group under this center. Then I think they should develop the program, keeping the maximum verticals including in that very center, And then we can think of, because unless until there are institutions, say for example, you are having the transmission electron microscope, I do have the, spectral microscopy. Others, they do have good fermentation facilities like Professor Banwaila said. If all together, because unless until we don't understand the concept of biorefinery, unless until we don't understand the concept of biomanufacturing hub these days which is coming up, and we don't have economists with us, say industrialists with us, so that we can study the whole life cycle of, say, the whole process, we won't be able to achieve this. Because simply in form of developing a project, submitting to the DBT would be happy that something coming out, they can give their annual report. That will continue like that. Dr. Wadhwa will be annoyed with me, but this is how it has been happening in our...
But I think you have very well said that. I agree with that. Just the research proposals, which 90% are repetitions, they will not take us anywhere. It is truly that different institutes come together for one particular vertical, and then they take that into further. And in this particular context, also let me tell you that DBT should take care of this now, that any technology which is at least at TRL level 4, only that should be taken about. Don't give the projects which again start from the scratch level. Like, for example, in case we have got a project now from Government of India, they said that we had the technology at TRL level 4. We said that we'll take it up to TRL level 7, scale it up and show it working or not. From TRLs, now is the time for technology readiness level, not just starting from the scratch actually. So that's why your point is important. Those clusters of institutes which have technology readiness level at least 4 or 5, only they will be taken further within the paradigm of these 6 verticals actually. Thank you, Professor Quartz. This is very well said actually. I'll come to Dr. Gulshan again. I'll be, Dr. Gulshan, you know that in India's approximately 3.6 trillion GDP. At the moment, I think the bioeconomy constitutes about 150 billions, which India is poised to increase further. And also let me take part in that. India's contribution in this GDP, India's contribution to research and development is only 3%. Compared to China, USA, we are nowhere actually. How do you think that this percentage increase in the research and development, in the total GDP, is going to help in increasing the bioeconomy, in increasing the employability, and reducing the carbon footprints and ameliorating the environment?
Sir, the question which you mentioned is very, very pertinent, very relevant. Bioeconomy has grown more than what we planned in the last 5 years. And by 2030, we plan to have it 150 billion. So it is growing, and the growth is also reflected in the number of companies we have set up in the last 10 years. Through BIREC, more than 10,000 companies have been set up. Through MSME, we have set up more than 120,000 new companies, startups, and at different levels. So the growth rate is no barrier. As Dr. Kuhad has said, the first call for proposal for the BioE3 has gone only for organizing the system into 3 major components, that is biofoundry hubs, which will facilitate the setting up of the biofoundries, bio-AI hubs for facilitating the setup of the AI hubs and AI units, and biomanufacturing hub for facilitating the biomanufacturing units. So these 3 are the pillars of BioE3 policy, again at a different level. where the doubts raised by even Dr. Banwari Lal and Dr. Kohad will be taken care. And the next call will come, the calls, the technologies which are below TRL level 3 will be taken up by DBT, and technologies which are from TRL level 4 and above will be taken up by the BIREX separately. So that is taken care already. So I hope it will be a good initiative, and the way it is progressing. Of course, we don't have many people around the country which are entering into the field, which are knowledgeable, as has been said, but the bigger industries are not interested, or they are only interested in the proven technologies. So we have to rely mainly on the small industries and the startups and MSME.
Yes,
So
sir. You are very, it's very true.
Proven,
Okay,
sir,
please continue. Yeah.
once the technologies are proven, they will be definitely taken up even by the bigger industries. And I hope the growth rate will accelerate and we will cross the 150 billion target even before 2030.
Yes, sir, it's very true that big industries are not interested, because when I go to the market with my product, now we are also having license with the startups. Startups are also looking for the products where they can keep marketed, and BIRAC is now supporting startups also. There was a call for startups to give the project. We have also, in the collaboration with startups, given projects to the BIRAC, and I think this will be a better initiative rather than just depending on only academia or depending only on industry. Startups are very enthusiastic about different projects. That's what I feel actually. Dr. Kohar, again, one more question quickly, because they are just signaling me that, okay, fine, that you are an academician of such a repute. Their stakeholders are DBTs, ministries, MSMEs, public-private partnerships, but then academia can play a very role in skill development, because one of the skepticism which is coming is that in biomanufacturing, in biofoundries, where is the skill? Where is the skill? Skilled people are, they don't find it. That is one of the problems there. So how academia can play a role in giving, in this project, the skilled manpower?
Yes, very, very important. Because yesterday also we were talking about that how really we can enable these youngsters, how really we can make them skilled. For that, because as we have mentioned, and we know that we are in the process of developing the curriculum for undergraduate and master course in microbiology, and we could find when we surveyed that on one hand, biotechnology is bringing the policies and the programs considering the progress of our teaching program and the research going on globally. But they are really not taking care of that what is the standard of education and the curriculum program we have been running within our country. Hardly any master's course in microbiology might be having a course on biorefinery, biomanufacturing. I think sometimes the ministry people also should see these kind of things, and they should write to Ministry of Education, Ministry of Science and Technology themselves should emphasize that there is a lot of gap as far as the teaching program going on in our universities. You should make up. Because unless until we don't have the good trainers to train our students, facilities which are required, although DBT is helping even institutions to develop their lab, but that's a, वो कहते हैं ना कि ऊंट के मुंह में जीरा डालना, क्योंकि institutions are so many, उसमें से usually DBT select उन institution को कर लेती है जो आमी नामी ग्रामी institutions हैं, Rather, they should support those who are in a very, I think, far-flung area, rural area, so that they also get a feel of these kind of facilities and students get opportunity. The teachers should be trained. So I think academia has to play a very big role, Professor Prince, but certainly they also need that how their capabilities could be enhanced.
Sir, as we were discussing yesterday, internships are going to be very important in
very
industries.
important.
In
I
industries,
think
the real time or real training.
Dr. Wadhwa is here. I think you should recommend to DBT that they should spend, say, I think 50% of the grants available for supporting the internship of, say, these science students. If you really want to develop the scientific temperament in this country. Yes. Otherwise, number of startup registered, that will be a progress for the ministry. Yeah. But truly. Number of startup, how many of them are really successful? That's a big story.
Truly, sir, this is something really to be looked upon. I think Gulshan ji, they are making all efforts in DBT to make this Bio E3 policy a success, real success in the coming time, so that our country can really progress. There is a lot to discuss actually, but the time is very much limited today actually, and I had lots of questions lined up with me. I'll come back to you, sir. Just one question for the audience, for the students. Everybody understands what is Bio E3? Anybody who understands what is Bio E3? Raise your hand. Nobody raises their hand. Anybody knows what is red biotechnology? Look into this. This is the future actually which we are talking about. Get into this system. We come back to Biology for the last point if he wants to make. Let us be very positive as citizens that government will do very good in this.
So I think the biggest problem is skill. As you know that here students, they are taking the theoretical knowledge, but they do not have practical knowledge. Even the student doing M Pharma, even they have not seen HPLC, which is a very basic thing. So I think biggest problem is here, and government is also helping, and the government started another scheme, Vigyan Dhara. So the purpose of that policy is that they will provide the skill development to the young students, and they have given the target to all the corporates, industry people, that you have to give skill development training to so many students. I think academia also should approach them. The industry people will not approach. They have to approach them, and they will also give fellowship, some fellowship also. So government has made mandatory this. They are putting so much on industry people that you have to train them. But we have to utilize this. So I think that is the thing, that how we can develop the skill manpower.
We have been discussing things yesterday's also, that CSR, they should divert towards these things actually.
Because I'm trying to make opportunity of the representative from the government, and is again a good friend of ours, and we can really pass on the request. Wadhwa Saab, the problem is that government is asking the industries, it's a very good step, but industries are giving the CSR to the other agencies, and those agencies are not really serving the purpose. Therefore, I think the scheme should be slightly modified. Educational institutions should be given, say the grant, and you should have a list of the experts, and those could be assigned as, say, professor of practice or adjunct professor to those institutions. Facilities will also be developed, and we'll be getting the trainers, so that the chain will continue. That's my suggestion.
It's great manpower also. So with this, we have to finish this. I again urge the students that get into research, get into internships as much as possible. All those students who are really interested in research and internships, they can give their names to the organizers. I will select one of them, and I will give them free internship in my lab, anybody who is interested. From 1 month to 6 months, I will give that.
Thank you,
And
sir.
similarly, I will urge others also that give them internships to the advanced techniques free of cost. Thank you so much.
Thank you, sir, for giving this opportunity. And thank you to our one of the panelists, Dr. Gulshan Wadhwa, sir, to joining us online on the behalf of Dev Bhoomi Uttarakhand University. Now I request Professor Sandeep Sharma, sir, the Pro Vice Chancellor of Dev Bhoomi Uttarakhand University, to share a token of respect and gratitude to Professor R.C. Kuhad, the founding chairman of AMSC. Sir, I request you please join us in front. Thank you. Audience, can we have a huge applause for one of the panelists for this session, Professor R.C. Kuhad. Now I request to share a token of respect and gratitude to Professor Banwari Lal from TERI. Audience, can we have a huge applause for another panelist, Professor Banwari Lal. Now I request sir to kindly share a token of respect and gratitude to Professor Prince Sharma, the President of AMI, as a moderator for this session.
So we are very thankful to Gulshan ji. He could not be here, but we are really thankful for your comments, for your vision, and we really wish DBT and our country to be very successful in this policy.
Thank you, sir. Thank you very much.
Thank you, everyone. With this, it's a time for the lunch. I just have a special and important announcement that sharply at 3 o'clock, our chief guest will join us. So I request everyone to be here back before 3 o'clock so that we can start with the valedictory session. Those who have already, they have their lunch, they can be seated at their respective places. By the time I request all the people to go for a lunch and be here back by sharp before 3 o'clock so that we can start with the valedictory session. Thank you. Hello, 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 hello. Hello. I request all the students being sitting here, kindly put your phone on silent. Very soon we are starting with the valedictory ceremony of this annual conference. I request everyone to kindly be settled as soon as possible. In next a minute, we are starting with this valedictory ceremony. Hello. I request everyone to settle down fast. We are about to start with this validatory ceremony very soon. And I request all the people who are being here in the pandal, kindly keep your phone on silent. Your appreciation will be highly appreciable. Thank you. The people who are joining just right now, please settle down fast. We're about to start this validatory session. And I request everyone to kindly keep their phones on silent. Thank you. I request everyone to please settle down fast at the back side, as our chief guest is not able to join us offline due to the unforeseen, but yes, he's accepted our gracious invitation and he's joining online with us. So he's about to join us online very soon with us, so I request everyone to kindly please be settled. So good evening one and all present here. I welcome you to the validatory ceremony of the 66th AMI Annual International Conference and Research Conclave. It is a time to introduce our chief guest with you all, Professor V. Narayan, the chairman of ISRO. Dr. Narayan is a visionary leader and a pioneering scientist whose contribution to space research, technology development, and scientific information have placed India at the forefront of global space exploration. Under his leadership, ISRO has achieved remarkable milestones in the satellite technology, space mission, and application of space sciences for social benefits, including in agriculture, environmental monitoring, and disaster management. So we are deeply honored to have you as a chief guest at the valedictory ceremony of 66th AMI International Conference and Research Conclave. We are eager to look forward to your inspiring address and guiding us and all participants, researchers, and young scientists present here. Ladies and gentlemen, please join the warm welcome to our chief guest for today, Dr. V. Narayan, the chairman of ISRO. Can we have a huge round of applause? He's just joining in next 2 minutes. A very good evening, sir. We welcome you to the valedictory ceremony of AMI International Conference and Research Conclave. On the behalf of
Good evening to all of you.
Over to you,
very
sir.
Good evening to Honorable Minister is there, I understand. Very good evening to all of you. Hope it is audible to you.
It is already started, sir. Over to you. Please proceed, sir.
Yeah, but I only greeted all of you. So good evening to all of you. So,
a very good evening, sir.
Yeah, good evening.
So I would like to introduce once again to our chief guest for today, Professor V. Narayan, the chairman of ISRO. Dr. Narayan is a visionary leader and pioneering scientist whose contribute to space research, technology development, scientific information, and place India at the forefront for global space exploration. Under his leadership, ISRO has achieved remarkable milestones in satellite technology, space missions, and application for space sciences for societal benefits, including in agriculture, environmental monitoring, and disaster management. So we are deeply honored to have you as our chief guest for the valedictory ceremony of the 66th AMI Annual International Conference and Research Conclave. We are eagerly look forward to your inspiring address and guiding to all participants, researchers, scientific present here. Ladies and gentlemen, please join in welcoming our chief guest for today, Dr. V. Narayan, the chairman of ISRO.
Thank you. Sir, hope it is audible to all of you. Can you confirm? Okay. Very good afternoon to good evening to all of you. Most respected Education Minister, Government of Uttarakhand, Honorable Taban Singh Rawat ji, Vice Chancellor of DBUU, Professor Ajit Kumar ji, Vice President of DBUU, Mr. Aman Bansal ji, Founding Chairman AMC, Dr. R.C. Guhant, General Secretary of this AMI, Dr. Namida Singh ji, and distinguished personalities, members from academia, industries, and my dear young students. Very good evening to all of you. First of all, let me apologize that I could not participate in this very important event of 66th AMI Annual International Conference and Research Conclave, what Dev Bhoomi Uttarakhand University, Dehradun is conducting. I would have been so happy to be there. In fact, we are having our own office. I originally planned to come there, but then the demand from my office, I could not come. I apologize for that. And the theme of the workshop at the outset is set very nicely. Let me congratulate the organizers for conducting this conclave on a very holistic and strong theme: new generation microbiology, education, innovation, and research for economy, energy and environment. What a fantastic topic it is. And I could go through the type of papers presented, type of plenary talks, type of various presentations. It was really outstanding topics what you have selected. The next generation aspect emphasized the need for modern and innovative approaches to education, research and technology in the field of microbiology. And more importantly, it shifts the focus from pure academic knowledge to real world applications. That's what really, attracted me. Generally, I was looking when this conference, the proceedings, not proceedings, the deliberations, what has happened. Academic institute like this, when you organize, generally most of the papers will be purely academic oriented. Whereas here I saw a little difference, at least 50 to 60% were not pure academic oriented, it is application oriented R&D activities. Now, dear friends, when you come to Indian space program, and today there was a time we were working only on satellites, only on launch vehicle and the ground segments. But today, after 63 years of activities, today the Indian space program under the visionary leadership of our Honorable Prime Minister, Sri Narendra Modi ji, we are spreading our wings in multiple areas. As you are aware, today we are an organization working on the human spaceflight program, and the entire activity of the space activity started in the year 1962. And 1963 was a very, very important year for us. We accomplished the first major milestone on 21st November 1963. That was the day the first tiny rocket lifted off from Indian soil, a small tiny rocket lifted off from Indian soil, and we made the first achievement. And when I look back, the tiny rocket was given to us by USA, and the payload for that, a sodium vapor lamp, it was given by France. And the entire thing was assembled by our team, our elders, in a church building. There were no formal proper buildings we had. It was assembled in a church building, and that was accomplished. Today, when you look back, that time when we accomplished that, we were truly, if you make an assessment, we were 60 to 70 years behind advanced countries in the space activity. That was the stature. Just we started our activities. From that humble beginning, receiving a tiny rocket from USA and launching, this year, July 30th was a major milestone and accomplishment for us. We had an important mission called NASA ISRO Synthetic Aperture Radar mission. You know, for that mission, a payload called a L-band SAR payload and the antenna, 12-meter unfurlable antenna, was developed and realized by spending 10,300 crore by JPL NASA. And a equivalent payload, L-band SAR payload, we have developed, and the entire satellite was built by Indians in India, and it was placed perfectly in the orbit by Indian launcher. That is the type of growth what has happened over 63 years. Getting a small tiny rocket from USA, today placing the costliest satellite ever built in the world by Indian launcher. That is the type of growth what is accomplished. Next month, December 2nd week, we are going to have we have scheduled a targeted a very, very important mission. A 6,000 kg communication satellite of USA, purely on commercial basis, we are going to launch using our launcher. Now, if I summarize the Indian space activities, its accomplishments and achievements, from 1963, November 21st onwards till today, from India, 4,017 rockets are launched from Indian soil. Out of that, almost 70% belongs to India and 30% for other countries for their studies. And in the area of satellites, till today, 134 satellites. The first satellite was built using the support of other countries, USSR, and exactly 50 years back, April 19, the Aryabhatta satellite was launched in the year 1975. From that till today, 134 satellites of various types and capabilities we have conceived, built, and launched. And in the area of launch vehicles, we have conceived and built 6 generations of launches. The first launcher was done in the year 1979. The project director was none other than APJ Abdul Kalam ji, the people's president. But unfortunately, that was an unsuccessful mission. And then the second mission was made successful in the year 1980. This year, after 41 years of service, I took over as the chairman ISRO Secretary Department of Space on 13th January. And 29th January was yet another important day for India. We had the 100th launch, and we were very particular to get successful launch. And I am happy to announce and inform all of you that the 100th launch of India was a perfect, successful launch, and we have written in the golden letters of Indian space history, the 100th launch was the successful launch. And a country, at that point of time, what was the vehicle we were using for moving our rockets? There were no automobile vehicles. We had only old bicycles. So our rocket parts used to be moved in old bicycles for launch. And one of the first satellite, communication satellite, Apple, was moved in Bangalore for a test in Villacart. And you can Google and find out those photographs are all actual photographs. From that area, today, we have accomplished 433 satellites of 34 countries. 433 satellites are launched from India using our own launches. That is the type of accomplishment. And during these years, 10 places we are number 1 in the world. Chandrayaan 1, finding water molecule on the moon. India along with USA is proud to be the first 2 countries finding water molecule on the moon. And Chandrayaan 2, you all may be knowing, some of you may be knowing, who is having the best camera in the moon's orbit? The best camera available in the moon's orbit belongs to India, a 32 centimeter resolution on-orbit high-resolution camera, and the best photographs coming from moons belongs to India today, and we are number 1 in that place. Mars orbiter mission, we have to travel almost 68 crore kilometer, and 68 crore kilometer, and after traveling, after 285 days, we have to restart the engine and we have to capture our orbiter. And dear friends, I want to proudly say that India is the only country and first country successfully accomplished a Mars orbiter mission in the first attempt. And we are very proud that we are the first country accomplishing. And it was Russia which placed the first man-made satellite, the Sputnik, in the year 1957. And through a single rocket, they have placed 37 satellites. That was the world record at that point of time. We all debated in Thiruvananthapuram one day. We were thinking that we all are so many gold medalists, so many toppers from Indian universities, we are working. But then how to get number 1 position? Finally, a decision was taken. We will launch 100 satellites using a single rocket. And launching is very easy, but it should not hit each other, collide each other. But today, dear friends, this history, India is the first country which placed more than 100 satellites, that is 104 satellites in a single rocket, and we made that history. And cryogenic propulsion, a propulsion which was denied in the early 90s. In fact, I was a student from IIT Kharagpur, completed my post-graduation cryogenic engineering in the year 1989 with first rank and joined back in ISRO. And at that point of time only the cryogenic technology was denied. Today when I look back, I really thank the people who denied the technology to us. And to our credit, 3 propulsion systems we have indigenously developed, and we are one among 6 countries having that technology. And to study sun, till today only 4 countries have built the satellites. And we are one among 4 countries which has built the satellite called Aditya L1, and it is giving fantastic results. Almost 20 terabit data it has come, and we have placed in front of the scientific community. 23rd August 2023 was again a historical day and a proud moment for all the 140 crore citizens of Bharat. India, the first country, successfully soft landed near South Pole of Moon, and we made every Indian proud, and we integrated the every Indians across the globe. And this year, lot of accomplishments are made. One of the accomplishments, please understand, when you talk about docking. Imagine in Dehradun road, two cars are going with 100 km/h speed and without hitting each other, without stopping one vehicle, both vehicles in the moving, you have to connect and mechanically connect each other. You can imagine what type of difficulty it is. This year, two satellites rotating with 28,000 km/h in space, we have successfully connected, rather docked, using a set of cameras, algorithms, and hardware. And India is the fourth country which has accomplished this fleet this year. And we are a proud nation. This technology is going to be used for a lot of things. And today, we are having 56 space assets, satellites in the orbit. You know, during the recent Sintur operation, almost 2 dozens of our satellites perfectly worked to ensure safety and security of the citizens of Bharat and ensuring successful operation of the Sintur operation. Now we are going in a very big way, because when you look at 50 years back, there was no space application in the country. Today there are 50 space applications. And without the satellite technology, I cannot think about a country with so much applications. Telecommunication, television broadcasting, telemedicine, disaster warning and mitigation, weather forecasting, ensuring food security, contributing food security, contributing in water security, contributing the health management and scientific missions, human spaceflight mission. So much activities are taking place. In fact, let me explain one application. If I have to talk about application, this is not the forum to talk about application. You know, telecommunication. 40 years back, if you were to make a phone call from Dehradun to Delhi, I think elders will understand what was involved. Somebody has to go to a trunk booth and wait at least 1 hour, and then only you will get the connectivity. And today, that is not the system. And every one of you have got mobile connectivity, and any country, you can get connected at any point of time. Television broadcasting. You know, you all will agree with me. There was a time 35 years back when I was a student in IIT. And there used to be a program called Chitrakar. And every week, 5 songs used to be played. And if you miss that songs, you have to wait for a week. But today, you see that situation. Like that, connecting real-time train and Navy applications, informing 9 lakh fishermen every day where they will get the potential fishery zones. Like that, so much application. Weather forecasting, disaster mitigation, lot of applications are there. Now coming to microgravity related research in the area of biology, just we have put our luck inside. In fact, I want to say this year we had a space microbiology experiment through our POEM payload. After the PSLV C60 vehicle, which was accomplished on December 24th, we put the upper stage. We did a couple of life science microbiology experiments, and we demonstrated the plant experiments with spinach and cowpea, tested their survival and growth in the space condition. This was the first biological experiment what India accomplished in the space. And you all know Axiom 4 program. Axiom 4, the Subhasnu Shukla, the first Indian to be sent to space, International Space Station, and the second Indian sent to space and first Indian sent to the International Space Station. There too, before going to the experiments, let me explain one aspect. In fact, he was sent by Falcon rocket on 25th June, and originally the launch was scheduled on 11th June. And 10th morning, June morning, we came to know there was a problem, leakage in the rocket. And it was Indians who went ahead and ensured the launch was called off. And after calling off the launch, when they inspected the rocket, there was a crack in one of the major feed lines. And the entire thing was corrected, and finally the rocket successfully lifted off on 25th June. But for our intervention, but for the Indians' intervention, this would have been a catastrophic failure, and we have ensured and we have proved to the world we are second to none, even in holding a human spaceflight rocket in the last minute to ensure the safety of the 4 astronauts to be sent to, who were sent to space. Now, Subhasnu Shukla, when he has come back, we have carried out 4 biological experiments in the International Space Station. One is the seed germination experiment. In fact, lot of seeds were taken, exposed to microbiological conditions, and it is now brought back because there are understanding that when the seeds are exposed to microgravity conditions, and when you bring back and the yield is going up. This is some of the literature source. So we want to experiment and understand, and if it is required, we have to put in application. And right now, the seeds are exposed to microgravity environment, and it is brought back. Further experiments are going on. Another important experiment: 2 strains of cyanobacteria to analyze their adaptability and how they might be used to recycle the human waste, produce food, and generate oxygen. This study will be useful in developing sustainable bioregenerative life support system for space travel. And cyanobacteria are also capable of nitrogen fixation and photosynthesis. Another experiment what Department of Biotechnology did was microalgae experiment. This experiment also has given very important results. And IIC Bangalore carried out another experiment, Voyager tardigrades. This experiment was carried out. And when you go through the literatures, other spacefaring countries like America or China, I think they have done lot of experiments are carried out. Huge literature is available. In fact, one can go through the literatures to understand what are all the important aspects they have studied. Even International Space Station, people have done lot of experiments. And I happened to see in NASA, when I was there, something in 2014 time frame, they were growing lot of plants under microgravity experiments, under microgravity conditions. These are all very, very exciting and important experiments. And when you talk about Bio E3 policy of Government of India, where we talk about biotechnology for economy, environment, and employment, the policy of Indian government, which was approved in August 2024, aimed at fostering high-performance biomanufacturing by integrating biotechnology with engineering and digitization. Its goals are to accelerate economic growth, improve environmental sustainability, and generate employment by focusing on key sectors. Dear friends, there is lot of opportunities for biomedical area, pharmacy areas, and medicine development, and microbiology area, biology area to carry out experiment in space. Today, we are working on building a Indian space station called Bharati Antariksha Station. The space station is a 52-ton space station which will be made in toto by 2035. And it is having, basically it consists of 5 modules. And the first module, right now design is completed, realization is going on, and we are going to place it by 2028. Almost third quarter it is going to be placed. We are going to announce opportunities for carrying out my experiments in multiple areas. And this community has got lot of opportunities. You can think and come back. Not only that, when the full space station is completely going to be constructed, we have to carry out lot of experiments. And your area is one of the very potential area we are considering. And we are going to have the human spaceflight program. In fact, the first crewed mission, we are targeting sending our own brothers using our own rocket in the year 2027. And towards that, we are working. And 3 uncrewed missions are planned this year and next year, followed by the crewed mission in 2027. And based on the direction and guideline from Honorable Prime Minister of India, we are also working on crewed mission to Moon. By 2040, we are going to land Indians on Moon using Indian launcher. In fact, the first launcher, whatever we realized and developed and succeeded in 1980, the capability of the vehicle was just 35 kg to low Earth orbit. That is the satellite what it can carry. Now this vehicle, what we are conceiving now, has to lift 80,000 kg to low Earth orbit. That is the type of capability what is required. And the rocket is a 40-story building type rocket. That is the type of capability what we are building. A country which started with bicycle and bullock cart era, today we have grown to greater levels. By 2040, Indian space program will be in par with any other space program in the world in terms of launcher, satellite, and application, and the ground infrastructure. Along with that, I am sure during that time frame, we will be carrying out lot of experiments in manufacturing, in material science, in biology, in biomedical area. So much experiments would be carried out. And this audience who are all here, I understand lot of students are there. Of course, you are yet to select your field, and there are a lot of professionals who are there already working in the biology area, and you have got a great role to play to think very smart way, carry out literature survey. You have to spend a lot of time in carrying out understanding what is done in the world and coming out with proper proposals. So we are there to handhold you to carry out experiments on almost free of cost, both in the space station as well as in the poem experiments. With that, I am extremely happy, and compliments are to the Dev Bhoomi Uttarakhand University, and the education minister himself is there. That shows the enthusiasm of the government, which is supporting this type of conferences to make scientific comfort among the youngsters, also for exchange of outstanding ideas among all the faculties and student community. And once again, let me thank the organizers for giving me the opportunity to address. Also apologize not participating directly. Whenever I come to Dehradun, I assure you I will visit your university and at the earliest opportunity. Thank you. Jai Hind.
So we are deeply honored to have you as a chief guest for today's valedictory ceremony. On the behalf of organizing committee, we share a sincere gratitude and respect for your presence, and definitely we look forward to see you once at Dev Bhoomi Uttarakhand University. Once again, sir, thank you so much for joining us online.
Thank you.
Now I request the dignitaries to please join us on dais, please. The guest of honor, Professor R.C. Kohat, the Vice President of the University, Mr. Raman Bansal, the Vice Chancellor of the University, Professor Ajay Kumar, the President AMI, Professor Prince Sharma, President-elect, Professor A.K. Punia, sir, and the General Secretary AMI, and, Dr. Namita Singh, and the Organizing Secretary, Professor Nabil Ahmed. I request everyone to please join us on dais. Audience, can we have a huge applause for all the dignitaries being joining us on dais.
With your permission, I have to go to airport. If you permit, I think I will leave. I will disconnect and leave. Please go continue.
Certainly, sir. Yes, sir.
Thank you. Thank you. Anyway, we'll meet you in your university.
Sure, sir. Sure. Thank you so much, sir. Now it is a time to honor to introduce. I request Namita ma'am to please join us on dais, please. Now it is a time to introduce the Vice Chancellor of our university, Professor Dr. Ajay Kumar. Professor Ajay Kumar is a visionary academician leader and inspiring administrator who has been instrumental in guiding Dev Bhoomi Uttarakhand University towards excellence in education, research and innovation. Under his leadership, the university has flourished as a hub of academic vibrance, industry collaborations and holistic development. With this, I request Sir to kindly present the address for this gathering, please.
Good afternoon, everyone. Honorable Chief Guest of the Day, Dr. V. Narayanji, ISRO Chairman, was connected online. Honorable Minister of Health and Education, Uttarakhand, Shri Dhan Singh Rawat ji, Honorable Guest of Honor, Chairman AMC, Professor R.C. Quad sir, Honorable Vice President of this university, Shri Aman Mansal ji, Honorable President of AMI, Professor Prince Sharma ji, Dr. Namita Madam, General Secretary of AMI, Dr. Anil Kumar Punia ji, Director ICR National Research Center on Camel, Organizing Secretary Dr. Nabil ji, our Assistant Vice President Nancy Madam, our Pro-Vice Chancellors, Deans, head of departments, scientists, researchers, participants, and my dear students of this university and from other universities. Dear all, finally we have reached on the 4th day of the conference and we are organizing the valedictory function. On this occasion, our ISRO Vice Chairman was connected online. So I really a proud moment for each and everyone. On the behalf of entire Dev Bhoomi family and my own behalf, I welcome the ISRO Chairman to the valedictory function. And on the behalf of the Dev Bhoomi family, I welcome the guest of honor, Kohad sir, Professor Prince sir, our Vice President, General Secretary to the valedictory function at this university. Truly, it's a historic moment and will be entered in the history of the university. These 4 days were utilized smartly. If I say this smartly, naturally, each and every session was very productive. In a very well manner, I hope the participants, researchers, academicians, and the scientists are really enjoyed and utilize these days effectively. So big hands for each and everyone. Sure, the outcomes of the conference will really help the beginners and other researchers to pick up the latest research areas on which they can start something in terms of research. So that is the beauty of the conference, because not only it was the conference, but it was the research conclave. It was the complete deliberations and the churning on the latest topics. So it was done wonderfully. In the conference, which I have taken the data, approximately 700 numbers as per the participants, speakers, and the co-chair and chair of various sessions across India and abroad were participated across renowned IITs, NITs, and from the various renowned universities in India and abroad. So it was the great part because the Dev Bhoomi Uttarakhand University witness for these academicians and these stalwarts. Apart from this, around 2,000 students from our university, they participated in various sessions because in parallel of this Pandal sessions, many more 4 sessions were there. So our students, they were participating in the Pandal also, main Pandal also, and the different seminar halls and other venues. I really congratulate the team of AMI, President Sir, General Secretary, and our team for this great success and for this great show of the AMI conference at this university. As the Vice Chancellor of Dev Bhoomi Uttarakhand University, I once again extend my sincere thanks for everyone for coming and attending the conference. I shall request to everyone, please carry positive things from here and utilize the outcomes of the conference for your research areas as an input and a name and fame in the field of microbiology. With these words, I wish you all the best. Jai Hind, Jai Bharat, DBU.
Thank you, sir, for your kind words. Moving forward, it is now my privilege to introduce the Vice President of our university, Mr. Aman Bansal. Mr. Bansal is a dynamic and visionary leader whose forward-thinking approach has been a pivotal in shaping Dev Bhoomi Uttarakhand University growth and global outlook. His constant encouragement and commitment towards fostering innovation, entrepreneurship, and quality education have greatly contributed to the university's continued success and excellence. So we are truly honored to have you present at the valedictory ceremony of 66th AMI Annual International Conference and Research Conclave. Ladies and gentlemen, please join me in welcoming Mr. Aman Bansal, the Honorable Vice President of Dev Bhoomi Uttarakhand University.
Good afternoon to everyone present here. First of all, again, that I quoted in the inauguration ceremony, it gives me a profound pride that AMI chose Dev Bhoomi Uttarakhand University as the first ever private university or institution in the country to host their 66th AMI International Conference Research Conclave. I would like to thank our chief guest for today's occasion, the chairman of ISRO, that from a very busy schedule, he gave a confirmation of attending the conference physically and gracing the occasion and blessing all of us. In the morning, we got a message by him that he has an emergency meeting at the PMO, because of which he could not turn up. But then still, he joined us online and blessed us, all of us, with his profound knowledge about ISRO, about the industry, microbiology, and biotechnology. Thank you, sir. I would like to again thank our chairman of AMI, Dr. R.C. Kohad, sir, the guest of honor for today's ceremony. I'm sure, sir, on this, in these 4 days, it was very tiring, but we could not see that tiredness on your face at all in these 4 days. Over the panel discussions, your talks, it was mesmerizing, sir. Thank you. The president of AMI, Dr. Prince Sharma, thank you very much, sir, for believing in us. And again, it was a very tireless job, but we could not see any kind of, tiring or any kind of exhaustion on your face at all. Dr. Namita Singh, the General Secretary, the woman behind the event, the planning, the documentation, inviting the speakers, from everything to anything. From 5:30 in the morning to almost 12 o'clock at night, she was on her toes. Ma'am, we salute to your energy and to your courage. Dr. A.K. Punia, sir, congratulations on becoming the president for the AMI, the president-elect. Thank you very much, sir. And at last, the people who actually chose microbiology as their domain of expertise. Over the 4 days, over 3 days of conference, sessions, seminars, panel discussions, what was not clear on 12th is very much clear today on 15th, in just 3 days of time. We think that research only happens on paper. But in these 3 days, a very significant and very clear of, a total clear of thought came to everyone's mind, I'm sure, that any research in microbiology, biotech or healthcare cannot happen on papers. It's a real life research happening on ground. So any paper which comes into existence is totally after day and night research happening in labs, experimentation happening. The people of pharmacy, healthcare, allied sciences, kudos to you, kudos to your thought that you chose this domain. And imagine that the dignitaries sitting on the dais, they chose this domain almost 20-25 years ago. That means they knew the importance of this domain back then when people were not even talking about this expertise and this domain at all. When supercomputers and computers came into existence, people were talking about technology. And those were the times when these people, when the scientists sitting in front of us were talking about and thinking about microbiology. I'm sure, sir, with everyone, all the delegates, all the keynote speakers, all the scientists who have come from their respective universities, institutions and places across the country, you must have encountered many questions, and I'm sure you were troubled by the students, by the delegates, by the audience a lot with their inquisitive questions. I'm sure you have sufficed them to 100% of whatever they had in their mind. Ultimately, there is a saying that confusion is the first step to understanding. So our teacher in school, the math teacher, used to say this. And today, we actually realize it. जब तक confusion नहीं होगा दिमाग में, you will not ask. And till the time you will not ask, there will be lack of understanding of the concept. Over these 3 days, so I was always there attending the session through the live technology, what we have. We came across tremendous discussions, tremendous questions being asked by the audience, to the panel members, to the keynote speakers, to the presenters. And I do not think that the students mainly would get this kind of platform anywhere. I would like to tell the students and encourage them that if you have any conference coming up in your vicinity, it's an urge, go and attend it. Whatever you will learn in classroom is just through books. Probably you will be taken to your labs for experimentation. But whatever you will learn in a conference, in a research conclave, will be a finding of a scientist, will be a finding of an expert who has given his or her life to that one domain to expertise in it. So probably whatever you are thinking that you will become 25 years from now, that gap can be bridged to at least 15 years. Because whatever you think that this knowledge I have to acquire, you will get it right now instead of getting it 5 years from now. The scientists you heard over the 3 days are renowned ones who came from government institutions, government agencies, who have their own name in their industry. Probably many students, many people sitting in the hall today would not even meet them ever because they are unavailable. Getting them here is a very, very tedious job. And everyone, we at Dev Bhoomi Uttarakhand University, we feel lucky that we heard them. I'm sure people have learned from them. You will not get this kind of, platform, this kind of occasion anywhere else. After this, you will be indulged in your corporate jobs, in your livelihoods. You will not get time to do any kind of research. But trust me, research is the root of any invention, anything which is coming in the world. It will come from a research. The gap analysis and the solution to it, the research is very much important and evident in all our lives. The escorts who were made of the guests, if you have not asked questions from them about their industry, about your own industry, that's a huge loss you guys have made. You were with them for 24 hours for 3 days. A too learned person was just next to you and just a call away because the numbers were shared to every one of you. And the scientists, the teachers, whoever we have, they have one quality of not saying no. To whatever questions you would have asked, they wouldn't have said, कि बेटा time नहीं है अभी या नहीं. They must have said that this is not the right time, we will explain it to you maybe later in the evening. But they will never say that मुझे नहीं आता या मैं नहीं बताऊंगा. If you guys have asked them questions, if you guys have encountered your problems to them, I'm sure you guys have learned, and if not, ultimately it's your loss. The people who came in over the 3 days, they are well established. No one in the industry, no one in the country has that platform or that identification that they had. So they do not need you. It is us who needed them. So I'm sure everyone took the best out of it. And trust me, the research and the technology has gone. The new era will be of healthcare and microbiology and biotech, food technology, all the eras of allied sciences. Like in the inauguration, and everyone talks. Gone are those days when we used to say that the veto power, the veto country is the country with a good army. Now the veto power is the country with best healthcare infrastructure and facilities. Because ये जो healthcare war है ना, ये बहुत बहुत आगे है. What you cannot, any one of us cannot even think of it. Just a passing of a virus or a microbiome and the country is devastated and finished. Never, no one even thought of it. And no one can fight in the court of law against it. Because microbe का, virus का, organism का control किसी आदमी के पास नहीं है. So who will you actually fight upon with? Which country? So we have to be very much attentive that we do not encounter or we do not get any infectious virus to our households, to our homes. And parallelly, tremendous amount of research has to happen to make the country, our own country, India, best for the infrastructure of microbiology in terms of research and domain expertise. After all of this, true I'm too much thankful to all the people, the scientists, the teachers, the delegates, everyone who came. Arrangements, the team is there. Invite karna my responsibility duty hai. The dignitaries of AMI will come because it is an occasion of AMI, but it was a grand success when we got almost 200 people in the campus talking about different topics, talking about different panel discussions about their research work. Their contribution and their presence in the organization made this conclave a success. Thank you to everyone from bottom of my heart, and I'm sure we will be hosting similar kind of conferences in near future. And the people of microbiology domain, kindly register yourselves as live members. Go attend this conference whenever and wherever it is happening. Because learning की कोई सीमा नहीं है. जितना आप सीखोगे, उतना आपका कैरेक्टर, उतनी आपकी नॉलेज बढ़ेगी. When you talk amongst people, they will not see how you are dressed or what you are looking like. They will be actually noticing what you know, what you have in yourself. Thank you very much again. Thank you.
Thank you, sir, for sharing insightful thoughts with us. It is a time to introduce Professor Prince Sharma, the President of Association of Microbiologists of India. Professor Prince Sharma is a renowned microbiologist and a visionary leader who has significantly contributed to advancing microbiological research and education in India. Under his guidance, AMI has continued to promote scientific excellence, foster collaborations between academia and industry, encourages the young scientists to pursue innovations in microbiology. Sir, your insights and the experience are valuable to all the present here. We are eagerly look forward to see your address as a valedictory ceremony at the 66th AMI International Conference and Research Conclave. Ladies and gentlemen, please join me while welcoming our AMI President, Professor Prince Sharma.
Good afternoon, everyone. I can see so many people enthusiastically listening to all the dignitaries here. And we have just heard ISRO chairperson elaborating the country's progress, success in space research, in satellites, and how he has explained it so beautifully that how India is poised to be number one when it comes to the space technology, the satellite technology. I listened to the ISRO chairperson and I was filled with such pride that my country, India, is taking so much leap in the space research. Not only so, he has also added the dimensions of biology and microbiology and biotechnology to the space research also, if you have heard carefully. As we have been telling you since day one, microbes are everywhere. And I have been telling you that before we came here, maybe not many people were knowing about the microbes and the microbiology, but after we leave, I think everyone will be knowing what microbiology is and what are microbes and their power. He has very deftly elaborated that microbes, cyanobacteria, research on cyanobacteria, the plants, biotechnological importance of the space research. And just a few hours ago, we have been discussing BioE3, the BioE3 policy of our country, where one of the verticals is the space and marine research, where India wants to take leaps, where India wants to go ahead in the space research. And now you see that space biotechnology, and when I say biotechnology, I've been telling you microbial biotechnology as integral and the very most important part of biotechnology. So in one of the pillars here, which is the space biotechnology, it is getting fulfilled here. My dear friends, This has been a 4-day research conclave where various talks have been held. Again, when I am talking about the 6 pillars of Bio-E3 policy of India, I think we have tried to cover each and every vertical in this particular conference, whether it's biomanufacturing, whether it's industry, whether it's agriculture, medical, marine biology. We have tried to cover all the aspects of biotechnology which India is including in the Bio-E3 policy. But the story doesn't end here. AMI this particular time had thought that research talks, let us not just restrict this particular conference to the talks only or to the research work only. And for the first time, we have included the next gen microbiology education programs also in this. For the first time, I was talking to the Vice President, Mr. Aman, that for the first time in this particular conference, we have added these 4 panel discussions also, which have never been done before. And in collaboration with Dev Bhoomi University, I think these have got a big success. And this is a start only. This is a start only. We had panel discussions on the next gen microbiological education. We had on the creating enablers. Very importantly, we had the panel discussions on including the industrialists and the startup to have the startup culture in India. And today we had on the Bio-E3 policy. What else do you want? All the students. What else you want besides having, as Amanji has said, that besides having about 200 speakers from various fields, not just the speakers, but the experts in their own areas, in their own fields, who have come from far and wide and have talked to you. I think not everyone, I can say, is listening but there were some students who were listening to them very avidly and were asking questions also. My only surmise is that in all the talks, the hall should have been filled this much as it is today. In future, make the examples, it should as much filled in all the panel discussions, when speakers come. For a speaker, you know that the most important thing when the hall is filled. That's the most satisfying thing for him, because he has come from far. He's talking to you. He's wanting that, take my expertise, learn from this. So in these 4 days of conference, which we have, AMI has taken in this particular region, for the first time or maybe after a very, very long time, it has been a, we can say, synergistic relation between Dev Bhoomi and between the Association of Microbiologists of India. That's why it has become a great success also, a grandeur success. I believe that in future also, you will be having many such conferences where the talks will be more and more on the panel discussions, more and more on the, as we have been discussing, on the internships, besides being the talks on by the speakers, because now the world is moving away from theory, it's more moving towards the labs. It is more towards to get the training. And I am urging since day one to all of you, get into the research as much as possible, get into the internships as much as possible, get into the contacts with startups and industry as much as possible. That is the future. Get into the newer technologies, include newer technologies into your syllabi like AI. This is the future. This is the future, because otherwise, I understand Indian students, they have a lot of potential. When they go to US, when they go to different countries, they do excel. Best of the positions. they are held outside also by Indian students. Why not here? Indian government is very much wanting through all these Bio-E3 like programs that there should be development of a skill which can take this country to Atmanirbharta, to make in India. In these times of trade tariffs, India is direly wanting to make it in India, make itself, rather than importing, rather than depending on other countries. And who will do this? Who will do this if through the next gen education, if through proper education, through proper research programs, through proper trainings, you have the skills to do that. You have the abilities to go for startups towards the industry. So in these 4 days programs, we have tried to inculcate in you the microbiological next generation education temper, microbiological sciences research, the startup and industry research, the internship programs, culture, as much as possible we could do, because one of the mandate of AMI is not just the conferences, but to develop the scientific temper, not just microbiological, but scientific temper among the students. And we try as much as to develop this temper. In the Bio-E3, Bio-E3 panel discussion just a few hours ago, I have been telling that go for internships. You can apply as many people, they can give it to the organizers. I'm ready to host one student in my lab for 1 month to 6 months, as much he wants, to have the flavor of the recent technologies in my lab. And so I will also urge other scientists also, if they can have at least one student and they can give the internship and the expertise, they can learn the expertise in our lab. They're open to that. So Amanji, you can sponsor any one of the students to my lab. I will be freely giving whatever knowledge I have, technological programs I have, I will give it to him or her. With this, as valedictory word says that it's a farewell. Vale means farewell. And in the valedictory speech, the last day has come, fortunately or unfortunately, that we don't want to leave this place, but we have to go. It is the farewell. So I wish all the organizers of this conference, the president and the vice president and the vice chancellor, and all the organizers, that they have taken care of us, they have organized it very well, and I wish all you students a very good and bright future. Thank you so much.
Thank you so much, sir. Now I request the Vice President of Dev Bhoomi Uttarakhand University, Mr. Aman Bansal, and the Vice Chancellor of the University, Professor Ajay Kumar, to kindly present a token of respect to the President of AMI, Professor Prince Sharma. The audience, can we have a round of applause for the President of AMI, Professor Prince Sharma. Thank you, sir. It is a time to introduce Professor A.K. Punia, the President-elect AMI. Professor A.K. Punia, President-elect of Association of Microbiologists of India, is a distinguished microbiologist and dairy scientist recognized nationally and internationally for his pioneering research work in the human microbiology, microbial biotechnology, and probiotics. Presently serving as the director at National Research Center on Camel in Rajasthan. As a mentor, innovator, and a scientific leader, Professor Punia continued to inspire young researchers while contributing to the development of Indian dairy and a microbial biotechnology sector. His association with AMI underscores his commitment promoting the microbiological sciences and fostering collaboration among academia, industry, and research institutions. Now I request Sir to kindly address the gathering on this valedictory ceremony.
Thank you very much. Though it was not scheduled, but I will take only few minutes only. I will be with you because there will be a lot of time. I will be keep on interacting with the students and other faculty and colleagues. First of all, very many thanks to the audience. In most of the conferences, wherever I have attended, there remains a very thin population, especially in valedictory section, as our Honorable President has already said. So, Honorable Kuhad Sahab, Honorable Prince Sharma Sahab, Aman Bansal ji, Vice Chancellor Sahab, Madam Namita ji, Nabil, Professor Pabbi Sahab, I can see Kamar Sahab, my senior Joshi Sahab and Vasudev Ji, and other colleagues, faculty and students of this very prestigious institute. The name itself indicates, जब आप इसका नाम भी लेते हैं तो आपको बड़ा पवित्र सा महसूस होता है, देव भूमि। एक मिठास सी आ जाती है बोलने में, ये शब्द इतना अच्छा है। आप लोगों ने बहुत अच्छा शब्द पिकअप किया है अपनी यूनिवर्सिटी के लिए। तो मैं कुछ चंद शब्द कहना चाहूँगा। मैं आज ही आया हूँ। आई कुड नॉट अटेंड द होल कॉन्फ्रेंस बिकॉज ऑफ माई अदर commitments elsewhere, but what I could learn that lot of good session and lot of interaction and lot of learned people from world over, they came over here and interacted with the young minds. I will say that challenge before these scientists and students, those who have opted for this subject, is that we are working on an organism which is not visible. It is very easier to work on plant and animals in comparison to microorganisms because they are visible and we can see their morphology with naked eye. But when we cannot see something, whether it is good or bad, because microorganisms are both, and we cannot think of surviving on this planet without the involvement of microorganism in our day-to-day life. So they affect us and infect us both. So they interact with the human beings in both way, negatively and positively also. It is the duty of or it is a very sagacious duty of all the scientists that we have to isolate and identify and screen those good microorganisms for human welfare, for global welfare. I used to call them as the small wonders of the world. ये बहुत ही वंडरफुल ऑर्गेनिज्म है जो बहुत तेज़ी से ग्रो भी करता है और बहुत तेज़ी से आपको लाभ भी देता है. आई विल टेल यू कपल ऑफ गुड थिंग अबाउट माइक्रो ऑर्गेनिज्म. दे नेवर टेक रेस्ट. When you incubate something in your laboratory in the evening and morning you come, you will find them in your in your flask or in your petri plate. So they are working day and night for you. All other organisms, whether it is animal, plants and any other, they take rest, including us. जैसे इस valedictory session के बाद भी हम सब लोग रेस्ट करेंगे और सो जाएंगे. So that is the beauty. Secondly, they are very secular also in nature. Irrespective of caste, creed and race, they will infect and affect you equally. They will not bother you whoever you are. Whether you are a senior person, junior person, poor person, rich person, they will affect you equally. So we have to learn from microorganisms how to see this world so equally. Then a couple of other things about the organizers and this AMI. It's a very beautiful, not only very beautiful campus. We came to this campus couple of months back and we decided that this 66th AMI is going to be organized over here. So the organizers have proved by inviting all of you and arranging everything, because it is a big program, so there may be some little bit tidbits and all. That is always possible when you arrange and make this thing. But arranging such a big program is also not an easy task. It is really an uphill task. We learn it when we really organize ourselves. So thank you very much, Amanji. And another indication I got, so Amanji, you are not a biologist even, I think. So 3-4 दिन में आप लोगों ने इनको इतनी माइक्रोबायोलॉजी सिखा दी है. He was speaking like as if he is also from biological sciences.
आपने देखा भी होगा उनकी स्पीच में, और उन्होंने भी सीख लिया है। तो क्रेडिट गोज टू ऑल माइक्रोबायोलॉजिस्ट सिटिंग ऑन द डायस एंड सिटिंग ऑफ द डायस। सभी लोगों के लिए ये मैं कहूँगा तालियाँ। और माइक्रो ऑर्गेनिजम्स इतने इम्पोर्टेंट हो चुके हैं, दे आर इंटरट्विन टू ईच इन आवर लाइफ। पहले हम बोलते थे यत्र नार्यस्तु पूज्यन्ते रमन्ते तत्र देवता, कि जहाँ जहाँ नारी है वहाँ देवता निवास करते हैं, लेकिन हमने इसको मॉडिफाई कर दिया है, यत्र माइक्रोब्स पूज्यन्ते रमन्ते तत्र देवता। तो जहाँ जहाँ माइक्रोब की पूजा होती है वहाँ पे भी देवता निवास करते हैं। मैं फूड साइंटिस्ट हूँ, तो फूड के बारे में भी एक दो चीज़ मैं आप लोगों को बोल देता हूँ कि देयर आर 3 डिफरेंट कैटेगरीज ऑफ माइक्रो ऑर्गेनिजम्स। One, which we do not want that they should come to our food arena or food environment, जिनको spoilage causing organism. Second category is they can infect us, जो बीमारी पैदा कर सकते हैं, and we call them as foodborne pathogens. And third one is the starter organism. Without them you cannot convert your one product into another. वो तीसरे हैं जिनको हम स्टार्टर कल्चर या फ्रेंडली माइक्रो ऑर्गेनिज्म, डायरेक्ट फेड माइक्रो या प्रोबायोटिक्स या दूसरी संज्ञा दे दें, नाम कितने भी हों लेकिन वो बहुत ही बेहतरी के लिए हैं। एंड विदाउट द इन्वॉल्वमेंट ऑफ दोज माइक्रो ऑर्गेनिज्म इट इज़ नॉट पॉसिबल टू कन्वर्ट वन फूड इंग्रेडिएंट इनटू अनदर। एक मैं आपको एग्जांपल देता हूँ, बच्चे लोग सुन रहे हैं कि नहीं? लास्ट में जोर से बोलो, वंडरफुल। I give you an example. You can never convert milk into curd by any other means, chemical and all. But these beautiful microorganisms, over the night, they work for you. आपकी मदर घर में या आपकी मिसेज या आपके बच्चे या कोई भी या खुद आप, you incubate them, you put them, आप बैक कल्चर, बैक स्लोपिंग कर देते हैं और अगले दिन you get a beautiful and human friendly or beneficial curd. And that gives you lot of benefits. It is not possible otherwise. This I have given you only one example because of the paucity of time, और मेरा scheduled भी नहीं था बोलना, तो मैं बीच में ऐसे ही आ गया, like a contaminant but beautiful contaminant. So जो लोग समझते हैं कि भाई कंटामिनेशन होता क्या है, और तो मैं एक अच्छा कंटामिनेंट साबित हुआ, तो आप लोगों की तालियां भी आई, माइक्रोब्स के लिए आई है, ऑफ कोर्स नॉट फॉर मी, क्योंकि दूसरी बातें। और मैं इन लोगों को, सब लोगों को, जो मेरे सब सीनियर लोग हैं जो सीट पे बैठे हुए हैं, इन लोगों से आई हैव लर्नड ए लॉट, सो आई बो माई हेड टू देम। उनको मैं नमस्कार भी करता हूँ, सैल्यूट भी करता हूँ। और आप सब लोगों को, यंग लोगों को, आई इन्वाइट यू so that you do good in microbiology, because this is the only field left where you can do wonders. And जैसे मैंने कहा कि माइक्रो ऑर्गेनिजम्स आर स्मॉल वंडर ऑफ द वर्ल्ड, यू आर द विजिबल वंडर्स ऑफ द वर्ल्ड। कल के दिन में आप लोग भी कोई ना कोई बहुत ही बेहतरीन काम कर पाएंगे। और जैसे हमारे प्रेसिडेंट साहब ने आप लोगों को इनवाइट किया है एक बच्चे के लिए, मैं एक से ज़्यादा को इनवाइट करता हूँ, जितने भी आप आ सकते हैं उतने आ जाओ हमारे यहाँ। और ऑलरेडी जो हैं हमारे पास जो हैं इतने इंटर्न आते हैं, और हमने सबके लिए अपना गेट खोल दिया है। हमारे यहाँ इको टूरिज़्म भी है, आपको जब भी मन करे तो टूरिज़्म भी करने आएँ, दूसरे भी करें, MOU भी करें हमारे साथ, हमारी लेबोरेटरीज़ में रिसर्च भी करें। इतना वाइडली ओपन हमारा इंस्टीट्यूट है, वंडरफुल। मेरे को तो शब्द भी नहीं मिल रहे कि किस तरह से मैं आप लोगों को आमंत्रित करूं। और सभी लोगों को भी और आप लोगों को भी बहुत बहुत धन्यवाद भी, साधुवाद भी वाइस चांसलर साहब को, और जो इसके मालिक हैं जो आज हैं नहीं, बंसल साहब, तो वो उनको भी धन्यवाद। मैडम नमिता जी को आपने कहा कि ये इन्होंने भी बहुत पेन लिया, लगातार ये आपको लिखती पढ़ती रही, सबसे संपर्क किया। फिर हमारे वाइस चांसलर साहब ने, प्रिंस शर्मा जी ने, वो ही टुक लॉट ऑफ पेन। तो हमारे अग्रज भी हैं, वो बड़े भाई भी हैं, और कुहाड़ साहब जो हमारे लिए प्रेरणा के स्रोत हैं। फिर बंसल जी जो नए नए मित्र हैं, मेरे अच्छे मित्र राजे, और नबील, और वो जो लड़की है निर्जरा, उसके लिए भी एक बार तालियां कर दें, वो भी बहुत ये कर रही हैं। और बाकी लोग जिनका मैं नाम नहीं ले पाया, मेरे यहाँ 2-3 स्कॉलर भी आए हुए हैं, और बाकी सब आप लोग, बेस्ट विशेस टू ऑल ऑफ यू, थैंक यू वेरी मच।
It was truly an honor to have the opportunity to work for your labs, for our students. It was truly been commendable, sir. Thank you so much for that. Now I request once again to the Vice President of Dev Bhoomi University, Mr. Raman Bansal, and the Vice Chancellor of our university, Professor Ajay Kumar, to present a token of respect and gratitude to Professor A.K. Punia, the President-elect AMI. Audience, can we have a huge applause for the President-elect Professor A.K. Punia for AMI. Students telling you what you dream you would be having, that what he has given a fulfill to you now. Thank you, everyone. Is it a time to listen from our guest of honor? It is now my honor to introduce Dr. R.C. Kohat, the Chairman of Association of Microbiologists of Indian Sciences Council, AMSC, who is working as a Vice Chancellor at Swami Vivekananda University, Assam. Dr. Kohat is an eminent scientist and academic leader with a vast experience in microbial biotechnology, environmental biotechnology, and research administration. His visionary guidance has been instrumental in shaping policies, promoting scientific research, and fostering collaboration between academia, industry, and government. So we are privileged to have your presence today to look forward for your inspiring thoughts and guidance on this valedictory ceremony. And I mentioned it is Dev Bhoomi, so that's in these 4 days, he bring me that level that yes, we can a part of Dev Bhoomi University. Please, एक बार जोरदार तालियां हो for Kohat sir, who's always mentioned, ताली की आवाज रुकनी नहीं चाहिए.
Thank you very much. But I could see only 27% of the audience has clapped so far.
Dev Bhoomi, show your level of the clap at the back side.
ये आप पे डिपेंड करता है कि जब मैं बोलना बंद करूं कि क्या आप 100% मार्क्स ले सकते हैं, ये आप पे डिपेंड करता है. Ladies and gentlemen, it's a mixed occasion, or occasion of a mixed feeling, you can say, when you are coming to the closure of any program, जिसे विदाई का समय कहते हैं, उसके लिए आप और हम यहाँ पे एकत्रित हुए हैं. ये बातचीत आप लोगों ने सुनी, वार्तालाप सुना कि क्या कुछ इन 4 दिनों में हुआ है, वो आप सबके सामने प्रस्तुत किया गया है. मैं अभी भी देख रहा हूँ कि कुछ हमारे यंग लोग ही नहीं, हमारे जो जवान कलीग हैं, वो अपने मोबाइल पे कुछ एक दूसरे को दिखा रहे हैं. ये तो आप 1 घंटे बाद जो है ना खूब कर सकते हैं. जैसे मैंने 12 तारीख को कहा था, तो अपने अपने मोबाइल जरा अपने अपनी जेब में रख लीजिए. ओके? कितनी अच्छी मुस्कुराहट आपके चेहरे पे जो दिखाई देती है, तो यहाँ बोलने वाले को भी अच्छा लगता है. और जब आप मायूस बैठते हैं तो ये पता लगता है कि मजबूरी में आपको यहाँ बांध के रख रखा है. तो ऐसा नहीं होना चाहिए, क्योंकि श्रद्धावान लभते ज्ञानम. तो अगर आपकी श्रद्धा नहीं है तो कुछ नहीं मिलने वाला. और अभी हमारे एक साथी ने आपको बताया
कि देखिए जो हमेशा आपके साथ रहता है, अगर उसकी परवाह आप नहीं करेंगे तो आप नुकसान में रहेंगे। आपको बताया था मैंने, आप लोग ज़्यादा नहीं थे, कुछ और नए लोग आए हैं आज, कि ये जितने भी माइक्रो ऑर्गेनिजम्स की बात हमारे प्रेसिडेंट साहब ने बताई, हमारे आने वाले प्रेसिडेंट ने बताई, हमारे इसरो के चेयरमैन साहब ने भी कुछ स्पेस माइक्रो ऑर्गेनिजम्स की बात की, को छोड़ेंगे आपके साथ और कोई नहीं रहेगा, ये सिर्फ माइक्रो ऑर्गेनिजम्स रहेंगे। और जो आपका ऐसा साथी है उसके बारे में आप समय नहीं निकालेंगे, श्रद्धा नहीं रखेंगे, तो क्या होगा? चलिए, as tradition, like respected chairman from ISRO, our Honorable President Association Microbiologists of India, Vice President Dr. Aman Bansal ji from Dev Bhoomi Uttarakhand University, Dr. Ajay Kumar, Vice Chancellor, Dev Bhoomi Uttarakhand University, my dear Professor Amita Singh, General Secretary of Association Microbiologists of India, Dr. Nabil, Organizing Secretary, Dr. Nirzara as well, my all colleagues, those who are sitting in front of me, their family members, deans, directors of this beautiful university, which is taking lead in transforming education, not only in Uttarakhand, but within the country, because there are many students. I heard that you have students almost from the 20 or 22 different states. Persons from media and press, dear students, ladies and gentlemen. ये विदाई का समय है, कुछ उदास आप भी हैं, कुछ उदास हम भी हैं। बिछड़ते हैं मिलने के लिए, तो अगर हमने 4 दिन खुशी से बिताए हैं तो हम भी ये याद रखेंगे कि आपने कितना अच्छी तरह से, कितना भव्य आयोजन करके और हमें खुश रखा है। आगे भी जब मिलेंगे तो इसी खुशी के साथ मिलेंगे, और यही एक मानव को ध्यान में रखना चाहिए कि ऐसे तो बिछड़ो, ऐसी अवस्था में तो बिछड़ो कि कहीं रेलवे प्लेटफार्म पर मिल जाओ तो एक दूसरे को हेलो तो कह पाओ। तो आई थिंक वी आर इन ए पोजीशन टू डिपार्ट with the same feeling. Dear friends, it's very true that in these 4 days, or maybe one meeting we had earlier with the Vice President and President of this university, and of course during the course of arrangement of this organization, or this conference, 66th Conference of Association of Microbiologists of India, Dr. Aman has come across the microbiology as a discipline, but he said one thing, one word which I could catch: confusion. And having confusion is not a weakness. This is the major strength, if you ask me. If you will have confusion, hello, students, देखिए, Dev Bhoomi University is spending not less than 50,000 for my presence here. And if you don't listen me, then what's the use? I'm not telling you about the kind of technological work and the researches are going on. I'm not a great scientist like Professor Prince Sharma and Professor Abbi or say my other friends. Professor Om Prakash is here. I'm a student like you. I know about your problem and that I would be discussing. Confusion takes you to development of the curiosity. And if this confusion can make you to think, to be curious, to find out the solution, then you will have the zeal to question somebody. Question your teacher, question your mentor, question your father, question your mother, or your friends. And curiosity leads to invention. More the invention, you will lead to or bring out more and more researches. Therefore, having curiosity is the first step to make you a person with scientific temperament. Once you have scientific temperament, you will be able to develop passion. And once passion is developed, then you will be a great scientist. Therefore, dear students, it's very, very important to listen. When you can listen, again I will say, connect with me. Okay? Connect with me. With curiosity, you will learn. When you will learn, you will acquire. Why I'm saying that? Even Dalai Lama says that when one speaks what he or she knows. But when you listen, you learn more. You can experience yourself, you feel yourself. Therefore, for students, it's really, really very, very important. And I said in the beginning that the conference, which has focused on a specific theme, that next generation microbiology, education, research, innovation, for energy, economy, and environmental sustainability, is immediately accurate and highly beneficial and informative. And I'm sure those who have visited the posters specifically, the research students of your age, they came out their work. I could get an opportunity to visit, say, some of them, not all of them. And it's really, they have carried out the wonderful work. And I think the way we used to present the posters during our research time, when we were young, they have really transformed the newer ways, modern techniques they have used, and even their presentation techniques, presentation ways, presentation methods have been improved. They are very, very clear. I was very happy to see the progress. Dear friends, in this conference, we have also emphasized, and I think in the history of the science in this country, this association has taken a note of that we need to work more or focus more on education part as well instead of only research. And that's what we have talked about, the next generation education and how next generation education needs to be organized, need to be developed so that we can align with National Education Policy 2020 or vice versa. That how we can really make a plan, execute our national education policy, keeping in mind that we would be able to provide and using all kind of technology which are used to transform your ongoing education, match it with the next generation education. What are the requirements of the next generation? We have worked on that. We have been able to develop almost 60 different courses for UG and PG microbiology. The complete curriculum has been developed, which will be a unique and novel type of, say, the curriculum in the time to come. And here I would like to request the Vice President of this university that by March we'll be able to complete that course, and I think this should be the first university to use that course for your undergraduate and postgraduate courses, so that will be a great, I think, encouragement to the Association of Microbiologists of India, because that will be a first success story, say, for our association academia as well. Dear friends, as you have been briefed by our President, certainly Association of Microbiologists of India and Academy of Microbiology has taken a decision that we will be providing platform for assisting or facilitating, say, our students, UG and PG students, for getting the internship in different institutions. We will be creating the data, say, required for that in terms of the institutions, in terms of the supervisors. And we will be developing a platform for that, and I think that way we'll be able to help you. Of course, on individual basis, as Professor Anil Punia, Professor Prince Sharma, they have offered. Likewise, my all colleagues who are sitting here, they are ready to accept, if not more, at least 5 on average, the interns. So that sort of information you will have on our website, and you can make use of that. We are here. Our objective is that how we really can popularize microbiological sciences among masses, because I don't have any hesitation in saying that it's everybody's science. It's not, don't think of that, okay, I'm a man of political science. Fine, you be a political science graduate or postgraduate, but you are a microbiologist as well. You are a man of microbiology. Microbiology is with you. You are not with microorganisms. Microorganisms are with you. Therefore, it's a kind of discipline which you should love. If you start loving, you are having the pets at home, your home. You are domesticating them. Okay? You need to be friendly with these microorganisms. You need to learn how to domesticate them. You have to take care of them. If you take care of them, meaning by, if you take, say, nutritionally rich food, specifically from the plant origin, you will see that your microbiota, the microorganisms in your gut, will be much stronger. And if your microbiota is, say, the stronger, you won't be ill. Take these words from me. I can give you guarantee that microbiota of this gut can take care of your all kind of organs, their functions, their physiology. They produce so many compounds which take care of even, say, controlling the cancer kind of the disease, diabetes as well. But you have to make your microbiota, gut biota, rich. Even some kind of the capsules, they are available in the market, which people use in order to enrich the microbiota. Because you might be hearing that take a fibrous diet. So how this fibrous diet helps you getting the better nutrients? There are anaerobic bacteria, specifically the methane producer bacteria, which act on this fiber and produce more energy. And of course, that helps in movement of your balls. That's a different thing. Because more fiber doesn't help in digestion, but helps in energizing your body. This is the way that microorganisms, the morning you get up, what you eat, throughout the day and night, every product, you'll find that there is a intervention or involvement of microorganisms. So microorganisms are so important, and all aspects of microbiology have been studied here. Dear friends, I'm very sure those who are from microbiology, they might have been benefited. And I'm very much pleased to say that the way Dev Bhoomi team has organized this conference, I salute them, I congratulate them, and I thank them on behalf of my all colleagues sitting on the dais and colleagues sitting, say, in front of me, off the dais you can say, and we are going with a very good experience. Bye-bye. Thank you very much. Walaikum salam. Soinara.
Thank you, sir, and I'm sure we'll look forward to see you in future once again at Dev Bhoomi Uttarakhand University. Now I request once again to the Vice President of Dev Bhoomi Uttarakhand University, Mr. Aman Bansal, and the Vice Chancellor of our university, Professor Ajay Kumar, to present a token of gratitude and respect to our guest of honor, Professor R.C. Kohad, sir. Audience, 100% clap has to be there. Students, I'll tell you, he's just not an eminent scientist, but he's a great motivator, what we have seen in last 4 days. Thank you, sir. It is a moment to introduce Professor Namita Singh, the General Secretary of the Association of Microbiologists of India, AMI. Professor Singh is an accomplished microbiologist and a dynamic academic leader and has been actively involved in organizing and coordinating the 66th Annual International Conference and Research Conclave. Her efforts has assumed the smooth conduct of various sessions, workshops, panel discussions, and collaborative dialogues throughout the event. Her insight has always reflected the contribution of the speaker, participants, and organizing team has made this conference success. Now I request ma'am to share her insightful thoughts with us on this valedictory ceremony.
Good afternoon to everybody. Honorable dignitaries at the dais and off the dais. Esteemed speakers, respected faculty members, and dear participants. Being the General Secretary of this 87-year-old prestigious association, Association of Microbiologists of India, it is my privilege and honor to extend a heartfelt vote of thanks on behalf of the Association of Microbiologists of India and the Academy of Microbiological Sciences to the Dev Bhoomi University, Uttarakhand, organized from 12th to 15th November entitled Next Generation Microbiology Education, Innovation and Research for Economy, Energy and Environment. First and foremost, I extend my sincere gratitude to our distinguished Chief Guest, Dr. V. Narayan, Director of ISRO. Chief Guest of inaugural function, Dr. Ranjit Kumar Sinha, Secretary, Department of Higher Education, Government of Uttarakhand, for gracing this event with his invaluable insights into the globalization of higher education. His word has truly inspired us to think beyond the conventional academic boundaries. A special thanks to Professor Prince Sharma, our President of AMI, for his unwavering support and encouragement in organizing this conference. I would also like to express my appreciation to Professor R.C. Kohat, Chairman AMSC, for his warm welcome address, for hosting this workshop with great enthusiasm, his dedication and commitment to academic excellence and commitment for the education. A big thanks to President-elect Professor Anil Kumar Punia, Professor S.S. Dudeja, Professor Sunil Pabbi, Professor Ruplal, Dr. S.S. Kawar, Professor Lily Ganju, Professor Dr. Meenakshi Prasad, and other CC member, Dr. Vasudevan, Dr. Joshi, and other present here participants, as well as our EC, CC, and life members of association. A special thanks to Organizing Secretary, Professor Nabil Ahmed, and the Joint Secretary, Dr. Neeraja Singhvi, and their team for their dedicated efforts in making this conference a great success. We are thankful to the Vice President and the President of the Dev Bhoomi University, who provided a very good environment. I would say Dr. Punia already explained regarding the Deva Bhoomi itself is a very holy place where we are organizing this academic activity. As we know, that's total, the organization, we organize 2 different workshops during the conference. That is hands-on bioinformatics workshop for genomic analysis and patent writing, to creating a hands-on experience, and then it was supported by the UCOST. So I thankful the UCOST for the gracious support, DBT, DST, NIRF, as well as the DRDO for giving the financial support to organize the successful conference of AMI. This conference includes total 11 themes, which are microbes for transforming waste to wealth, metabolic and molecular frontiers in microbial biotechnology, microbial manufacturing, Make in India, AI in microbial research, bioprocess and bioeconomy, current and future trend in antimicrobial resistance, the promise of vaccine, microbes for the restoration of degraded environment, computational microbiology, data science and digital health, microbes in agriculture and crop improvement, microbes in traditional and contemporary food products, next generation microbiology, exposing a school student to a promising career in the field of microbiology. In this conference, a global gathering of over the 400 national and international speakers and the participants throughout the globe from USA, UK, Japan, South Korea, Kuwait, Uzbekistan and China. The total dialogue talks delivered by the prominent researchers from across the globe, including 5 dialogue talks, 3 panel discussions, 14 keynote talks, 34 invited talks, 23 invited power talks, a special lecture in the memory of Dr. K.R. Dadarwal Memorial Award, industrial talk, 6 Young Scientist Award talks, 46 faculty oral talks, 75 students oral talks, and total of 267 poster presentation during all the 4 days. The conference also included a panel discussion on Circle of the Science Bench to Business, the startup journey. On the occasion of Children's Day, a special session was organized for a school student from class 8 to 12 to showcase their microbial literacy and create awareness about microbes. One of the conference theme was also dedicated to this activity on 14th November 2025. Through this initiative, the school students experienced the fascinating world of microbe, and the activity encouraged them to consider microbiology as a promising career. I extend my gratitude to the organizing committee, technical staff, and supporting team, and their behind-the-scene efforts in ensuring the seamless execution of this event. Without their hard work, this conference would not have been possible. Finally, our sincere thanks to Dev Bhoomi University Uttarakhand, providing excellence venue and infrastructure to host this significant activity of AMI. As we conclude this conference, let us take forward the knowledge shared, the discussion held, and collaborations formed enhance the microbial literacy and education with the National Education Policy 2020. Let this be stepping stone towards a greater academic excellence and innovation. With this word, I thank all the participants and organizing secretaries and the staff of Dev Bhoomi University. And then finally, I request one of my students who can come, and we will announce today all the awards from AMI side. May I?
Yes, ma'am. You can proceed for that.
Before going for the award ceremony, we can honor our partner of the
Ma'am, before you would like to give a session of gratitude, on the university behalf, I request to our Assistant Vice President, Mrs. Nancy Bansal, and the Vice President of the university, Mr. Aman Bansal, to please share a token of respect and gratitude to the General Secretary of AMI, Professor Namita Singh, ma'am. Jaldi do. My dear audience, can we have a huge applause for the General Secretary of this AMI, Professor Namita Singh, who was rigorously been there for organizing this 66th AMI International Conference and Research Conclave. Thank you so much, ma'am.
From our side, I request our President, Chairman, and the President-elect to honor the Vice President of Dev Bhoomi University. Shawl and
It is a time where the AMI would like to share a token of respect and gratitude to the Vice President of our university, Mr. Aman Bansal. My dear audience, can we have a huge applause for the Vice President of our university, Mr. Aman Bansal.
Dr. Now the Vice Chancellor of Dev Bhoomi University, Dr. Ajay Kumar. Sir, please अभी आगे रहिए. अब आप पीछे नहीं जाइएगा. आप लोग.
It is a time to honor and felicitate a token of gratitude to the Vice Chancellor of our university, Professor Ajay Kumar.
Dr. Nabil.
It is a time to felicitate the organizing secretary of this 66th AMI Annual International Conference and Research Conclave and the Dean Research at Dev Bhoomi Uttarakhand University, Professor Nabil Ahmed. Audience, can we have a huge applause, a man of a success who brings this conference alive. I request Nirjara ma'am to please join us on stage. It is a time and honor to give a token of respect and gratitude to Dr. Nirjara, a joint secretary for this conference, 66 AMI International Conference and Research Conclave, a lady who was standing 24/7 for these 4 days and bringing this conference to a success.
Dr. Shubhang Walia.
I'll announce for my own self. Thank you so much for giving me this respect and gratitude, and I love to have such conferences in future at Dev Bhoomi Uttarakhand University.
ताली बजाइए आप लोग.
It is an honor, sir. Thank you, sir. Thank you so much.
सर नहीं प्रेसिडेंट का सर उनका ऑफिस में देंगे, अमन सर को देते हैं, डॉक्टर अमन को दे देते हैं, प्रेसिडेंट सर का. प्रेसिडेंट सर का ऑर्डर.
Now I would like to invite our Assistant Vice President, Mrs. Nancy Bansal on dais. Audience, to take the honor and respect of gratitude on the behalf of our President of the University, Shri Sanjay Bansal. Audience, can we have a huge applause, a man who brings this conference to this organization, and thank you to the Dev Bhoomi Uttarakhand University for hosting this international conference. A woman behind the success, truly said by Professor Prince Sharma, the President AMI.
Time to announce the award. Okay, other than this, I want to we selected or the collected 11 innovation ideas. Okay, so all the idea belongs to the different fields. So we will organize one meeting and select one from each field, and that will be announced and then informed to individual later on. So as we know that in all fields we are awarding the different fields of microbiology with the poster session award. So in the field of agriculture, Bhavana M Agriculture, IIRI New Delhi. Bhavana M, IIRI New Delhi. She's not present.
Is she here?
Yes. Vijay Lakshmi Medical, Vijay Lakshmi, my medical field, King's George Medical College, Lucknow. She's here?
No, ma'am.
Sakshi Dogra, Environmental Sciences, CSIR IHBT Palampur. CSIR IHBT Palampur. Is she here? No.
No, ma'am. No, ma'am.
Sushovan Bhattacharya, Molecular Biology, Birla Institute of Science and Technology.
Ma'am, somebody is coming for his award.
Sushobhana, yes. Sushobhana ka nikalna.
Audience, can we have a huge applause for this that she presented a conference research paper here.
Certificate, we will provide the 2,500 cash prize that we will transfer online after collecting information from you. Yes.
She's been honored with a 2,500 cash prize for this success.
Nishu Yadav, Industrial Microbiology. Nishu Yadav, Industrial Microbiology. Huh? J.C. Bose University, Delhi. Delhi? See?
Not here, ma'am.
Okay. Varsha More, Food. Varsha More, Food. Bhabha Atomic Research Center. Varsha More, she also left. Anshika Birla, who?
Ma'am, somebody is coming, I think, on her behalf.
Yes. Your name? Varsha More. Girl or boy? Okay. Okay, you can. Varsha More.
The volunteers standing at the back, please ensure all the research people who are here, if they are standing outside, tell them to please be inside for the valedictory.
AMI Springer Award, Anshika Bila, Punjab University.
She is here, ma'am. Can we have a huge applause?
Anshika Bila. Priyanka, University or Allahabad. तो एक बार टिक करते जाना किस किसका चला गया. प्रियंका, यूनिवर्सिटी ऑफ इलाहाबाद. डॉक्टर रंजना भाटी, बुंदेलखंड यूनिवर्सिटी. शीतल ठाकुर, IIT दिल्ली. रागनी ढोले, सेंट्रल यूनिवर्सिटी ऑफ राजस्थान. भारती दत्तन, MDU रोहतक, हरियाणा. सुसाद खलीज, ICAR. स्वाद.
Ma'am, somebody is coming.
श्रेया माहेश्वरी. भावना मनराल, देवभूमि यूनिवर्सिटी. भावना मनराल.
Yes, she is coming, ma'am.
सरिता, पंजाब यूनिवर्सिटी, चंडीगढ़. राणा मेमोरियल, ऊपर जाके देखो कौन है?
This
राणा
clap should
मेमोरियल
not
डॉक्टर
stop.
रेणु सिंह, झांसी वर्मा मालविका सहगल, यूनिवर्सिटी ऑफ जम्मू, time to announce the oral presentation award from AMI Springer with the 300 euro book prize. Nishchal Thakur, ICR NDRI Karnal. Vatsal Bhagwan, University of Delhi. Pooja Mahadev Rao Sonwane, Government Institute of Sciences. Sonal Gupta, Department of Zoology, University of Delhi. Dolamani, ICAR IRI. Vivek Kumar Gaur, MET University Noida. Koli Bhavya, ICR Indian Agriculture Research Institute. गवर्नमेंट हायर सेकेंडरी स्कूल क्या हो गया? स्कूल का बच्चा तुमने उसका नहीं हो रहा है? भोपाल यूनिवर्सिटी वाले, भोपाल यूनिवर्सिटी वाले एस मलार विजी, गवर्नमेंट हायर सेकेंडरी स्कूल अनाथन। स्कूल वाले का कैसे हो गया? स्कूल नहीं है। शुभम अग्रवाल, IIT रूड़की। दिव्या वेणुगोपाल, ग्राफिक एरा यूनिवर्सिटी देहरादून। प्रणामी भारद्वाज, इंस्टीट्यूट ऑफ एडवांस स्टडी इन साइंस एंड टेक्नोलॉजी। मिस नेहा, पंजाब यूनिवर्सिटी। अच्छा, you are working there, na? Okay, no problem. You can. So she is in a school teaching. Okay, no problem. इनको यहाँ से देते जाओ। हाँ? रागिनी गोथलवाल। Fine. In the end, I congratulate all the winners.
Can we have a huge round of applause from both the sides? I request the dignitaries to please stay back.
देखिए भाई, बैठने का कोई फ़ायदा नहीं। देखिए, किसी को अवार्ड मिलता है तो जो नीचे बैठे हैं उनको खुशी होनी चाहिए और परमात्मा से प्रार्थना करनी चाहिए कि जब मैं वहाँ जाऊँगा मुझे अवार्ड मिलेगा, और जिसको अवार्ड मिला वो बैठा होगा तो वो ताली बजाएगा। तो ये बड़ा ज़रूरी है। ये टीम हमारे प्रेसिडेंट AMI की है और ये वाइस प्रेसिडेंट देव भूमि यूनिवर्सिटी उत्तराखंड की है। जो जीतेगा उसको 500 रुपये का इनाम। हाँ, ये हुई बात। अब हम याद रखेंगे कि देवभूमि के जवान बच्चों में कुछ ताकत है। आइए, कौन बचा अभी? और शायद हम अपने यंग दोस्त को ये सिखा चुके हैं अभी कि आगे से ये छोड़ेंगे नहीं आपको जब तक ताली नहीं बजाओगे।
Students, I'm telling you, he's just not an eminent scientist, he's a great motivator. So can we have a huge applause, Professor R.C. Kohad.
आपके जो आसपास में ग्रे हेयर वाले बैठे हैं, आप देखोगे वो धीरे से बजा रहा है। ये ग्रे हेयर होते इसलिए हैं कि धीरे धीरे बजाते हो। जितना तेज बजाओगे बाल काले नहीं होंगे जिंदगी भर।
Thank you, my dear audience. Now I request Joint Organizing Secretary Dr. Nirjara to please join us on stage and share some more inciting awards by ASCS and ASM. Over to you, ma'am.
Thank you, Shubhang sir. So moving on with the award ceremony, congratulations to winners of the AMI award, and I'm here to announce two more segments of awards. The first award is sponsored by ACS, which is American Chemical Society. They have sponsored 6 Best Poster Awards, and the winners are May I request our honorable dignitaries to kindly felicitate the President-elect of AMI, Professor A.K. Punia.
Congratulations, sir.
Thank you, sir. Moving on with the awards. Awards by ACS for the best posters. The first award goes to Priyanjali Tripathi from University of Allahabad. They have been awarded with a certificate and a cash prize of Rs 3,000 each. Priyanjali. The second award goes to Nancy from Pondicherry University. The third award goes to Ms. Priyanka Singh, Dev Bhoomi Uttarakhand University. The fourth award by ACS goes to Sambit Kumar, Bharampur University. Congratulations to the winner. Can I have a huge round of applause for our winners? Thank you. The next award goes to Dharmender Kumar, CPRI, Shimla. Mr. Dharmender Kumar, CPRI, Shimla. The last ACS award goes to Ms. Apurva Singh, Dev Bhoomi Uttarakhand University. I am re-announcing the awards. The first award was for Priyanjali Tripathi, University of Allahabad, Nancy, Pondicherry University, Priyanka Singh, Dev Bhoomi Uttarakhand University, Sambit Kumar, Bharampur University, Apurva Singh, Dev Bhoomi Uttarakhand University, and Dharmender Kumar, CPRI, Shimla. Once again, a huge round of applause for our winners of the ACS Best Poster Awards at the 66th AMI Annual International Conference. The next set of awards are by ASM, which is the American Society for Microbiology, and the society has given 1 year of free membership of ASM along with access to its journal and e-books worth $200 for each winner. And the winners are Dr. K. Veena Gayatri from Stella Maris College. This is for the best oral presentation. These 6 awards by ASM are for the best oral presentation during the oral sessions of the faculty and student sessions in our conference. The next award goes to Dr. Darshana from BARC, Baba Atomic Research Center. We have a student from Dr. Darshana's lab who will receive the award. The next award is Neha Hatwar, Niri, Nagpur. I still cannot hear the clapping going on. It should not stop tonight. The next winner is Harish GS from IIT Roorkee.
Sir.
Our next winner is Dr. Vijay Lakshmi. The winners are being awarded with a 1 year membership of ASM with access to all its journals and $200 e-books. The last award under the category goes to Ms. Neha Rawat from Dev Bhoomi Uttarakhand University. Vijaya, announce कर दिया मैंने? Vijaya Lakshmi. ये रही Vijaya. Can I have a huge round of applause, please? Once again, congratulations to all the winners who have received the ACS and ASM awards at the 66th AMI Annual International Conference. I request Mr. Shubhang Walia to kindly take over with the conference proceedings. Oh yes, we have 2 more awards. We announced a pitching round in between on the second day of the conference, and 2 of the students who have won award for the best pitch are Yashvi Rajput and Parvez Alam. They have pitched their startup under the mentorship of Mr. Shubhang Walia. Can I have Yashvi Rajput and Parvez Alam on stage, please? And I request our honorable dignitaries to kindly felicitate the winners of the pitching round. Congratulations and thank you so much. Over to Shubhang Walia.
Thank you, ma'am. It is a moment and a privilege to invite Professor Dr. Nabil Ahmed, the Organizing Secretary of 66 AMI International Conference, and to deliver a vote of thanks. So I request to share a vote of thanks to this eminent audience, please.
खुदी को कर बुलंद इतना कि हर तकदीर से पहले खुदा बंदे से पूछे बता तेरी रज़ा क्या है. A very good evening to one and all present here. Honorable Chief Guest V. Kumar, Guest of Honor, Founding Chairman AMSC Professor R.C. Kuhad, Vice President Dev Bhoomi Uttarakhand University Mr. Aman Bansal, President AMI Professor Prince Sharma, Vice Chancellor Dev Bhoomi Uttarakhand University Professor Ajay Kumar, President-elect Dr. Anil Kumar Punia, General Secretary AMI Dr. Namita Singh, Assistant Vice President Ms. Nancy Bansal, respected dignitaries on and off the dais, distinguished member of AMI delegates, speaker, and my dear colleagues. It's hard to believe that the wonderful journey that was started since 7th month past is coming to an end. So I would like to just give you a simple story where it just came from. I just received a message in the month of Feb 2025 from Honorable President Sir, Mr. Sanjay Bansal, and I think today our dream has been turned into reality. A huge round of applause for our Honorable President Sir, Mr. Sanjay Bansal ji. On the behalf of organizing committee, I take this moment to express our deepest gratitude to all distinguished guests, speakers, delegates, and participants. Your presence and active engagement have made this conference truly memorable. A special words of appreciation goes to the Association of Microbiologists of India for its continued leadership in nurturing scientific thoughts, collaboration, and innovation across institution and generation. Behind the success of this conference lies the tireless work of many invisible hands. The organizing committee, faculty coordinators, student volunteers, and shadow and technical staff work round the clock to ensure every detail was executed to perfection, from scientific sessions to the hospitality, logistic, and cultural program. And event success lies in a team work, and can't be more thank you to have a work with the best organizing committee who have no stone unturned to cater this 4 days event with 80 plus speaker and 500 plus participant. I would like to begin by thanking a hidden face whose support is invisible, Mr. Arjun Singh. Next in line in our very supportive digital and website team under Mr. Rahul Bhatt for meticulously planning and promoting the conference to every possible level. Mr. Ajay Kumar and Mr. Anuj for maintaining the website and registration portal 24/7. Digital team Shivani, Amit, Udit, Mohit, Asif, Mehak for supporting the conference in digital and print. A loud applause for the accommodation committee under Mr. Iqbal for arranging every last minute room. Team transportation under Mr. Sachin for ensuring smooth pickup drop facility. Mr. Paramjeet, MTO Pankaj, Tawfiq, who I believe have not spelled from past 4 days to ensure every delegate is settled well in the hotels. A huge round of applause for the Team IT under Yugalji for setting up entire IIT setup for a smooth conduction of the session in online or offline mode, from providing Wi-Fi facility in the campus to setting up the room for the meeting as well as printing purpose. Admission team for helping in all aspects. Every event is remembered for its food, and under the team Dr. Molly and Mr. Surinder, the food was not nothing less than a master chef support for the serving most delicious food. A special thanks to our venue coordination committee and Mr. Shubhang Walia for managing timely conduction of the session. Regards for Dr. Digvijay and Mr. Bhupendra for untiring and endless support in all logistics. A special thanks to Dr. Sandeep Sharma, Dr. Subhashish, and Mr. Sunny Verma for support and guidance. No event would have been completed without the next few names: Meenakshi ma'am, Ramesh ji and his team, Raghav ji and his team, Abhishek ji and his team for arranging everything so perfectly on time for every session for past 4 days. We are equally grateful to our funding agencies supported by the DBT, ANRF, DRDO, CSIR and UCOS, sponsors, collaborators and partner organization, High Media, Thermo Scientific, AppDos, Gale, IGL, Aster India, Global MedKit, Lab Technologies, ZSI, LN, American Society of the Microbiologists, Microbiological Society of the London, KIT Bhubaneswar, joining us as the knowledge partners, whose support was invaluable in making this event possible. Your encouragement and cooperation strengthened the bond between the academia, industry, and research institution and open new avenues for meaningful collaboration. So on the behalf of the entire organizing team, I extend my heartfelt thanks to each participant, your time, enthusiasm, and scholarly spirit. We wish you all the continued success in your research and academic pursuits and a safe journey back home with pleasant memories of these inspiring days. I would like to end with the famous quote with one of most famous Allama Iqbal. वो कहते हैं, गिरते हैं शहसवार मैदान जंग में वही, वो तिफल क्या गिरे जो गिरे घुटनों के बल. इसका मतलब ये है, जो काम करेगा गिरेगा, जो घुटनों के बल चलेगा वो कभी गिरेगा नहीं. So thank you all once again for making 66 AMI Annual International Conference a great success. Jai Hind, Jai Bharat, Jai DBUU.
Thank you so much, sir. Now I request everyone to kindly stand at their respective places. Okay, before everyone stands, ma'am, I have some announcements, please.
Okay. On behalf of the Association of Microbiologists of India, I invite all of you for the next 67th Annual International Conference, and very soon we will announce the place where we are going to organize 67th AMI Conference.
Thank you, ma'am. Now I request our Assistant Vice President, ma'am, to please join us on stage just for a minute, and I request everyone to give a standing ovation to our Vice President and Assistant Vice President on their wedding anniversary today. Can we have a huge round of applause for this beautiful celebration that we are doing all together today? Sir, मुझे तो मैं सर ने बोला, I've done it. Guys, can we have a huge round of applause and can I give a lot of blessings to both of them on their wedding anniversary? Thank you. I request everyone to stand on their respective places for the national anthem.
Thank you.
everyone. The photographs, e-certificates and souvenir would be available on the website of this conference, so all the participants can download out from there. And I request the e-certificate can also be downloaded from there. And I request all the dignitaries to please join us on downstairs for a group photograph. I request the authorities at the backside kindly make the students waited and the participants kindly wait for a group photograph, then may you proceed. We would like to have a photograph, then you may proceed. Nirjara ma'am, I request you to please join. Thank you so much. Thank you everyone for being a part of this 66th AMI Annual International Conference in Research Conclave. And I'm thankful to the entire AMI team and Dev Bhoomi team for this organizing a beautiful conference for the people. Thank you.